these VMs and other things. So all these things together, and then once everything is verified and structured deployment is using some of that, and is able to create the resource under the resource pool and authenticate it. So this is how you can actually manage
Michael. Hello, sir.
Morning. Hello.
So we'll start in another 5 minutes.
Okay.
So I would like to welcome you all back to the AMI International Conference. I hope you all had a great lunch. So we'll now have a plenary talk by Professor Andreas Bechtold, and I would request Professor Roop Lal to formally welcome him.
So, while want to welcome you to Andreas to this international conference organized under the aegis of Association of Microbiologists of India and also INSCAR, I want to thank you for the efforts and time that you have spared in order to be here. And many of my colleagues and students, they are very well familiar with your work and the ideas that you have been giving to our group, and your association really helped us a lot. So before I really uh, get into your lecture, I would now pass on the mic to Nirjara, who will formally introduce you. But once again, thank you very much.
Thank you.
Thank you, sir. To chair this session, we have Professor Rooplal Terry and Professor Yogendra Singh, University of Delhi. Professor Andreas Bechtold is presently the head of the Institute of Pharmaceutical Sciences at University of Freiburg, the Department of Pharmaceutical Biology and Biotechnology. Major areas of his research are aimed at production of new therapeutic agents by combinatorial biosynthesis. Other project deals with the investigation of the mechanism leading to antibiotic activity of these oligosaccharides and characterization of the mechanism leading to the antibiotic resistance. He has received several scholarships and honors, notably from German Research Foundation, Japan Society for Promotion of Science. He has also been awarded the Phoenix Pharmacy Science Award in 2008. May I now request Professor Andreas to enlighten us with the BLDA and Cyclic Di-GMP, Natural Product Regulators Special. Over to you, sir.
Thank you very much for the invitation. I prepared a PowerPoint presentation with my language included, and now I'm starting to start this.
Yes, sir.
Hello, everybody. My name is Andreas
Sir,
Bechtold.
it's,
I'm a professor at the
I'm sorry to
University
interrupt you, sir. It's not visible. Please share the slide.
Sorry?
I want to thank the
So please share the slide.
organizer,
It's not
Professor
visible.
Lal.
Is it not working? 1 second. I must go back. So I have to.
Share screen, sir. The
Yes,
green
I did
button.
this. Is it now fine?
Not yet, sir. They want. Yes,
Now?
sir. It's coming.
Yes,
Yes,
now
sir.
we're running?
Yes, sir. Now run it. Sir.
Okay.
Hello, everybody. My name is Andreas Bechtold, and I'm a professor at the University of Freiburg, which is located in the southwest of Germany. I want to thank the organizer and especially Professor Dr. Rublal for the friendly invitation to give a talk on this conference. The title of my talk is BLDA and Cyclic DGMP, National Product Regulators Special. In the beginning of my talk, I would like to mention a few topics which are in relation to my talk. First, we are facing a corona pandemic, but we are facing for an even longer time an antibiotic resistant crisis. Each year, around 1 million people die because of antibiotic resistant bugs. Second, national products are very valuable. Most antibiotics on the market have been invented by nature. Third, Streptomyces are very talented gram-positive bacteria. The number of national product gene clusters in the strains is higher than the number of products produced by the strains. The challenge is to activate gene clusters which are silent under normal lab conditions to find novel antibiotics. The activation of silent gene clusters is the topic of my talk. As in all other organisms on Earth, in Streptomyces, DNA stores all the information. DNA is converted to mRNA by RNA polymerase, and the RNA is being used at the ribosome as template to generate proteins and enzymes. In Streptomyces, cyclic DGMP is involved in regulating transcription, and the tRNA BLDA controls the translation process. I want to start to speak about BLDA. The gene BLDA is coding for a unique tRNA able to translate an UUA codon into the amino acid leucine. UUA codons are a very rare property of GC-rich Streptomyces genomes. The impact of BLDA on morphology can in parts be attributed to the regulatory effect of BLDA on the translation level, because TTA containing genes can only be translated into their corresponding protein in the presence of a fully functioning BLDA gene. In addition to the visible effect of BLDA expression on the phenotype of, for example, Streptomyces cerlicolor, BLDA mutants were also deficient in antibiotic production. Our work on BLDA started a few years ago when David Zechel came on his sabbatical to my lab. He brought to Freiburg a strain named Streptomyces calvus. Streptomyces calvus, which has been described in 1956 as producer of nucleosidine, a potent translation inhibitor. But in the lab in Canada, the strain not only failed to produce aerial mycelium, but also failed to produce nucleosidine. David remembered a paper from David Hopwood and Keith Chater, who described, also a few years ago, the life cycle of Streptomyces strains. The life cycle starts with the germination of spores, which grow out to form a branched network of vegetative hyphae. Under environmentally adverse conditions such as nutrient depletion, streptomycetes initiate a complex developmental program whereby the vegetative mycelium serves as a substrate for a new so-called aerial mycelium. This stage of development usually corresponds with the production of secondary metabolites such as antibiotics. A BLDA mutant of Streptomyces cerlicolor resembles Streptomyces calvus, as it also is not able to generate aerial mycelium. David Zechel came up with the hypothesis that Streptomyces calvus might have a mutation in the BLDA gene, and he decided to sequence the BLDA gene of the strain. As you see on this slide, a mutant was detected in the BLDA gene, which most probably resulted in the formation of a non-functional tRNA molecule, shown in B. When we complemented the strain with a functional BLDA gene, the strain started to generate aerial mycelium, which is shown in D. It was now important to investigate whether the strain is now able to produce nucleosidine. As shown by fluorine NMR, nucleosidine production was restored in the complemented strain, shown in B, as you see here on the slide. When we started to investigate Streptomyces calvus in more detail, we found the strain lacking BLDA was able to produce the national product WS9326A. But when we investigated Streptomyces calvus containing a functional BLDA gene, beside WS9326A, two additional compounds, which we named Animycin A and B, were produced. From this result, we learned that BLDA is not controlling biosynthesis of all compounds, but there are compounds like nucleosidine or Animycin A and B, which cannot be produced without BLDA. We then ordered a couple of strains which have been described in the literature as strains which do not generate aerial mycelium easily. BLDA was expressed in these strains, and in more than 50% novel compounds were generated, here highlighted in yellow. We started to isolate compounds and performed structure elucidation. For example, compound 11 is a streptophenanthine which exhibits moderate antibacterial activity. Peak number 15 consists of two compounds, glomeromycin A and B. And compound 17 is a curatazole which exhibits potent cytotoxic activity against AC2116 and HOS cancer cells. Actually, we recently cloned the curatazole gene cluster. The compound is a ribosomal made national product with an interesting biosynthesis. As a summary of the first part of my talk, I can say that BLDA is a very useful tool which can be used to wake up biosynthetic gene clusters. I'm coming now to the second part of my talk. I would like to speak about cyclic D-GMP, which you can see here on my slide. It is known that cyclic D-GMP is a global bacterial second messenger that controls a wide range of cellular processes that contribute to surface adaptation, biofilm formation, cell cycle progression, and virulence. Here you see the principles of cyclic D-GMP signaling. The CD-GMP monomer exhibits two-fold symmetry with two GMP moieties that are fused by a 5'3' macrocyclic ring. The synthesis of CD-GMP is catalyzed from GTP. It can be hydrolyzed into PGPG or GMP respectively. Conversion of GTP to CD-GMP is catalyzed by degranulated cyclases, DGCs, through the cooperative action of their two catalytic GGDEF domains. Specific phosphodiesterases, PDEs, that contain EAL domains hydrolyze CD-GMP into PGPG or GMP, respectively. CD-GMP is a key effector molecule in bacteria. In Streptomyces follicular, the transcriptional regulator BLD controls the expression of ADPA, BLDA, as well as dozens of other development-related genes. Interestingly, the regulatory action of BLDD is controlled by CD-GMP. In the presence of CD-GMP, a dimeric BLDD complex bound to 4 CD-GMP molecules is formed, which then proceeds to bind to target promoter sites. In Streptomyces follicular, this binding results in vegetative growth. If BLDD is not able to bind to DNA, sporulation is observed. Before I continue to speak about CD-GMP, I want to introduce our model strain for this project. It is Streptomyces ghanaensis, the producer of moenomycin. Moenomycins are the only known national inhibitors of peptidoglucan glycosyltransferases, enzymes involved in cell wall biosynthesis. Moenomycins show a broad spectrum of action against pathogenic bacteria, including vancomycin-resistant Enterococci and methicillin-resistant Staphylococcus aureus. In Streptomyces ghanaensis, moenomycin biosynthesis is controlled by a couple of regulatory proteins which are not encoded by genes located in the gene cluster. The pleiotropic regulators ADPA and BLDA are involved in direct and multi-layer control over moenomycin production, whereas another protein, EPSP, limits ADPA abundance via ribonucleolytic activity. BLDD is controlling expression of ADPA and BLDA, and we were able to show that deletion of BLDD led to a mutant which is not able to produce moenomycin at all. Our aim was now to find genes controlling the formation of CD-GMP in order to control BLDD function. 7 genes were detected in the genome of the strain, encoding proteins belonging either to the phosphodiesterases with EAL domains or to the cyclases with GGDEF domains. In 4 cases, both domains were identified. In the beginning of this project, we focused our efforts to investigate the function of RDMB. Deletion of RDMB led to a mutant showing increased production of moenomycin. Complementation of RDMB restored moenomycin production. We were able to show that transcription of genes of the moenomycin cluster is increased in the RDMB mutant, shown here for the structural gene MoE-E5. We expressed the gene in E. coli and checked the protein for activity. It did not show cyclase activity, that are not shown, but the enzyme was able to convert CD-GMP to PGPG as shown in this slide. This RDMB is acting as phosphodiesterase, reducing the level of CD-GMP in the cell. All 7 genes have been investigated in a very similar way. Moenomycin production was mainly increased in mutants in which a phosphodiesterase gene was deleted, and in mutants in which a cyclase gene was overexpressed. There's only one gene, RDMA, which seems to encode both the phosphodiesterase and the cyclase. But now let's come back to RDMB. I've told you that moenomycin production was increased in an RDMB mutant. When we analyzed the strain for the formation of further compounds, we found 2 new peaks using HPLC MS analysis. The structure of one compound was elucidated as oxohydrodidine, a compound with antifungal activity. The second compound accumulated in vehicles formed on the surface of the strain. This compound was shown to be deferrioxamine, which is an iron chelator which is used as drug for the treatment of iron overload. So to summarize my talk, BLDA and genes involved in cDGMP formation and hydrolysis are useful tools to wake up biosynthetic gene clusters, and these genes can also be treated for the overproduction of compounds. At the end of my talk, I want to thank my co-workers for a wonderful job in the lab. Especially I want to thank Olya, Daisy and Roman, here indicated with a red point. I want to thank Professor Dr. David Zechel and Professor Dr. Thomas Paulolat, as well as Professor Dr. Oliver Einsle and Dr. Sonja Chang, who also were involved in this project. Money came from the Deutsche Forschungsgemeinschaft. We are members of two RTGs, the RTG 1976 and the RTG 2202. And at the very end, I would like to thank you for your attention.
Thank you so much, sir.
I can't hear. I have no.
Just a second, sir.
Okay.
Sorry for the disturbance, a bit of technical glitch. Thank you so much for this amazing talk. Now we'll take few questions. I would request Professor Yogendra Singh, he want to say something. Sir, over to you.
Yeah, Andrea, this was a great talk showing that BLDA regulates synthesis of many molecules. And BLDA is controlled by the cyclic di-GMP, which binds and allows the transcription of many genes. And cyclic di-GMP is controlled by the phosphodiesterase. What controls phosphodiesterases? Because their synthesis is going to control the amount of cyclic di-GMP. So question is, what regulates phosphodiesterases?
Sorry, the question is what?
How the amount of phosphodiesterases are regulated, because they are going to regulate the amount of cyclic di-GMP.
That's a very good question. I think we found in some cases that in the promoter side that there are also recognition sites for BLDD. So the phosphodiesterases controls BLDD and BLDD controls the phosphodiesterases. This is a very complex regulation going on. And yeah, we are trying now to focus also on how much of cyclic GMP is controlling the expression of the phosphodiesterases. This is very complex because to our understanding, cDGMP is not measured in the whole cell, but it is measured in parts of the cell. So there are compartments maybe going on which have to be controlled.
Okay. Can you ask one more question?
Yes, sir, sure.
So many times we have seen that in bacterial spore germination, there are many kinases and phosphatases are involved. Here you have mentioned that deletion of BLDA probably did not germinate, isn't it?
Yeah. So
I'm saying if you delete BLDA gene, then the germination will not happen of these spores. Is that right?
Yes, that's correct. That's correct. I think if you take all the mutants which do not have the BLDA gene or have a mutation, they cannot sporulate.
So what are the proteins which actually regulate germination after BLDA?
Oh, there are many other genes involved.
Are there kinases and phosphatases?
Oh no! To my knowledge, they're not. No. They are the Y genes. They are structural genes, so nobody knows really the function. It's very difficult to predict. But to my knowledge, there is no phosphodiesterase involved in this process.
But last question of mine. So BLDA is going to regulate synthesis of many molecules. So are there large operons so that BLDA can bind at different places? So the structure of the molecule is going to be changed.
Oh, I think BLDA is not binding there. I think BLDA is only used for the translation process.
Yeah.
It's being used there. And if BLDA is not there or not in sufficient amount, it stops.
Does it activate transcription?
No, only translation. It's only involved in the translation process.
Okay, thank you.
Thank you so much, sir, for these interesting questions. I request Professor Ruplal to say a few words before we end the talk.
You want me to? Thank you, Andreas. It was a wonderful. I know it's a very difficult system to work with actinomycetes, and you have been sticking on to this and have become now a leader in this particular field. So it was really a very informative talk, and I'm very sure all our colleagues and participants who were waiting to listen work on polypeptides and on newer this particular aspect will be benefited. Thank you very much once again.
Thank you, Rup. I must thank my postdoc, Dr. Roman Makitrinsky, who really is really good in this, and he has most of the ideas come from him.
Thank you so much, sir. This was indeed a very interesting talk to know about such regulatory genes. Thank you so much, sir, once again on behalf of AMI and INSCR for being a part of the conference as a speaker. Thank you, sir.
Well, thank you for the invitation.
Thank you so much, sir. Now, moving on with the conference sessions, I request Dr. Princey to kindly introduce our next set of speakers and the conference theme.
Thank you, Nirjara. Good evening, everyone. So moving on with the next conference session, welcome to our special session on translating innovative ideas to enterprise. We are pleased to have with us Dr. Amulya Panda, Director NII, Dr. Mrityunjay Swar, Director General R&D KIT University as chair of the session, and Dr. Mona Singh, Fixed and Private Limited, and myself, Dr. Princey, as co-chairs for the session. Now it gives me immense pleasure to welcome Dr. Swar on behalf of AMI and INSCR. Dr. Swar is currently professor at the School of Biotechnology at KIT University, India, and also he is Director General R&D Department. His research interests center on developing a recombinant safe mucosal vaccine candidate against enterocolitic pathogens using Salmonella as a model organism. He has also explored the differential virulence properties of Salmonella typhimurium and Salmonella enteritidis. His research is mostly translational in nature, and he believes in developing platform technologies for common man which are sustainable and user-friendly. He has published many research articles in international peer-reviewed journals and also promoted 7 startup companies in the area of biotechnology and engineering. We would like Dr. Swar to kindly initiate with this session. Over to you, sir.
Thank you. So, thank you, Tripinsi. Thank you, the AMI. Special thanks to Professor Ruplal, Professor Yogendra Singh, sir, all of you for setting up this stage to talk about a special session on translational science. How do you build from science to enterprise? So, I hope I'm audible, Prin.
Yes, we are able to hear it.
Okay, thank you, sir. So I think we're a bit early because I told other speakers to join at 2:10, but anyway, I'll manage that. So today, what I'm going to speak to you for a little that, we have been talking about science since ages, and that's very important. I feel I have gone through a similar stage of doing PhD with Professor Ruplal and then doing postdoc, and henceforth I joined KIT. Now the time has changed, or we have to think differently today, because I think all of you will agree the learning from COVID-19 was different. And where I, as a entrepreneurial professor, I think I learned team building is the most important lessons we have learned from COVID-19. Forget about developing other stuff. So when the world is going through crisis, I think people realize that we have to build team with complementary expertise. And scientists have sometimes underestimated the potential, that what potentials they have. So with that, I think COVID-19 has taught us that you have to build our team, the policymaker, the scientists, the investors, the industries, innovators, startups, all grouped together. And that was, I think, in my life, my period, I've never seen this kind of very, focused strategy everybody was talking about. And today, the session which we are going to talk, I think this is going to be like that, because there are some targets by the government, by all of us, that how to build, enterprise which is coming from the scientific laboratories, like Professor Ruplal is talking about, or Professor Yogendra Singh Sahab is talking about. Now, this is very, very important, because we have done science and we still will do science, but how some of our scientific activities lead to technology development. And finally, we should not claim our technology is already there. It has to be in the market. It has to do business, commercials. So it has to take the shape of an enterprise. And this is very easy to say, but the task is not even impossible. It's quite difficult, but not impossible. And very happy to tell you that we have several agencies coming forward today and focusing on translational science. So scientists like us, because also I have a laboratory in KIT universities and having 10, 12 PhD scholars like all of you have. And this is message I want to give to all my scientific community, that you have to think differently. And today, the time has come, you can start thinking differently. You can do whatever job you're doing, publishing papers and all, but beyond that also you can do, and even for the students who are doing PhD, masters and all. So during the presentation of some of our speakers, you'll realize, even though they are scientists, they are doing things differently. They're also doing good science, but they are translating some of their scientific discoveries or developments into enterprise. I think nobody will disagree with me. There's no harm in getting more wealth or the finance out of your activities of scientific. So with that, I think we'll talk about, I'll talk about a few of our speakers who are going to start, but I think I have to call them because it's a bit early. So let me tell you what is happening in the country at the moment. Now, the best thing what is happening now, if you look at the whole funding scenario has changed. Now, the post-COVID, the new policy, you might have seen that even though science, they're putting more money, but most of the money going towards the development of innovations-driven R&Ds. It means we have to change our mindset, change our working style. So few of you have already done it, but you have probably have not done a structured way. So can we build this structured process, and what I call them through a special vehicle government is pushing called technology business incubator. Now, this is very, very important, because this technology business incubator is not a, not in laboratory, just like what we have been working with our students or all of you have been working. It is a different kind of entity which brings you laboratories is only a very minimal component, but it provides you several mentorships. It means you'll find people like Professor Ruplal and Professor Yogendraji, but also you'll find people like Dr. Manish Diban, who will speak to you from BIRAC side. Then Dr. Sriram Raghavan, who comes from industry side. There'll be regulatory people, there'll be design people, there'll be professors, there'll be policymaker, there'll be investors coming into picture. Now, why I'm saying these things are very important, when I meet people, even my Professor Ruplal is now practicing to be an entrepreneur. He has founded a company. This is important because we have been doing science that is go on, and that will go on because we are paid for it. But if you really want to bring more impact, can you build some of your activities in the laboratories to enterprise and for social impact or commercials way? Now, if you look at the Indian context, now all ministries have now set up their own vehicles. For example, DBT has now put up BIRAC, which is also a offshoot of DBT, is a Section 8 company, which is promoting, pushing the startup culture in the country. And we're very happy that myself, even though I'm a professor from KIT universities, And then, I'm a CEO of KIT TBI, which promotes startup companies. And we have so far, I think 250 odd companies we have promoted, not only in biotech, all the sectors. And somehow we have been doing quite good in these sectors. Now, why I'm saying, because I'm also paid to be a professor, I'm paid for to be a professor, but then also I can do some things which is beyond a professor's job, but which is also driven by science. Now, there, biotech plays a major role, which Dr. Manish Diwan is going to tell you in due course of time. The second one, like DST is now pushing through a scheme called Nidhi scheme. And believe me or not, Nidhi scheme is a huge budget. It's, more than what I think the science budget they have. And they're pushing several incubators, they're pushing seed fund, fellowship funds, prototyping fund. I think I'll be requesting all my colleagues, my the seniors, professors, the teachers who are in this platform, please go through it, encourage your students, your colleagues to be a part of it. And one of such grant, if you get, the whole body language will change in due course of time, because you will be asked in a different way in terms of accountability, in terms of development. So this is going to change some of your perspective. I think all of you should get into this. Similarly, now the things have changed, AICT, UGC, everyone. Now defense has come into picture. There's a program called Innovation in Defense Excellence program called IDEX. Now you'll be very happy, some of our students, they've been doing extremely well in this. Even one of our students who is a master's in biotech topper now has found a company, and the company is doing quite good. Couple of gross turnover companies, an enzyme-based company, and Dr. Bisakha is the co-founder. She's also going to speak about it. So things started happening because you need a different set of mentors program, different type of professors, different type of ambience. You need regulatory people, everybody into picture. Now, by doing this, what you're going to achieve, you'll achieve something which you cannot expect at the moment, because once you start building your technology, which leads to an enterprise and the business happens, you can really do a lot of exciting things without the money from government. You can really fund your own projects. And sometimes I'm very happy that some of our companies have really done great. They did some science led, they built a team of it, and then the team took it from science to technology and what we call technology readiness level. So all of you must learn these languages of TRLs. So which TRLs you have. And so when I was doing my PhD, I go to all conferences, everybody talks about I have a novel technology. But what the technology is, who is going to certify that? So there's a process for it. So incubators are built to look at this, help you, and then mentor you to go to next level of development. Now with that, I think the country is doing quite great, and I'll be requesting all of you that at this moment, if you really want to do something called translational science activities, which lead to technology, development and enterprise development, this is the best time I think we can have, because so much of funds available, grants available, investment available. Now, just looking at 5 crores, 10 crores, 50 crores investment is not a big deal. Now, why I'm saying, I'm also a professor, but I get research grant to my laboratories. When I go for incubation activities, innovation activities, when I push some startups, they get in multi crores, which is much, much higher than what a scientist. Of course, not all, but most of us don't get it. So that's why you have to change your language, the body language, your writing skills, the packaging of grant proposal skills. Everything has to be changed so that finally why you are going to do this project in next 2-3 years. What are going to be deliverables, not just like some manuscripts. Is it going to take something to a product, to a new process? Is there any value of it? So why I'm saying, because most of us, we talk about even patents. So the number of patents wise, the number game is not going to be now drive our ecosystem. It's going to be the potential of the patent, how much value it has, it is going to drive a potential. So this is what I think I can tell you. And so what I'm trying to tell you now, 5 speakers we have, and selectively, thanks to Professor Ruplal, that they agreed, they have to be onboarded. And we have Dr. Manish Diwan. I think he's very popular at the moment in Indian context, because he heads the BIRAC division called SPED division, which promotes strategic partnership and entrepreneurship development. And he has been the forefront in promoting biotech startups in the country, and you'll hear from him. And then we have Dr. Sriram Raghavan, who is a very seasoned, he's a scientist, hardcore scientist, and currently he is the Vice President of Iranum Private Limited in Coimbatore. Formerly, he was with a Swiss company called Evolva. And so then we have, so he's going to tell you about the programmed live biotherapeutics. Then you have a speaker, Dr. Namrata Misra, so who is, practicing, promoting startups in KTBI. She's also a PhD. So you look at all these people are all PhD, they're all scientists, but they have taken a different path, sometimes a hybrid path like me. Dr. Namrata is, PhD in bioinformatics and but then she's doing now promoting all biotech innovation in KTBI and quite successful. We have Dr. Vishakha Raine also, who did PhD from Professor Ruplal's lab with me. And she's also faculty associate professor in KIT University doing good in science, but she has taken up one more aspect that can I take some of my laboratory things to enterprise level. So she could build up a company called Kriya Food and Beverage, and very good that last year they were doing around 3 point some crores turnover. Look at, so science is going on, but simultaneously she builds a team with a master student and a couple of them. I think they have now 30 members team in the company, and she's growing. And we have Dr. Soundray Chaudhary, and she's also a scientist at Tripura University. She also got the innovation grant from BIRAC, but then she's also building some of the, from microbiology research. So she's going to talk about, she's not well, she's undergone a surgery. I'm going to play a recorded talk for you, very interesting talk, going to speak on microbial technology for clean environment. So these are the speakers from scientists, policymakers, the people, those who build companies, and then enablers like Dr. Namrata from KTBI. So they're going to speak about all these activities. I'll be requesting all of you to put up your questions and make it more and more interesting for them, because these sessions are not going to come to you in all the conferences. And a couple of examples I can tell you. When we talk to students in our universities, we talk to them about different aspects of these incubation activities, and some of them have taken this and they have built very big companies. Now, not the biotech one, the 3 students came to incubator in 2010 or 11, and today the company, 3 students of us, now the company, the owner of 3,500 crores valued company. Look at what can happen. They were engineering students. So some of you have to think differently, and now India, things are happening. Everywhere things are now happening. Take advantage of it and make it everything interesting for all of you. So thank you. I think what I'll do then, for the interest of time, I'll because Dr. Sound Chaudhary is not well and she has undergone a surgery. She has but I think why? How can I? Yes. So let's see.
So should we present?
Yeah, I think what I'm going to do, I'm going to present her slides.
Okay.
Right. Hold on, please. Yes. So Dr. Sound Chaudhary, she's working as associate professor at Tripura Universities, and she did a PhD in biophysics, molecular biology from Kolkata Universities. And he has also started a company that is called Waste to Wealth Technology LLP. And she's currently working on adequate microbial treatment of dairy wastewater for converting it into liquid biofertilizer, which replaces use of chemical fertilizer. Quite successful. And she received several awards, including BIRAC Big Grant. Also received Visitors Award in Technology from President of India in 2019. So she's not well, so she has sent recorded slides. So let us hear what she wants to say.
Excuse me, sir. So there's no sound in the lecture. So no, maybe when you share the screen, you have to put an option, share sound.
So I think she has sent the slides to you also, right?
Yes.
Can you do that at your end?
Yes, sure, sir. We can do it at
Do
our
that,
end.
please.
Yeah, please. Thank you. Just a moment.
Good evening, everyone. At the outset, I would like to thank the organizers for giving me an opportunity to present my work at this conference. My group, the Microbial Technology Group, has been working in the area of tailor-made consortia development for wastewater treatment. And that's what I'm going to talk about today. So the topic is microbial technology for clean environment. Our approach to lead towards clean environment is to develop processes either for minimizing pollution during a product formation or through pollution abatement through bioremediation, mostly using bacterial systems or microbial systems. The five different kind of wastewater treatment that has been developed by my group till date are the dairy wastewater treatment, petrochemical wastewater treatment, municipal wastewater treatment or the agricultural runoff treatment, ammonia-rich wastewater treatment, mostly coming from the aquaculture or the refineries, and tannery or the mining effluent treatment, which is rich in sulfate. Each of the technology has been IP protected through filing of patent, and some of these patents, be it Indian or international, have already been granted. As you can see from here, each of the technology developed by the group has a distinct advantage over the conventional process that is available. Actually, three of these have already been recognized by different awards. The problem that we try to address is mainly three separate problems, but they are absolutely linked to each other. First, the freshwater crisis, which is a global problem, the increasing population, which demands higher food production per unit land. That again is a global population, though the extent of severity might vary. And the third is the need for treating the enormous amount of wastewater generated from the industrial activities, which is also a must, keeping in mind the population increase. Now, as far as the freshwater scarcity is concerned, the United Nations report and also the Niti Aayog report states that within no time, about 40% or more of the global population would be subjected to water stress. This is going to affect or threaten the day-to-day activity and also the food security. Agriculture is a principal player in it, where it consumes about 80 to 89% of the fresh water that is drawn every day in countries like India. The increasing population demands higher food production, and that in turn results in use of enormous amount of chemical fertilizer. Only a small portion of it is utilized, and the rest is washed off, creating environmental problem. Now, the need of the hour is a process whereby the production would be increased, but the environment would not be damaged. And hence, we are looking towards organic farming. On the other hand, the large amount of wastewater that is generated because of the industrial activities that cannot be turned down drastically, though there are processes being developed to reduce the amount of water that might be generated, but nevertheless, the water that is generated needs to be treated adequately so that it doesn't cause environmental pollution. And that's what my talk is going to deal with, this one, especially where we are going to address the pollution caused due to dairy industry. This is one of the most highly polluting industry which utilizes large volume of water and generates copious amount of wastewater, about 1.5 to 8 meter cube of wastewater per meter cube of milk processed. This wastewater, unless treated adequately, leads to eutrophication of water bodies and hypersalinity of soil. So, the available technologies that are there are tedious and extensively cost intense, and they apparently tend to use about 1.5% of the annual turnover of the company. So from here you can understand that the small and the middle scale dairy farms cannot really adopt these. There is a requirement for new techniques or technologies whereby this nutrient rich wastewater from the dairy processing plant can be converted into a value added byproduct, could be a liquid biofertilizer. This liquid biofertilizer, because of its large volume and the nutrient composition, could actually replace the use of chemical fertilizer and fresh water for agriculture if it could be suitably done. Such an attempt could make not only the treatment at the dairy ETP self-sustainable, but also profitable. That's exactly what my group has been working for, had been working for quite some time. So here what you see is a conventional process of dairy wastewater treatment, a 7-unit operation. Often you do have one more unit here, an 8 units operation, where you have an anaerobic system placed for degradation of the pollutants. Now, the approach that we pick up for developing a tailor-made microbial consortia is to first understand what are the pollutants that need to be converted or treated, that is either removed or converted to another form. Here in this particular case, we could check that these were basically the nitrogen, phosphorus, carbon and potassium components, either the total or the available form. So our approach here would be to convert these into a form which can be easily utilized by the plants so that the need for adding them externally through the chemical fertilizer is not there. We don't want to lose the nitrogen here through bioremediation to nitrogen gas. We don't want to convert that because then we'll be losing the nitrogen, causing pollution because of release of nitrogen gas to the environment, and at the same time there would always be an economic need because it's a energy intense process of generating the nitrogenous fertilizer. So if we lose it, we not only pollute the environment, but we continue the need of producing nitrogen based fertilizer. So our objective would be to convert these components into a plant utilizable form along with the water so that complete use of fresh water and chemical fertilizer can be removed in terms of agriculture. So here, in order to do that, the next step is to understand what is the kind of metabolic process we want the microbes that we select for the consortia development to have. So from the nitrogen metabolism pathway, we can understand that what we want is microbes with the respective enzymes that can result in conversion of nitrate to ammonia. And we are surely not looking for microbes which would convert the ammonia back to nitrate or to nitrogen gas. So from here we can make out what are the pathways which should not be there in this particular organism or these organisms that we select, and what are the desirable pathway that we are looking at. So mostly conversion, the end product should be ammonia in case of nitrogen. Now, we started with isolation of cultivable bacterial population from the activated sludge of mother dairy ETP at Dankuni, West Bengal. These isolates were characterized at length, and each of them were even tested for their bioremedial activity, be it removal of phosphate, nitrate, nitrite, protein, carbohydrate, or production of ammonia. We also checked these isolates for their ammonia producing ability under suspended as well as immobilized condition, that is in the form of biofilms. So now we know which among these isolates that we were able to get enrich from the activated sludge could be involved in the remediation or the conversion that we were looking for from the dairy wastewater. So the next question was, which among these could be the potential candidates so that they lead to the conversion of the nitrogenous component to ammonia and not through denitrification lose it as the nitrogen gas. So in order to do that, the draft genome sequence of the organism, some of the isolates were subject to in silico genome analysis, and we were able to get or identify candidate bacterial isolates which had the requisite genes for ammonification, but they lacked the genes that were required for denitrification. Again, they were isolates which had the respective gene for production of protein that would provide protection against the reactive nitrogen species. There were also genes present that were responsible for production of the precursor of indole acetic acid, that is tryptophan. So all these indicated towards these microbes being potential candidates for plant growth promotion since indole acetic acid was there, or for production of ammonia. Now, once the isolates were understood, they were well characterized, we had selected them as per the approach I mentioned earlier, they were combined in different proportion and different consortia were developed. Among this, the one which produced the maximum amount of ammonia was taken, and the ammonia production by this consortia was compared with that of the individual isolate. The result clearly showed that the consortia had a much better production of ammonia as compared to either of these isolates, and hence for subsequent study the consortia was used for further studies. Now, this particular consortia was then further characterized in terms of the optimum bacterial inoculum to be added, not only in terms of the ammonia production at different inoculation percentage, but also in terms of the nitrate reduction, phosphate reduction, protein reduction. Now, this consortia was also tested for the temperature it could tolerate and the pH range it could tolerate, as well as to determine the optimum temperature and the optimum pH for it to produce ammonia. This consortia was tested for its biofilm forming ability, and the comparison of ammonia production under suspended condition and under immobilized condition was also carried out. Here again, we saw that ammonia production under immobilized condition was much higher for the consortia as compared to the under suspended condition. So here 4% inoculum was selected for subsequent experiments to be carried out. Here what you see at the bottom panel is the same image of the biofilm that has been formed by this consortium. Here you have a look at how the process has been scaled up from a 125 ml biofilm reactor till 11,000 liter per day processing capacity reactor, which was at OMFIT Bhubaneswar. This 9,000 liter per day processing capacity reactor is at Gomati Dairy in Agartala. And actually this was initiated, the entire process was started in April 2018, and it is continuously performing with that single charging. For each of the reactor at the lab scale, we had carried out the time course of ammonia production, and from there we could get that 16 hours of hydraulic retention time was the optimum time for ammonia production or conversion of the dairy wastewater into liquid biofertilizer. So the larger reactors were designed based on this finding with the HRT of 16 hours. The larger reactors at the two different industries were tested for their performance on a regular basis, and as we see here, the reactor could start performing their function from the very beginning, be it nitrate reduction, phosphate reduction, or ammonia production. However, the efficiency was different in the two cases. That could be because of a lot of reasons. The influent characteristics was different in both the dairy. The process used for formation of the biofilm in the two reactor was also different, and the climatic condition was also different in the two regions, one at Agartala, the other at Odisha. That's Bhubaneswar. So from here, what we get to see that irrespective of where we place it, irrespective of the geographical location, this particular system can work efficiently. Here, the next step that we wanted was to understand if at all a 16 hours of hydraulic retention time was required for this conversion of the influent into the effluent, that is the dairy waste water into the liquid biofertilizer. In order to do that, we did a spatial distribution analysis where samples were collected at different point 1, 2, 3, 4, 5, 6, and 7, and they were tested for the ammonia production and the nitrate reduction. What we could see from here that the ammonia production reaches saturation at point 2, and there is no further increase in ammonia production as the sample moves through, effluent moves through this entire path. Similarly, for nitrate reduction, the maximum reduction on almost saturation was there within the point 3. So from here we could see that the actual time required for the process was much less than what we were thinking. This is another indication that this process is scalable and its efficiency increases on increasing the scale of operation. In order to confirm this finding, what we did was that we tested this removal or ammonia production in a smaller reactor of 27 liter in continuous mode, and the time course of ammonia production or nitrate, phosphate, protein, COD and BOD reduction clearly showed that the desired reduction was taking place within 4 hours of incubation, and the ammonia production also was more or less achieved within the 4th hour. However, though it was much higher, slightly higher at 16th hour, I would say, if you try to consider all these points, but nevertheless, within 4 hours, we had substantial amount of ammonia that was being produced. Hence, this process is by far the fastest dairy wastewater conversion process where within 4 hours the entire wastewater could be converted into a liquid biofertilizer. Now, as I said, that the system has been running for a long time. So the next question that came to our mind was whether this particular consortia that we had charged into the reactor was retained even after 17 months of operation. It is expected that other microbes would come in just because we were treating actual effluent under ambient condition. Nevertheless, we wanted to know what happens to the native population, whether it was retained or not, the native population of the biofilm that we had generated to start the bioreactor performance. Metagenomic analysis shows that that particular community, of course, continues to be there, but there are new members who are added. So overall, the new members which were there in the wastewater could not replace the existing biofilm. Of course, they have added to it and they are also present there. So though indirect evidence of the consortia being there was there from the fact that ammonia production was consistent throughout, nevertheless, it was reconfirmed through metagenomic analysis. Now we know that we have a system which produces a water solution or a solution which is rich in ammonia, also has phosphate. So there is a scope that it might work as a plant growth promoting solution or as a biofertilizer, liquid biofertilizer. But the real tests had to be done and the field trials were carried out for Mungbin, which showed an increase in production of 1.56 fold. We had black gram, which showed an increase in production of 1.04 folds. We had maize, which showed an increase in production of 1.19 folds. And when we looked into the nutritional quality of these produce, we could see that they were at par with chemical fertilizer grown crop. But in case of Mungbin, we saw that the biofertilizer grown crop was much better than the chemical fertilizer grown crop. For Remy fiber, there was a 1.23-fold increase in production. For aloe vera, the biomass increases, the gel content increases, and also the viscosity of the gel increases, which is responsible for its medicinal property and the polysaccharide that's there. So it's a positive indication. When we checked for sorghum Sudan grouse, there was a minor difference in this experiment. Here, this is a fodder crop. So we actually had tested without any fertilizer when compared to the grass grown with liquid biofertilizer, because one does not grow fodder crop with chemical fertilizer. When such a trial was done, we could make out that there was an increase in biomass yield of 2.53 folds. And actually from here we could actually understand the reason or the mode by which this biofertilizer was working. And what we could see was that the route for the biofertilizer grown plant was much shorter as compared to the control, indicating that the nutrients, plant growth nutrients, were available in the root zone, and so the roots didn't have to really travel deep into the soil to get the nutrients. So it is the easy access of the nutrient which is ensuring that there's a healthier growth of the plant with this biofertilizer application. It was tested out for centered rice, and we had an increase in percentage filling of grain. It was tested out for lemongrass as well. Not only the grass biomass, that is, increases, but also the oil content increases. And the increase in the biomass is 2.82 folds. So from here we can understand that if anybody plans to adopt this biofertilizer as compared to a chemical fertilizer, there's a substantial increase in the income that the farmer would get, in addition to the fact that he or she would not be using any chemical fertilizer, so would not invest for that, would not be using fresh water. Looking at the impact that this process, this biotreatment plant would have on the environment, not the fertilizer and that's its impact, but the energy consumption for conversion of the dairy wastewater into the liquid biofertilizer is much less. So because the energy consumption is less and the time required for treatment is also less, if we convert it to the carbon dioxide equivalent emission, then what we find is there's a 89.92% decrease in CO2 equivalent emission. And the investment that anyone adopting this technology would need to make would also be 78.83% less as compared to the conventional system. Now, the customer value proposition who picks up this system as compared to a conventional system, it's a benefit not only in terms of a reduced investment for the biotreatment plant purchase, but also there would be a continuous revenue that the customer would be able to generate from the sale of the biofertilizer, unlike the conventional system where one has to go on just spending continuously. At a glance, our technology, it's a 1 or a 2-step process depending on the wastewater nature as compared to a 7 or a 8-step conventional treatment process. The retention time varies between 4 to 16 hours as compared to 120 hours in a conventional system. The energy required for treating a 500 meter cube per day system, the energy required for running it would be 3 to 6 kilowatt in our case, while it is 70 kilowatt in a conventional system. There is no water that would be discharged in our case. There is no scum formation, and there's a value added product which is generated, that's the biofertilizer. So considering the ETP with the surrounding farm, it is a zero discharge technology with agricultural sustenance and yield enhancement. On top of it, there is saving of fresh water from being applied for agriculture like purposes, which really doesn't need the fresh water. The smaller dairies can be compliant with the pollution control guidelines without adding to the cost of the production of the milk, especially in this highly competitive market. Another information was that the COD of the indigenously produced biofertilizer was much less as compared to the commercially available biofertilizer in liquid biofertilizer available in the market. Hence, that again proves that this is eco-friendly. At a glance, what we have been able to, our approach or what we have been able to develop is a process whereby environmental microbes selectively screened have been put into biofilm, and that biofilm in the biotreatment system converts the milk processing wastewater, generating 89.99% less carbon dioxide. It converts it into liquid biofertilizer that can sustain production of organic crop. The quality is maintained, the quantity is either maintained or enhanced as compared to chemical fertilizer, while giving all the advantages of an organic food. This work has recently been published in Journal of Cleaner Production in January 2021. These are the group of scholars and students who have been associated with the microbial technology group and the different processes that I showed at the beginning. And I'm extremely thankful to my collaborator, Professor L.M. Gantar, formerly from Bhabha Atomic Research Center. He's the chemical engineer in the group and is responsible for the designing of the reactors. I'm thankful to the granting agencies, the Biotechnology Industry Research Assistance Council. Under the big scheme, I received the funding from where this dairy work could be carried out at industrial scale. I'm thankful to Ministry of Human Resource Development. It is Ministry of Education now, Government of India, under the FAST scheme for the Center of Excellence in Environmental Technology and Management that they had given to my lab. I'm also thankful to DBT, UGC-DAE, BRNS, DST, ICAR, and IISWBM for the fundings they have given from time to time. I'm extremely thankful to Tripura University, where I'm currently working, to KIT TBI, who actually helped us in implementing the big project since we were incubated with them, and still we enjoy the hand-holding from them. And I'm thankful to Center of Excellence in Environmental Technology and Management, which is at Maulana Abul Kalam Azad University of Technology, West Bengal, where some of my scholars are still there, and it is part of the group. I'm thankful to the dairy associations, especially Mother Dairy at Dankuni, India, who gave us the activated sludge for isolation of the microbes. Gomti Cooperative Milk Producers Union at Agartala in India, where the first biotreatment plant was set up and it is running till now continuously. And also OMFED India in Bhubaneswar, that's Odisha, where we were able to establish the second biotreatment plant. Thank you.
Thank you. I think for all of your knowledge, Dr. Son is, I think she underwent some surgery. Son, if you are there, can you pick up one question maybe? Because I know you'll not be there for long. So Son, there's a question for you. I think the professor has appreciated. There's one question, Rajani Gupta, saying in consortia, which organism were used in which ratio?
Okay. This is a combination.
Are you still in the hospital? Are you fine?
Today only I've been discharged because tomorrow was the operation. So I've just reached, home. So what I wanted to tell was this organism, it's a combination of Bacillus aeromonas acinetobacter, and they've been combined, it's 6 isolates combined in 1:1 proportion, because that's what gave the highest production of ammonia that we use.
Any questions? One more questions maybe? Professor Ruplal sir, if you allow, one questions for her.
Hello. Hello.
Yes, sir.
I mean, I think, I don't know whether I have missed your presentation. I'm curious to know where exactly you are using your consortium and what is the turnover and how much you could actually eventually earn from your this startup or whatever it is.
Okay, the consortium, sir, it's we use it as biofilm in the packing matrix that's inside the bioreactor. This is for the consortium use. As far as the turnover or what we have earned, no, we have not started earning anything yet because we are still waiting commercialization. With the BIREC funding, we were able to get into 2 industrial trials, and that is it for the moment.
Okay. Thank you.
Thank you, Son. I think get well soon. Wish you all.
Thank you.
Yeah, thank you.
Thank you.
Thanks. So now to move on to the next speaker. This is the first time for all of you. I think the session got preponed, and we have the next speaker. I think all of you know, we have talked about him. Like Dr. Son also have been the beneficiaries of BIG grant of BIRAC. So let me introduce Dr. Manish Devan. I think Dr. Devan, you are around now.
Yes, Dr.
Yes,
Mithun,
yes.
very much.
So Dr. Devan is a very known face in the bioinnovation community. Dr. Diwan now heads the Strategic Partnership and Entrepreneurial Development at DBT BIRAC. We call the SPADE division of BIRAC. And prior to this, he's also a scientist, very knowledgeable scientist. He worked for many industries like Daiichi Sankyo, Ranbaxy, Dabur Research Foundation, and he is a PhD from Jamia Hamdard University and also in the ICGB on pharmaceutical science and immunology. And over the years, we have seen he has been instrumental in promoting the biotech startups, the biotech incubator in the country, and building the right entrepreneurial ecosystem through BIRAC, BIG, and other programs. So Dr. Diwan is going to speak about how BIRAC is promoting this in the whole country. Dr. Diwan.
Thank you, Dr. Mrityunjay, and I also acknowledge the organizers of this event for inviting me over and sharing some of the thoughts in this direction in the translational sciences, which is a unmet need and a huge area where a lot of contribution can be done from academia as well as the stakeholders who are participating here. So is it okay, Dr. Mrityunjay, can I share some slides?
Yes, I think they will allow you.
All right, let me try.
They must have done it.
Yeah. Let me know if you are able to see my...
yes, make it slide mode. Perfect, sir, perfect.
Looks good, okay. Fine. So I've been asked to share what BIRAC has been contributing in building a biotech innovation ecosystem in the country. So in next 20-25 minutes, I would try to share some of the overviews and in this direction. To begin with, I think we are such a large population, almost like 1/6 of the entire world. And this offers both opportunities as well as challenges. And if you all notice that there are a lot of unmet needs which are still there and needs to be needs attention and contribution. And as we all say that charity begins from home. So can we not solve our unmet needs by the large pool of talent that we already have, and look at the resources and develop such affordable, customizable solutions which can address the unmet need of our Indian society, our local requirements. For example, if you have very high-end medical diagnostic tool, let's say a high-end ultrasound machine, it can work fairly well and it can serve all its purpose when we are using it in a metro city, a tertiary care center. But how about a place where you don't have a regular supply of even electricity or a limited bandwidth of internet? Can such solutions still be used although they are world class? So we need a lot of, probably not. And I think that is one large area. And 65% of our population lives in such low resource settings. So that's one example. Within medical devices itself, we are importing 70 to 80% of the equipments and diagnostic tools and consumables every year. Just to so that's a large imbalance which we are catering to and carrying forward. Likewise, other segments also one can pick it up. Is it that we don't have enough talent in the country where we can come up with such solutions and cater to these requirements? Is it that our resources are limited, or there are no opportunities where these ideas, innovative ideas, can be nurtured? I think probably one thing is clear, that there is large unmet need, and there is a clear opportunity that translation of our knowledge can progress into tangible products and technologies to address these unmet needs. But why is it not happening, or is it happening to some extent or not? Let's look forward to maybe in next few slides I will try and address that. So when somebody identifies a pain point, an unmet need, to create a solution and translate the solution into a tangible product, you would need clarity what differentiation you are aiming at, uh, technicality around it, whether that particular solution is technically feasible, and a complementary team which bears that know-how of the techniques to put it and implement and translate into a tangible outcome. Uh, More often than not, we would hear that there is a knowledge creation in the research institutions, publication of papers, educating many people in that particular subject matter and expertise to create a large, a pipeline of such talent pool. But then not all such knowledge gets translated and gets reduced to a product or technology. So commercial aspect, business perspective of that particular solution is also very important. And I think uh, this dissemination of understanding, dissemination of this knowledge has actually reflected back. And you all know that COVID stuck and unannounced, and everybody was on a same threshold, be it from India or from outside or international at the world level. India is the one of the front runners in vaccines production as well as new vaccine candidates. So there is no dearth of talent in terms of innovations and contribution to even first-in-class drug discovery. We could see that when COVID struck, there were no diagnostic kits. In fact, we had to send samples from India to US to get it tested whether the person is COVID positive or not. We had no testing centers also. Today, we are making 5 lakh plus kits every week and addressing unmet need within India as well as exporting it to the world. Today, we have more than 2,500 diagnostic test centers for COVID. So likewise, the PPEs, face masks, we were all importing it. Now we are actually exporting the surplus to the world. So there's immense amount of talent, immense amount of uh, examples where this translation activity has been delivered through the startup ecosystem, and that biotech innovation ecosystem has reached to a critical mass where we have successful examples. uh, People can pivot and bring it forward, just like the Sean was mentioning the technology know-how understanding she has been able to convert into a tangible product too. So for a national level such activity, BIREC has been specifically carved out from DBT as a not-for-profit Section 8 company, which facilitates all aspects of translation of knowledge into commercialized products and technologies. And this is brought across the biotech sector, which is a fairly interdisciplinary. And just like microbiology, this is also touches health, touches living beings, and wellness, and even industrial biotechnology. So it is integrated, and I think this particular forum and attendees for this from these backgrounds can directly relate to such activities. From BIREC, we have developed facilitations mm, and programs, schemes that can cater to all the stages of an idea generation to to proof of concept to validation of that prototypes and the proof of minimal viable prototype, and then taking it all the way forward to commercialization. So based on the stage of that particular idea, be it a student, a faculty, an entrepreneur, a research scientist in an industry, or a scholar, or a startup, medium scale industry, or large scale industries. So for all of them, there is there are specific specific stage related facilitations that are in place to promote and help translation and building the biotech innovation ecosystem. On the left side of the screen, you could see the map of India. Each red dot reflects an incubation center, which are breeding centers uh, specifically designed to develop biotechnological solutions and translate ideas into minimal viable prototypes. So these incubation centers are spread out across the country, and at present we have about 55 BioNest incubators, and if you these have been spread out in a sense that it reaches to tier 2 cities as well, so that entrepreneurs and startups can engage from all parts of India and connect to the biotech innovation ecosystem. Some of the states, some of the unoccupied green area, where we do not see red dots, in next couple of years we are adding 10 incubators every year, so we will be able to cover a lot of the other grounds which is still unmet. If you look at distribution of biotech startups in the country, these red bubbles on the right hand side graph, you could see that we have provided these incubation centers in those clusters of startups where this biotech innovation uh, innovation culture is more advanced. And in fact, if you look at progression of number of these startups over a year from 2017 to let's say 2020, we have seen a rise of more than 2.5-fold in terms of the number of such startups. And I think our incubation centers presence is actually providing a a life a bloodline to lifeline to all the startups in these regions to nurture and thrive. So we are reaching out to such areas where so that each an individual entrepreneur or a startup does not need to relocate just because there is no incubation next to or in the vicinity to that person. And these incubation centers or these funding provisions or facilitation provisions are done across these sectors, as I mentioned before. This is a glimpse of different incubation centers which offer high-end access to high-end infrastructure, laboratory environment. You can have your own cubicles, you can incorporate your own company, and you can utilize the address of such incubation facility to for your startups. At depending upon the stage of the of the entrepreneur, startup, medium scale company, or a large scale company, so we have created provisions as an entry level for students which are at undergraduate level, e-Yuva scheme, for example, with a fellowship amount, to an entrepreneur for biotechnology ignition grant, or equity investment schemes such as seed, SIBRI, BIP for where you have industry partners coming together with the academia or industry alone. Uh, there are various schemes which have been created just to facilitate different stages of movement of an idea to a tangible commercialized product. For faculty entrepreneurship also, there is a dedicated scheme, PACE or AIR. As you see a progression of the idea into prototype to a product, you would require increased amount of funding. So that is what is the provision from fellowship to all the way to $1 million for a particular startup is being provisioned. There is also mobilization of private equity uh, through the venture funds, angels, and high network individuals into the biotech ecosystem through fund of fund and other mechanisms. Just like we are talking now, so that there is awareness that what are provisions have been created through the Government of India initiative, BIRAC DBT. Likewise, these several schemes, they also reach out and let people know that there is now a defined path, that if you intentionally take a decision to move into entrepreneurial journey, there is a hand holding from stage A to stage B, stage C, and henceforth, so that you can become not only you can become job creators rather than job seekers to begin with. And the startup uh, culture in the country is not a new thing now. I think India is now recognized as the third largest nation of the startups. So and biotech startup ecosystem is being is evolving, and I think there is great amount of opportunity which is sitting. So the creation of pipeline is consciously done so that we have the base of the pyramid so broad that we have many such innovative ideas translate into tangible products at the end of the day. Just a glimpse that within these 55 bio incubators that we just spoke about, within the COVID days of let's say 6 months or so, we got 125 products, COVID related products, which got commercialized. So that's the power of innovation ecosystem, pivoting of entrepreneurs, startups to address a high unmet need for the country as well as for the world. Through BIREC's various activities, not the academia environment trains us so that we have deeper technical understanding. But there are regulatory, there is how to handle the intellectual property, what should be our go-to-market strategy, how to reach out to industry, how to create and identify these vendors where we can source in our components, or how to design a product. So all these mentorship, all these additional know-how, which is not a structured program in universities or colleges or research institutions. So entrepreneurs, startups do require this, and this gets disseminated and provided with through incubation network across the country, and special programs, special workshops, hand holding, high-end skill development, evangelization to all the way to investor connect. So all these activities are done across the country, and we touch institutions, as I mentioned, research institutions, as well as incubation network. And uh, this is not left at national level only. There is clear conscious attempt that we also provide and bring in international orientation to the entrepreneurs and startups, so that tomorrow when they are creating a product, they create a product which is world class, and if it is successful in India, can also be deployed outside India. So facilities and uh, these support system is now has reached to a level where anybody in the biotech domain who is working as startup or entrepreneur or intends to do, they are one way or the other gets connected through the biotech ecosystem of BIREC and DBT. And I'm glad that Mrityunjay is one such example, the kit Bionest, which has created a mature ecosystem in the eastern side, and they also run and lead one of the regional centers for BIREC. So through the international exposures, we bring these startups, entrepreneurs at a level where they can compete at the world level, and based on merit. And we are very happy to see that they win accolades, awards, even investments from foreign investors, and have been able to deploy their products outside India too. And one is that individual person learns from their own experience and it takes some time. Other is that within such peers, startup peers, entrepreneurs peers, we adopt and learn from somebody else's experience. So these peer-to-peer interaction are very meaningful. Just like any technical conference where we learn what is the cutting edge technologies which is happening, and I think this particular conference also provides that opportunity. Likewise, imagine all startups, all entrepreneurs coming together and then talking about their aspirations, opportunities, gaps, and strengths. So these kind of networking, showcasing, and outreach activities are done regularly through the BIREC network. There are several aspects. One, I said that those several schemes, which I think details one can look at BIREC's website, where you decide what program, what project, what pain point you want to address. And those are bottom-up theme identification or problem identification. Likewise, we do several top-driven or theme-driven activities as well. And BIREC websites keeps on telling what are the new challenges which are on board. This is an example where we recognize women biotech entrepreneurs. And I think we just heard Sean. Sean is one of the successful women entrepreneur and was also created startup. So such people are recognized through winner award, for instance, by BIREC. We also bring the strong strength of partnerships. BIREC being a central agency enabler, so we can engage with not only the ministries, government departments, agencies within the country, but we can also connect through G2G alliances internationally. So those country partnerships, country engagements, uh, the investors, private funds, industries, NGOs, philanthropic organizations, not-for-profit agencies or corporates. So all that partnership strength is also made available to each and every single individual entrepreneur and startup to benefit from. As I mentioned, we take we pay conscious attention towards exposure and mentorship and expertise to startups at the international level as well. So various countries we we have brought together programs and activities so that our startups can go and compete at international level. And there's an ecosystem to ecosystem connect. India's ecosystem has reached to a certain stage of maturity. There are more developed ecosystems outside country, whereas in some countries the ecosystem is still evolving. So we have opportunities for both learning and penetrating in developed ecosystem markets, whereas in the evolving international ecosystem, those pain points are fairly similar to pain points that we identify at India to leverage and deploy our solutions. So that's an extended market too for our entrepreneurs and startups. India's is a success story, and we promote the destination India at international platforms too. Reasons are self-evident. We are our strength in the vaccine biopharma industry, uh, the number of manufacturing plants, capacities, and these are all in increasing uh, uh, in in a stage where it is all growing. So, for example, today we are having 3,500 biotech startups, which is a jump from the previous year of about 2,700 or 2,500. Likewise, the capacities are increasing year on year. So we are looking at India's bioeconomy, which is currently pegging at $70 billion to $150 billion in next 4 to 5 years. Basis because the biotech innovation ecosystem has that strength and has demonstrated that opportunity, which can have multiplier and cascading effect in actually contributing significantly to our national target of achieving 5 trillion USD by 24-25. This is another opportunity which is in the same breadth and same direction. Global Bio India is a national platform where we can demonstrate and share the strength and opportunities of India's biotech ecosystem and present it to the international community. So the next edition of Global Bio India, I'm pleased to share, it is coming on 1st to 3rd of March, which is done through digital platform, and it would encompass all the biotech elements and sectors and subsectors. So feel free to engage and utilize that opportunity. I think going forward, the knowledge clusters through the academia will get strengthened and create a conduit through the technology clusters, where there would be translation of knowledge from incubation centers, academia, entrepreneur startups, maturing uh, into production, into validations, and leading to large industries. So that's the overall goal for the biotech innovation ecosystem. We are looking at creating India as 100 billion biomanufacturing hub by in next four years or so. And there is already demonstration to that extent. We have few examples where such one is the incubator network, which is growing and thriving. Then the next stage is the manufacturing zones that have been created in some segments, like Andhra Pradesh Medical Technology Zone, um, uh, MedTech Park. That's one one of the subsectors. Likewise, if we intend to create such technology clusters and connecting them through the knowledge clusters, which are at the institutional levels, university research institutions, contributing towards the growth and mobilizing and creating more startups in the country for the biotech India. So we are anticipating that in next by 24-25 we will be looking at threefold increase in the biotech startups in the country, and their valuation, collective valuation would also be sizable, and we will be able to contribute to the nation's the biosciences to bioeconomy to about $150 billion in couple of years. Maybe this is my last or second last slide. I would request, since there are many people from academia in this congregation, couple of things if we can consider. It's my submission, or uh, that if we can include, for example, in our curriculum or our activities, typically we have industry visits as one of the activities. Kindly include a visit to an incubator or a visit to a mini industry, which is a startup, as one of the flexibility, and within the course curriculum, engage and create that opportunity. Let some of the thesis or dissertation work, some of those can also adapt such problem identification, which has a potential to get translated into a tangible, into culmination, into a startup, or a proof of concept that can give you engagement with an incubator. So what I'm saying is that one is that academia industry connect everybody intends to have and strive for as a convention. What I'm suggesting is kindly consider incubator as your path to industry and startup as paths to industry. And these could also be focal points for interaction. And you will find they are more agile, uh, conveniently accessible, and you could do a lot more, and a lot more in a more informal and a more crisper and a faster manner. Of course, connecting with the industry is not a substitute, but do consider such alternate areas also. And within our teachings, our sharing of knowledge, let there be a conscious effort in knowing and identifying what are the unmet needs, what are the pain points in the present day, as well as in next 4 to 5 years down the line, so that our activities, our undertaking of projects and going deeper into that segment stays connected and has a viability and an acceptability at the other end. Some forward pull is realistic. So if we take a conscious evaluation of these opportunities, might be helpful for us to move forward in a more tangible manner. I think these new policies, they promote faculty entrepreneurship, creation of venture companies. So kindly exploit and explore those provisions as well. So this is I will stop here, and these are all the details of schemes and programs. I would encourage kindly visit BIREC's website. You can also follow BIREC at his Twitter handle or LinkedIn, etc., so that you can have a fairly clear and up-to-date knowing of what is going on. There is a dedicated e-portal that has been created, which showcases in-market solutions by the biotech startups in the country, which has been supported through BIREC. So there are more than 100 plus such solutions which are listed down. So feel free to have a look at that and get inspired and get connected and contribute more as much as possible. Thank you so much. I'll stop here. Thank you, Mrityunjay. And
Thank you. Thank you, Dr. Devan. I think in a nutshell, In 25 minutes, you spoke about how to create $150 billion things in the Indian context. And that's a good one. I think for all of you, what he mentioned that, it's not only in science, it happens that you do PhD, then go for postdoc or outside postdoc. If you're into enterprise creation through science or technology, if you're from Uttarakhand, you can go to US and UK. From Faridabad to Finland also you can go, right? So I think from India gate to Israel you can go, from east of India to Estonia you can go. So everything can happen now in the country. And everybody is pushing this. It's not a Dr. Diwan, the whole ministry, the whole country is pushing this ecosystem development. And I think let us build it all together. The body language is to be changed. And also he mentioned what is existing at the moment with all of us, what needs to be done. So like, can you do something in incubator? Can you do a decision incubator with a startup company? So I think this language is to be changed from the top, like the professor, scientist. So I think, thank you, Dr. Diwan. I think we'll pick up one questions, probably a couple of.
Yeah.
So,
Maybe I have can I ask one question?
Of course.
Yeah. Thank you, Dr. Manish Diwan. It was a very wonderful information. In fact, this is what is lacking, and many of the academia or professors working, they only work for publications and probably are not aware of the initiatives that government has taken up. And Mrityunjay, of course, is an exception who's propagating this and doing very well. But I have a question because, while in academia, most of the time your emphasis is on publishing papers and you are not really properly guided. But fortunately, because of the guidance we got from Mrityunjay, and also we had a couple of leads, and one of the lead was to produce a rifamycin analog molecule that is effective against resistant strains of Mycobacterium tuberculosis. And we got a big grant, and I want to thank actually Barak for helping us and now to confirm the proof of concept, and we really could produce a sufficient amount of this analog, which is really will be very helpful eventually if it is commercialized. So we only want to probably, if it is possible, how exactly we go further from here. We have already successfully completed this first part of the big grant. So can you really suggest now, or can we actually discuss sometime when you have time? I know this is not the platform where we can discuss in detail, but is it possible to guide us now further?
Manish, you have to unmute. Manish, you have to unmute.
All right. So I think there was an overriding by the administrator. I could not have unmuted. Thank you, administrator, for allowing me to unmute. So, Dr. Roop, essentially to progress from one stage to another, as I mentioned, BIG is the initial stage where we are looking at innovative ideas to move to proof of concept. Once we have the proof of concept, you can explore the next program like SIBRI or BIPP. From academia, you can also look at CRS. So these are provisions which are specifically created based on what stage you are. So kindly have a look at BIREC's website and look at funding mechanisms beyond proof of concept. So I think you will find quite a few of options there. And congratulations for moving forward there.
Thank you very much.
And sir, I think for you, the best would be the CRS scheme. We'll discuss offline on this. I think we'll also take Dr. Diwan's help in CRS. So there is one more question from Dr. Banwari Lal. I think he did an introduction. It's from Terry, a very big professor, which is an enterprising professor. Sir, questions from your side.
Thank you very much. Manish giving a nice talk, and that BIREC is really serving scientists, especially entrepreneurs. So I can give my story. As you know, around 10-15 years before, we were struggling to get even 25 lakhs from DBT and other funding agency because our research running expenses are very high. So we are at the verge of closing of our research lab. Then we thought we should do some application oriented. Then in 2007, I started a startup company without any grant from anything. That time BIREC was not there. And now this company, 2007 I started, and now we have completed 13 years. We have generated more than 300 crore rupees. I have given 100 crore rupees as an income tax to government. So like DBT must have given me 5-10 crore rupees for research grant, but in return we have given more than 100 crore rupees. Large number of jobs are created. So I think that is the really learning that we learn how to do the business, whatever we do. Otherwise, we are depending projects after projects. Our job was only writing projects only. And if we write 10 projects, we used to get 1 or 2 projects, and write 50 lakhs, the DBT used to give 10 lakhs, 20 lakhs. So our situation like beggars cannot be choosers. So whatever they give, we used to accept and do whatever. But I think with this entrepreneurship, I can say now we have bid a tender of $1.3 billion in international.
Amazing, amazing. Congratulations, sir. I think that's the inspiring story, and we must hear from you so that everybody kind of realize this. If you can do it with the right intent and right technical know-how, then imagine each institution. Just I'm saying, like within an institution, let's say if you have 50 to 100 faculty, or let's say 20-25 different specialty labs. So imagine if 1 year each one strikes, and we could have those many number of such such successful stories coming out. So it can happen. I'm very optimistic about it, and I think you are a true example. And we just heard about from Dr. Roop also. So these are examples which are now real examples. We just need to multiply these examples all across, and that would be our biotech innovation ecosystem. And I think that's what the vision and of the nation is, and for the world is also looking at India now as an innovation hub. So together we can make it, sir.
Yeah, sure.
Thank you.
Sure.
Thank you, sir.
so
And
much.
thank you, Dr. Diban. I know you have to leave now. Thanks for encouraging all of us and providing inputs about what BIRAC is doing in the country. Thank you, Dr. Diban.
Thank you so much. I enjoyed my interaction.
So now we move on to the next speaker. I think you heard Dr. Diban, who says money is not a problem, funding is not a problem. Now, what is the problem that, ideas, how do you build enterprise? This is not a job of all scientists. So scientists can build part of it, but then who takes it over? So let us hear from one of the scientists who has been working in industries, who has built companies, and he's Dr. Sriram Raghavan. He's also in many of the committees of BIRAC and others. I think he's currently the vice president of Yananome Technology Private Limited in Coimbatore, and with about 18 years of corporate research experience, spanning diverse life sciences segments. He's also very closely working with the central and state government in Asia, including Indonesia, Singapore, and Malaysia. And I always invite Dr. Sriram for motivating our faculties in KIT School of Biotechnology in the university, and also he works with many of our startups, the incubator. So he brings a different perspective that how from science you can build business. He's a business technology parks and drafts framework policies to help government and companies for boosting the economy via bioentrepreneurship. And also he's right now in the board of ABLE. And he always talks about nurturing startups. So Dr. Sriram, next 20 minutes for you.
Thank you so much, and really want to thank the organizers, and thanks to you, Dr. Mrityunjay, and of course such an energizing talk by Dr. Manish as well previously. So if you remember the previous slide that Manish showed in terms of innovation, he showed an equation, so I want to continue from there. So with your permission, I'll go off video to save some bandwidth so that I can actually focus on the slide better. I hope that is okay, if that is fine. So. Yeah. Now, so if you remember the previous slide from Manish Diwan, he showed the slide saying innovation is equal to, he showed a very nice equation, say idea plus leader plus team plus plan. And here I just wanted to show you an idea in terms of what was an idea, how did we really take it, and then what are we presently doing. So I'm going to present one of my US company. So it's called eLife Biotherapeutics. So I've given the website. So those of you who are interested, you can actually go and see a little bit more. So this concept of eLife Biotherapeutics is all about programmed live biotherapeutics. So there are 3 words in it. So obviously, when you know biotherapeutics, it's basically about therapy, curing a disease, and then there is bio, there is always a biological element involved in it. And when we talk about life, that's what makes it interesting, because there is going to be a live organism that is going to actually do the work in terms of curing a disease. And more importantly, when there is program, you are able to have an ability to control this live microorganism in terms of what it is going to do to the disease. So I'm going to give you a quick example in terms of what we did with this idea to actually work on small cell lung cancer. So I'll go to the next slide. So just to give you a brief background of where I am and what I've been doing, I've always been involved in terms of high risk, business of synthetic biology. That includes right from biodefense, theranostics, flavors and fragrance, crop protection, including aircraft and submarine polymers, biofouling for ships, astrobiology, including Formula 1 racing materials, unmanned jet fuels, cosmeceuticals, fiberceutics, biosilks, a lot of areas. But fundamentally, it boils down to 2 things with microbes. One is producing unnaturally natural products, which means those that are available in nature, but we produce in unnatural ways. For example, if you were to browse my name, you will see my patent on the saffron, which we try to make fully using synthetic biology. So the second part is unnaturally natural products, meaning that which is not in the nature, but we try to produce naturally. So you're going to see a classic example with the engineered live biotherapeutics in terms of what we have done. So we basically take a microbe, convert that into drug factory, which is not in the nature, but we have actually created it ourselves. If you look at the kind of molecular therapeutics growth, the first generation was antibiotics. You all know about it. The second generation was more in terms of chemistry, lead optimization and all of that. Third generation was clearly biomolecules. It was vaccines, proteins, and of course you have gene therapies and all of that. Of course, then the 3.5G is what we call, which is working with human cells, and that's more in terms of CAR T cells, which is now quite popular. But now, going forward in the next 10 to 15 years, that decade is going to be more in terms of microbial manipulation and how you solve the disease. Why is that important? I'm going to come back to that in a bit. But right now, I'm going to talk about a little bit on that particular 4G technology, so to speak. So why is that important? Because if you had been looking at this popular graph, the kind of FDA approvals have been always steadily falling in terms of the existing therapies. If you look at the biologic side, that's not doing any better because you see a huge patent cliff. And going forward in the next 10 years, by 2030, you'll see a lot of void there also. So, and also there are other issues. For example, the existing methodology of, let's say, small molecules, it's got ADMET problems, perennial dosage issues, and half the drugs that you actually see today have got hepatotoxicity. So the second main problem is in terms of routes of administration. So for example, if you were to look at a biologics, it is really, not very conducive to the patient, nor is it therapeutically very effective at this point of time. Of course, side effects are something that we are all aware of it, both in chemistry as well as in biologics. And of course, resistance to drugs, it's a very well-known issue now, right from cancer to various diseases have developed resistance to the kind of medications we have. So one way to look at it, of course, there are many ways to look at it. I'm not saying this is the universal solution, but one way to look at it, it's basically an idea is, you are made up of, let's say, 37 trillion cells, but you have more than 100 trillion microbes just in your gut alone. So if you were to imagine your company as, let's say, your body as your company, you are only 1/3 of the shareholder. 3/4 of your shares are held by microbes, especially the gut microbes. So now, can we do something with that? Yes, we can. So one way to essentially take a human commensal, engineer that with essentially the kind of genes that actually produces a specific therapeutic molecule, and then convert that for something called on-demand, on-site synthesis. So essentially, you actually put a bug into a specific area, what the target, where it is really required, and that's going to really produce the drug as and when it's really required. You will not even know about it, so the diseases gets cured. So right now, your gut microbiome may be doing it, but that's why, for example, some of you may not even, would have felt COVID, whereas some of you would have actually gone through a worse set of COVID symptoms because gut microbes play a lot of role, which is of course topic for another day. I'm just going to talk about in terms of how are we going to approach that problem. One of the key things that we can do is basically by putting on-off control switches. The sensor specifically takes the microbes to the specific location, and of course, biocontainment to ensure that the bug is clamped to the region where it is targeted to. So what are the key attributes, for example, of such an in situ drug production? So the first one is obviously the microbial host engages host immunity. That's very important because I'm going to show you an example in cancer, for instance, where it's important that not only the cytotoxic drug, but it's also important that there is a cell-mediated immunity, wherein the different immune system, different immune cells, components of immune cells that's going to come to the specific site, and they're actually going to look at and target the cancer cells. Typically, cancer cells have an innate ability to hide themselves from the immune attack. So that's basically flat. That's one direct benefit. And the way in which we are going to do it for cancer, for example, we have got sensors that specifically target the microbes to the hypoxic microenvironment, because at least in the lung cancer that we're going to work with, small cell lung cancer, there are a lot of hypoxic region, it's rich in hypoxia. So lung as a region, as you know, has got a lot of oxygenation, but if those pockets of hypoxia are flagged up, we have got flagella motors in our host bug. In this case, it's going to be E. coli inicil 1917, which will specifically target those specific hypoxic regions. And then, of course, the rest of the magic actually starts. So we have got a BioBricks-based engineering approach where you can actually engineer diverse payloads into the platform, and that actually helps us to do a controlled production at the site. So, so with this particular basic idea, so this is a slide I'll just spend a few minutes with, because this is what gives life to what you saw in Manish's previous slide. So this idea is all great. So right now, where are we? So before we go ahead and do that, there are certain things important. When we actually translate an idea into an enterprise, what is important is we need to risk mitigate at all the stages, because at the end of the day, of course, this project that you're seeing on the screen, it is not driven by grant. It is some of our own money and some of the investors' money in the US, Israel, Europe, India, and many other countries. So when you're going to put our own money, we have to risk mitigate as much as possible. So what happens is, the first one is we need to really see is, are you going to work only in therapeutics? Can we really diversify for risk mitigation? Yes, it's got a potential for diagnostics. So obviously we cover this in IP. So the second risk mitigation is obviously the founding team. If you remember, that's one of the parts of the equation that Manish had shown in the slide. So the founding team is very important. So for example, we have Stanley Goldman, who've got more than 30 years of experience in that space. So we actually align with the right set of scientific team. Without the right set of scientific team, it's very difficult to risk mitigate the project. And of course, we start a company, we have incorporated in Delaware, for instance, but then we are also risk mitigating in terms of using the same platform, not just for cancer, but for other diseases in future. This is very important to find good pharma partners. But of course, eLive is not just a drug discovery company, but basically we are looking at production of drugs on the site on demand. So here again, what we have tried to do risk mitigation is that, the idea of joining or using an FDA-approved, molecule for small cell lung cancer and constructing a pathway. So that's a 16-step pathway. But the more important point in the last step is there are 5 more things that are important when you want to risk mitigate, let's say, a therapeutic enterprise of this kind of risky nature. So we actually engaged with the pharma partners very early on, even when we are in idea stage. So we know what our pharma partners want, because we are very clear that we are not going to go to the market all the way. So with them, we actually got access to FDA. So we know the FDA's regulatory path for this. We picked up a disease which we'll get a fast track approval. We also got an orphan disease indication for mesothelioma, for instance. So we actually got the roadmap clear. Number 3 is that once that is clear, we actually spoke to several insurers, for the payer strategy, because at the end of the day, insurance companies needs to approve this kind of a therapy. So they have given us in terms of the roadmap for it. With that, we actually met underwriters. Now, underwriters will tell you what are the risks and pitfalls in terms of we need to avoid, what is it that we need to do. So that we actually did it. And with underwriters' help, we actually went and met several, in terms of lead investors, as well as people who can give us an exit. And there we have planned in terms of listing this in NASDAQ stock exchange in about 7 years from now. So notionally, we are going to look at, let's say, about $25 million investment. And we are looking at about exiting at a value of about close to $150 million. So that's the kind of, which means notionally we are talking anywhere between 5x to 6x, if not 10x. So we are very clear in terms of what's the return we are going to get, when we are going to get, how are you going to get, and what's the risk mitigation plan. So this slide from that perspective is going to be very important in terms of how we actually manage the whole, risks and how we convert that idea into an enterprise. The rest is basically a broad theme. I don't have time to go into technical details, but if any of you want to know more, I can actually have a separate discussion. Essentially, we are trying to convert a drug, a bug into a drug-producing factory by engineering all the sort of genes in it. Remember, biosimilars or bioengineering or biotherapeutics engineering, 1 gene or 2 genes. Here we are looking at 16 or 17 genes. That's because we have done it in the past, last 20 years. Probably one of the few groups in the world who could do it. And we are going to formulate it into a nebulizer, for instance, that's going to conveniently fit into what patients are already using, so they're familiar with it. And the platform has got multiple applications wherever there is a long-term resident therapy for cancer, fibrosis, infection. Now, all that is fine. So where are we today? Before I go, I just wanted to spend a minute in terms of the differentiation. This is very important when you are going to seek for an investment. We need to risk mitigate it all the time. If you really see in this particular segment, there are about close to 137 companies in this particular slide that has got investment into human microbiome space in the last 24 months. But we are very unique from that perspective that we are actually looking at synthetic biology of small molecule for lung cancer. So I think that differentiation is something which most of the investors would expect. And the second differentiation is that because of years of experience in synthetic biology, we have got the right toolkit, we have got the right host, we have actually got the right sensors, because it's very important you have to have the right sensors. We have the right switches, and switches here, what I mean is in terms of we have the logic gates, the AND switch, NOR switch, and the NOT switch, which is very common if you have worked in electronics. These are very important because you need to have one condition, two conditions, or three conditions satisfied for you to activate a gene cassette. So all of this could work in tandem. So that's the kind of circuit and switches ability that we have. And of course, we have other on-off switches as well. And let's say for the bug to detect when it should produce something, when it should not produce something. So in terms of the lung cancer, it's very important because a small cell lung cancer especially has got very poor prognosis. The radiation has refraction, chemotherapy has got resistance, and of course, biological treatments are not very good because only 1/5 of the patients respond to the lot of immune checkpoint inhibitors. So, and also most therapies do not consider inflammation in the immune system, and also in terms of the commensals that are involved in it, and of course, some diseases require long-term maintenance therapy. So what we are doing is, as I mentioned before, the idea is to put control circuits to control the dosage, sensors to sense the disease, switch on and switch off in terms of how the drug is produced. And that actually makes it a living medicine. That's a kind of a new area that we are stepping in. So the next 10 years could be, and 10, 15 years could be the age of living medicines. And of course, we actually use the whole platform by commensals, because commensals actually help in terms of faster regulatory approval because they're all GRAS approved organisms most of the time. But what's important is programming the organism. So I'm not going to go much in the details of this slide, except to say that small cell lung cancer has got a huge need because in the last 22 years, there are very poor prognosis. Essentially, the patient has got about 6 to 8 months to live once the patient is sensed with small cell lung cancer. And right now, first 30 days, they are actually given a very tough, difficult chemotherapy. They are sent home. But after 30, 40 days, they actually come back with relapse. And when the patient has relapsed, it is very difficult to go back to the same old platinum therapy or the cisplatin therapy that people are using. So it's a very difficult disease to live with. So that's what we have chosen. And we have actually engineered the entire pathway that produces a small molecule. We have engineered the circuits. Now the bug is working. We have actually we have conducted the early animal studies. We have got some very interesting data. So this one slide captures where we are. So right now, for instance, if you look at step 5 is done in terms of how we actually engineer switches. And remember, all the steps that you see on the screen, they are first time in the world. It's never been done before. This is the first time that you actually see here. And number 6, we have actually engineered the hypoxic and nitric oxide as an AND gate, which means both conditions needs to be satisfied at this point of time. We have also engineered the biocontrolled cell lysis circuit. Right now, we are working with several partners in the UK to formulate that into a nebulizer. And eventually, the manufacturing will be out of Australia. So it's a kind of truly global collaboration of more than 8 countries that will be involved into this particular work. I'm going to stop at this slide. This is a simple slide. As I was describing to you, typically a patient is diagnosed with the limited stage lung cancer. That means that the cancer is not spread to the second lung. It's with only one lung just to start with. And then he completes chemotherapy, he or she. And then what happens is there's a treatment-free period at home. And during this time, there is always a relapse. So that is the time when we actually, we'll be engineering this. And preferably in terms of a nebulizer, it goes in and it's a metered dose. It reaches a specific site. That's because the bug has got the right sensors to take it to that particular site, especially on the hypoxia in a lung. And even if it accidentally goes somewhere else, it is not a problem because it will be completely washed away because that's the way the bug is designed and the lung as an organ is designed. And also remember, we are actually talking about small cell lung cancer patient, that the patient has got probably about 6 months to live. It is very difficult to extend the lifespan, but what we are trying to look at it is quality of life. Because each day that the patient spends, he or she is going to spend in grueling pain. So this particular treatment is expected to have a large improvement in the quality of life, even though the actual lifespan may vary from a case-to-case basis. And eventually, we're going to look at both cytotoxic and immunogenic cell death, so to improve the patient quality of life. So with that, I just stop. It's basically to give you an idea in terms of how we could actually take an innovative idea, translate that into going forward up to a listing of NASDAQ, probably a good roadmap plan. There are a lot of things that could fail, but the idea is to have a plan and then move forward. So with that, I stop and then take any questions. Thank you.
Thank you, Dr. Siram. I think this is something probably very, very interesting, because we have been doing things in isolation and you are bringing a holistic picture that how to build up. So any questions for Dr. Siram from the participants? Any questions? Are they all there, or I see 112 of them?
Yeah, so maybe you can actually help them communicate that to me later on, so I'll be happy to answer that.
Yes, I think in Dr. Siram, we invited you because this is the Association of Microbiologists of India, and if you ask me, the biotech in the country in India is driven by the microbiological guiding, right? So all into.
Correct.
So I think this is a very good platform where you spoke about it, and I think we have to. Okay, so anyway, so thank you, Dr. Siram. No questions for you, and I'm sure in due course of time we'll get several questions.
Yes.
From these participants.
Thank you for the patient audience, and thank you all so much. Bye-bye.
Thank you. So now, you heard Dr. Manish, who talked about funding scenario, the direct perspective. Now you've heard Dr. Siram, who has come from several corporates, build companies, who said, how can you build? And he was giving a very bigger picture. So, where you have to bring science, business, regulatory understanding all together and to build your strategy. Now, to do that, there are many other people so Dr. Siram is a mentor and he is doing things for the company. And he's also a mentor who's helping startup companies and incubator like us, like KTBI. Now we have one more group, one more personality here, Dr. Namrata Misra. So she's basically, she's heading the bioinnovation at KTBI and working as also assistant professor at School of Biotech at KIT. And Dr. Misra has been actively involved in promoting innovations in life sciences and startup ecosystem across East and Northeast. And many of you might be knowing her. And she's a PhD in bioinformatics from CSIR, IMMT from Bhubaneswar. So even though she's pursued science, but she thought I should get into a system where I can help scientists to think differently and enable them to take it to the next level of technology development. So she has been promoting this in KTBI, quite successful. So I'll be inviting Dr. Namrata to tell all of you that what incubators plays a role in promoting, accelerating innovations out of scientific community. Dr. Namrata, 20 minutes for you.
Thanks. Yes. Thank you so much, sir, for the warm introduction. And a very good afternoon to all the esteemed dignitaries, participants, and a big thanks to the organizers, particularly Professor Ruplal, sir, and his fiction team, Vipul and Utkash, for giving this platform to share views. My session topic is role of incubators in accelerating innovation. It's actually a little bit of challenging to have a session after prolific leaders in innovation ecosystem like Dr. Divan and Dr. Shira, and particularly afternoon sessions are a little bit boring. That's why all the participants, I guess, they have vanished. But I tried my best to make this lecture session interesting and simple, particularly for students present out here. So I'm just switching off my video because a lot of bandwidth issue at my end.
I hope you're able to share your screen.
Yes, I can. I hope my presentation is up on screen.
Yes, please make it full screen.
Sure. So for the past 2 hours, we have been discussing and we all agree that innovation ecosystem in India is thriving and the entire country is buzzed with enthusiasm to make a difference. Every day, one hears of a startup story. Every week, a potential unicorn is unraveled. And in spite of all the regulatory and scale-up challenges, over more than 1,000 life science startups, I can say, have been registered in 2020. And particularly the flagship program of Indian government, that is Make in India. It has infused the entire country with a lot of spirit and zeal to bring out innovative products and support innovation across the value chain of product development, from ideation to proof of concept to validation, scale, and commercialization. Now, if we look into the source of innovation, I can say that there are three major sources. One is the academia, which is definitely the nucleus for any scientific discovery. Startups, which tries to translate those R&D discoveries into some novel product or process which has a commercialization value. And again, with the support of various stakeholders. And then most important is the social impactful grassroots level innovations that are need driven, and a good example of the same of the FPOs, where farmers are driven by the need to organize. So today we can say that India have all the ingredients for success in this innovation arena. Keeping in mind that India has a large public and private R&D network of world-class laboratories doing excellent work in various fields, India is also known to be the pharmacy of the world, with rapidly growing pharmaceuticals, biologics, medical device industry. We are also the largest suppliers of generics across the globe. We are the proven leaders in vaccine development and supply. And I would say the major strength lies in the fact that we are a democratic and young nation with 2/3 of the country are below 35 years of age. Having said that, but now to leapfrog technologically to move forward to the next orbit, definitely a massive push is needed to tap into this young entrepreneurial energies that we have, to motivate them to take entrepreneurship as a serious career option, to think out of the box and generate some fresh innovative ideas. So as Dr. Devan has briefed BIRAC with his vision to stimulate, foster and enhance the strategic research and innovation capabilities of the Indian biotech industry, particularly the startups and SMEs, they have played a vital role in enabling this startup and innovation ecosystem mission. So these startups and innovators, they are now beginning deployment of technologies, I would say, into core areas such as healthcare, food, agriculture, waste management that are very much critical to India's growth. But the challenge in this present complex and competitive landscape is that innovators, that entrepreneurs, they need more structural and fundamental support to carve their niche in this global startup ecosystem. Of course, the Startup India, the ambitious program, which is launched by the Honorable Prime Minister, it has brought a lot of positivity among the Indian entrepreneurs with the funds allocated to support startups, particularly the biotech startups, where investors, since various startups has a high-risk projects, investors, they have a little bit, apprehensive about it. But certainly, I would say that besides funding, startups require more than just funds to scale up to become enterprises. They need training, they need connections, they need capacity building, which is usually provided by technology business incubator. That is where the role of TBIs come into picture. Now, the question is, what is TBI? What's the objective of TBI? So there is a big misconception that incubators, they just provide co-working space. But please note that beside the co-working space, incubator provides a wide array of services to help startups and innovators, including the access to instruments at a very nominal charge, funding support, access to strong network pool of mentors, investors, and most importantly, to help the young student startups to think through. For example, during the mentoring or handholding, the incubator team, they ask right questions to the startups to help them think in different dimensions. And this can be about like customers, who are your customers, what's the unit economics of the product that you're developing, have you jotted down the cost economics, have you worked on pricing, is there actually a market for what you're developing? And this is usually they remain ignored. And believe me, this is the most common reason for failure of startups that we have seen. That is the identification of the right problem statement, which has both, remember, both social and commercialization potential. Another thing that incubators help the startups is to monitor them continuously to help them, clear the technical and the business hurdles, regulatory compliances, the support needed in IP. So these also are very important aspects which TBI plays a vital role in monitoring the milestones, the TRL levels, the technology readiness level. Now, another big misconception that generally students have, that incubators exist only for startups and they go hand in hand, they are synonyms, but that's not the case. TBI is actually a holistic ecosystem that brings in various stakeholders, scientists, technologies, entrepreneurs, industries, investors. They come in one common platform, that's TBI, to support one goal, our mission, that is innovation, supporting innovation, manufacturing, marketing in India. Like for startups, as already discussed, incubator, they provide market access, they provide funding support, getting connected to different funding agency, government funding agencies, then mentorship. Similarly, for research institute, TBI also plays a very, very important role. It helps the scientific discoveries to be translated into commercialization potential. That's very important hurdle that generally academia face. They are very good at, I would say, their faculties, academias, they're very strong in their technology. But when it comes to business, that's usually not the scientist's cup of tea. Therein, TBI helps in connecting the academia with startups. They build a team to facilitate their lab to market journey. Also, TBI helps academia industry need mapping, management of IP, tech transfer. That's the biggest challenge. Now, for investors, TBI provides a platform for collaboration and co-creation opportunities. So these will altogether conclusively impact the inclusive growth and government policy inputs also. Now, let's see what is the strategy of the incubators that we follow to support startups and innovators. See, to support anyone, first you need to understand what is the problem. And the same TBI does. It first maps the risks and challenges which the startups are encountering, okay, as they grow from small team to viable ventures. And believe me, these risks are different at different phases of startup journey, which is generally represented in the schematic representation, okay. The first phase, if we simplify, the first phase is a search phase where the startups, they search for an idea, a problem statement, they search for an idea which has a commercialization potential, they search for a business model, they search for a market connect, problem validation, and there they need help in terms of team building, they need help in terms of correct problem identification, immersion with various stakeholders. After that, the second phase comes, which is a build phase, when startups are building their prototypes, they're building the products, and they are trying to reach a product market fit. And there, the needs are different. They need support of incubators in terms of access to instruments, funding support, mentorship support. And then as a final stage, is a growth stage where startups, they scale up, and then it's a revenue generation. And of course, they need more funding support in terms of private investment. So what I mean is that incubator takes a very decisive choice about this support that needs to be provided to a venture, but mapping exactly at what stage, the startup is. Otherwise, there's no point. So remember, an incubator is really self-sufficient. It cannot work in silos. In order to adequately support the startups, the incubator has to partner with a lot of ecosystem players like investors, mentors for providing technical support, research institutes for idea generation, industry bodies for test bed manufacturing of the prototypes, and all the stakeholders, they work hand in hand with the aligned vision of promoting innovation and entrepreneurship. And this is possible only with a strong engagement strategy and the deeper engagement plan with all the stakeholders. Now the question is, how do the incubators do that? First, Incubators, they start with mapping the problem statement specific to the incubator focus. Like some of the incubators, they are focused on their sector diagnostic. They are focused on agriculture. Some of the incubators are focused on device and diagnostic. But again, these focus should be aligned with the region specific. Secondly, is to sensitize about different funding opportunities. Just an hour back, Dr. Devan spoke about various funding schemes that BIRAC provides to innovators for every stage of technology development and commercialization, starting from the ideation stage to proof of concept to your validation, commercialization. And there's so much of plethora of opportunities. But tell me honestly, how many, for the students present out here, this question for them, how many students are aware of all the schemes? The hard fact is that many young students, they don't know what is TBI. We have been to Northeast very recently and they don't understand what is TBI. So sensitization is very, very critical factor to promote innovation entrepreneurship. Then there are various capacity building training programs on emerging fields of deep tech that incubator regularly provides in collaboration, again, with a very structured partnership. And subsequently, we go for a intensive engagement plans with the startups which are onboarded after a very strict due diligence, after understanding that yes, they are on the right track. We go for the accelerator programs, boot camps for nurturing and the growth of startups. So this was a brief about TBI. Now, since my association with KTBI has been since a long time, like close to 5 years, let me briefly tell you the role of how KTBI functions as a case study. Since Procedure Sir is here, please correct me wherever I go wrong. So, although Orissa is one among the India's premier education hub, and we have a large pool of skilled workforce, however, I'm talking about like 10 years ago, no cluster-based framework existed to maximize the cross-disciplinary scientific collaboration. And honestly speaking, still the scenario is not so good. And if you come to startup ecosystem, it was very in a nascent form as compared to metro states like Delhi, Hyderabad, Bangalore. So to overcome this challenge, KTBI was established with a mission to create a conducive ecosystem for technology-driven research that has a commercialization potential. And it was built in a 20,000 square feet campus in Bhubaneswar, Orissa, which over the years, they have grown into pan-India ecosystem, innovation ecosystem. Particularly, we promote entrepreneurship and innovation startups across East and Northeast India. And KTBI was a pioneer. That time there was no benchmarking in Orissa to follow. It's the first university-based technology business incubator, which was established in 2009 with the support in the KIT deemed to be university as a not-for-profit organization, and with the support of course, NSTDB. And now it's one of the largest incubators in the country. Later, in 2012, Incubator was supported by BIRAC under the BioNS scheme to amplify the biotech innovation ecosystem. So sincere thanks to BIRAC for always supporting us. Now, what clearly distinguishes KTBI from other incubators has on the size, with a vision to augment, to do something in this part of state, India, to do something in Orissa, to do something for Northeast, and provide wings to the young innovators who have the dare to dream big. And KTBI supported knowledge network comprises of over 200 startups. A lot of them have got follow-on funding. We have now a regional center of BIRAC, or this is the BRTC Center, to promote innovation across Northeast specifically. Almost 67 patents have been generated. We have a strong pool of mentors and experts like Dr. Sriram Raghavan, who mentors the startups regularly. Then we have a national and international partners, which also has helped us in building a strong foundation for budding biotech entrepreneurs in India and outside. So additionally, KTBI is implementing partner of various startup funding schemes that Dr. Devan showed, particularly for BIRAC and other funding agencies also like DSD, MSME, Nidhi. So both central and state industry organizations, international bilateral agencies also to support product development across the complete value chain, starting from proof of concept to validation to scale up to until commercialization. So last year, BIRAC, in collaboration with Regional Center, KTBI, was established for the BRTC Center, which I already discussed. And for that, we have got a huge response from the young Northeast innovators. We got for the first time almost close to 180 applications from the big Northeast call. So we also have the TC centers, the recently technology transfer. We have a Spur Center to support innovative ideas in terms of social impact. So all the ingredients are in place to provide an enabling ecosystem to support innovation. So we have close to almost 90 innovative projects across the country who are working on biotech innovation, such as biomedical devices, agriculture, molecular therapeutics, hybrid enzymes. Even in agriculture, some are developing a seed drill for agriculture, novel wound care solutions, so many others. So I'll just give an overview of a few of the examples of innovators who had come to KTBI with a mindset to plunge into this difficult part of entrepreneurship and innovation, and how KTBI supported in crystallizing the ideas, helping them in getting the funding, helping in product development, and also connected them to various stakeholders, for follow-on funding. So let us come with this example, Dr. Sumona Mishra. So unfortunately, she was suffering with a induced hypertensive disorder called preeclampsia. And because of that, she had to undergo a lot of complications in her pregnancy. Then she realized that there is no such diagnostic kit available for this disease. And then she made up her mind that yes, this is a time where I have to use my scientific knowledge to do something in this field. A lot of mothers who are facing this problem, and this is the time, this is the need of the hour. So she approached to us and we supported her to get this BIRAC SIP fellowship program, and through which she went for the 6 months immersion. She went to a lot of hospitals, gynecologists, met a lot of patients, and then identified, crystallized her problem statement. And she got the big grant. She developed the product, and now it's no more looking back. She has a lot of accolades and awards. Similarly, Saika Lushra Sagabitra, she is developing a technology-driven molecular drilling device which can deliver the anti-cancer medicines to the affected cells, the precision medicine. So she came as a researcher. She said, but yes, I want to be a faculty, but I feel that I'll be best if I do something on my own. She always had this entrepreneurial mindset. And then we said that, yes, we need to support her. So she was strong technically, of course, but she needed assistance in regulatory, she needed assistance in funding, and also mentorship support. So then her entrepreneurial journey started from 2016 to 2019. She is a tremendous change. She is now a known name in the startup innovation ecosystem. Similarly, Sabir was recently MTech graduate, and he approached us with an idea that is to develop a formulation to stop bleed. She got a Nidhi Prayas grant, then got a big funding, and now he has launched his product and it's developed a lot of funding. So I think I've exhausted my team. I just want to say that the most important lesson that I've learned being associated with an incubator ecosystem for the past 6 years is to able to work in a group and build the right team. Group energy, believe me, has a very powerful impact on a way of thinking any innovative idea. And that holds good for including entrepreneurs also. So I end here by saying that one cannot solve all the problems, but of course one can make the best practice example. Let's build a conducive ecosystem. Let's bring together all stakeholders and find innovative solutions against challenging social and health issues. Thank you all.
So thank you, Thank you, Dr. Namrata. I think you really said everything can happen within an incubator if all they build up a right team.
Absolutely, sir. Completely.
Thank you. So any questions for Dr. Namrata?
And please reach out to me. I'm just posting my email ID, TBI ID, so you can feel free to reach out to me for any support needed.
So Namrata, somebody is writing, can we approach you with an idea? Of course.
Absolutely. Absolutely. And the good thing is that now the BIRAC BIG call is open and the deadline has been extended. It's now 15th of March. So you have ample time. We conduct a lot of grant writing workshops to help you to make a competing grant application. So definitely, please write to me. We will definitely help you out in every possible way.
Good. We do not. Can we have your email ID, please?
Yes, of course. I'm just doing that.
So, thank you, Dr. Namrata. So Namrata spoke about the enabler ecosystem like TBIs, how they are helping many of us to build the enterprise.
I'm just posting my email ID.
Thank you. Yes.
Thank you, everyone. Thank you for patiently listening to me.
Good. So now I think is the so we have next speaker. Can I request others to mute? Yes. So the next speaker, Dr. Visakha Raina, and now she's a different example. So she's a faculty and became now entrepreneur. Dr. Visakha Raina is now currently working as associate professor, and she's the associate dean of academics at School of Biotech at KIT University. And she did her PhD in molecular microbiology with Professor Ruplal in University of Delhi in 2004. Then she joined IAWAG in Zurich for postdoc, and she came back to KIT, joined School of Biotechnology in 2007. And a couple years back, she is always promoting different kind of work with the students, which are more innovation driven. So she picked up one of it and founded a company called Kriya Food and Beverages, also one of her master students, the major shareholder. And now I request Dr. Visakha to give a perspective of the microbial enzyme making safe food. Dr. Visakha, you have only 20 minutes.
Thank you very much for, am I audible?
Yes, very much.
Okay, thank you very much for this introduction. At the outset, I would like to thank the organizers, especially Dr. Ruplal, for being a guiding light throughout my academic and professional career. And I always look up to him for any advice whatsoever. So now I'm going to just share my screen and going to go off video. So are you able to see my screen? Is my
Please
screen
make
visible?
it a full slide.
Yeah, just a moment. Is it visible now?
Yes.
Okay, thank you. So I'm an associate professor at School of Biotechnology. There has been a journey from me being a professor, a faculty at a department, and then going on to entrepreneurship. So it has been not an easy road, because scientists love to do one tube to the next, and they love to do their experiments and love to publish papers. And making that shift from academics to entrepreneurship has been a very learning and an enriching experience. But always there is a doubt whether I can do it or not. And that's where these people from incubators, from BIRAC, from bioincubation, and all these, they handhold and help us to translate innovative ideas to enterprise. So let food be thy medicine and medicine be thy food. That Hippocrates told us, that you eat your food and you don't need to eat medicine. But there is a problem there. The problem is that nowadays the food which we are consuming, we don't know really what it is, what kinds of toxins and what kind of ingredients are actually added to enhance the flavor, the look, the taste, etc. So we are already aware of several food products in the market which have been in controversy because of the certain ingredients which have been found in food. And governments do play an important part in food safety, but ultimate responsibility lies with the food industry, who have to be responsible for how they are processing the food. So we know that when we treat food at very high temperatures, especially during food processing, there are a lot of things which are made. So basically there is a pipeline of where the food comes from, where it is grown agriculturally. Food processing takes place, packaging, there is point of sale, preparation, and finally it reaches to the consumers. And consumers have all the rights to know what they are actually consuming. So it's from farm to plate, are we having the right process to make the food safe? So we don't know whether the food we are consuming is the right medicine for us. So the food processing process is a complex process. Several pipelines are there, but a lot of products are added to it for giving it a nice shape, for making it commercially viable and attractive to the customers. So there are preservatives, additives, flavoring agents, coloring, antioxidants, and they have definite health effects, which we are all aware of. So what is the solution? So being a microbiologist, my first solution was to look at biocatalysts, which we get from microorganisms. And we have a lot of research which helps us to understand which are the microorganisms. So when we talk about food, by food, by training, I am an environmental biotechnologist, but food always interests me. I love food. And so my first preference was to do something in the food industry, which always I wanted to do. And in the food industry itself, we are using a lot of biocatalysts in different and various industries. So there my interest in food lied in baking. So I loved baked foods. I love to make them. I like to eat them. So my interest took me towards that. And when I started prodding into the types of enzymes which I used in the baking industry, and if I want to make baked foods, then what I would be requiring and what kind of processes would I be using? Would I be making a good product or not, etc. So that's where we, with the hand-holding of several agencies which provided us support, opened a startup which is Kriya Food and Beverages Private Limited. Our main vision and core principle was to design sustainable technologies to create affordable, innovative, nutrition-dense foods from local organic raw material, which is indigenous to the region where I'm living, with a no or a low preservative based strategy, because we are adding too many preservatives and many additives in the baking process also. So we established Kriya in a small laboratory, and we got a first grant from MSME, which was just 6.25 and lakhs, but it helped us to jumpstart our R&D and try to understand how to make different products and all. So everyone would think that baking is not very complicated. Everyone does it. Every city has a bakery, and it's a very common knowledge. But when you go into the chemistry and you go into the processing, there are so many things which people are unaware. For instance, the brown bread which you get into the market, it's not actually brown bread. Sometimes color is added to make it look brown. So we finally registered it in August 2015, and then only we started the entrepreneurial journey towards that. So basically, we look at several things. We look at food innovation, fortification, enzyme technology, which I'm going to talk about in my future slides, and alleviation of malnutrition by making safe foods, by creating innovative foodstuffs from raw material, which are indigenously found. And also we were looking at tribal empowerment, because the university I am, there is a tribal university which houses 30,000 tribal students. So I had a team, and I started my team with a master's student, Rahul Chatterjee, and he was my student, and he had the same ideas as I had. And we had the hand-holding, and we just started it. So I opened a company along with my master's student. And as today, I'm very proud to say that. So we have a 10,000 square feet manufacturing setup with the imported plant and machinery, and we got some funding. And as of today, since 2015, we make over 100 products with an annual revenue of rupees 3.2 CR. So we have more than 30 employees, and this is the team we have created with lots of product varieties. So what is the R&D we are doing? So it's just not we are looking at just a bakery and just doing business, but what exactly is the problem statement and what are we looking at? So earlier I told you that we are also looking at making safe foods. So we are an enzyme-based processing aid for reduction of acrylamide in different thermally processed food products. So in the baking industry, when foods are heated at a very high temperature, there is the production of a toxic compound which is called as acrylamide, which is a carcinogen, and it is produced during a reaction which is called as the Maillard reaction. So when proteins and glucose combine together, which is present in food and heated at very high temperature, it results in browning. Like tandoori chicken or toasting, and this is generally the common reaction which you see in baked foods. So this process also results in the formation of this toxic compound called acrylamide. So if you look at the risk assessment of acrylamide, we almost consume about 45 to 50% of it daily, and 300 times higher than the permissible limit. Also, in India, a lot of food products are kept in cold store, and that's called as the cold sweetening process, which also results in formation of acrylamide, which can be also present in with different tissues within the body. Children are the most exposed to it, and a lot of food products made from the food processing industry, which we every day on an everyday basis we consume, do contain very high concentrations of acrylamide. So the limits of acrylamide in water and food are actually very low, the recommended limits. So basically the consequences are that it is mutagenic, neurotoxic and genotoxic. So several agencies around the world, they recommend not having it during the food production process. So when we try to do a hazard analysis from different types of bread and bakery products from the market which is available, we found also there were news reports which are off and on coming when the food agencies are testing the food products. We find that bakery foods generally have addition of food improvers, which improve the look of the food. And the most common additives are potassium bromate and iodate, which are used in industrial applications. So we decided to do a food processing in the bakery industry, in my bakery industry, so that we don't use any of these improvers and produce the food products without them, despite having the taste and everything. So the innovation here we are looking at is the development of an enzyme product, which we are calling as asparginase enzyme, or denoting with ASN ASK. It's an acryl aid, and it is capable of removing acrylamide in the processed food which is being made, that is the baked food. So generally, L-asparginase is known to be a medicament. This enzyme is also used for treating childhood leukemia, and it is a known treatment. But here we are looking at asparginase from microorganism to be used in food manufacturing process. And here you can see that asparginase enzyme, which when added to the food containing sugars, it reduces the formation of acrylamide and the Maillard reaction is stopped. So there is formation of aspartic acid instead of acrylamide. So this innovation is, the USP is that it is cheap, it's affordable, and it's very little used during food processing, will not change the food process, and also does not change any kind of taste or organoleptic properties of the food also. So the basic reaction is that Maillard reaction will not happen. So I will show you a video at the end of the presentation. So what exactly we are looking at? We are looking at a strategy to make a useful enzyme for industrial biopurpose by choosing an engineering strategy. There are two ways: either you use rational design or directed evolution. And we have tried to do rational design by doing side direct mutagenesis and develop a mutant variant of a gene and transform protein, express it, purify, and then use it for the industrial process. So basically we are taking an asparginase gene and doing this entire process with this enzyme. So we have taken E. coli K12, which is generally regarded as safe organism. And from that we have tried to take the gene asparginase, then mutate it. And after mutation, we got several variants. And those variants are then checked for expression and purification of the asparginase enzyme. So all that process and purification was done. It's a 34.6 kilodalton protein, and the purification was achieved. And after purification, we do the rest of the temperature and pH stability studies. And studies showed that the highest results were between 50 to 60 degrees temperature, which is optimum for usage in food. So you have to always keep in mind that what are the temperatures you are looking at when you are adding the enzyme in the food process, when the food process is taking place in the industry. Similarly, the pH, the mutant which was taken, the pH studies showed that the activity of the enzyme is good at pH 7, and it should be more active in alkaline pH, which makes it more suitable for industrial applications. And enzyme kinetics also showed that the mutant was more efficient than the wild type in enzyme speed, and the Km value showed less values than the wild type strain. So again, it shows the potential of this particular enzyme towards the industrial scenario. So these comparative studies, we estimated that the variants which we have obtained, whether they are having good potential for reduction of acrylamide in food. So after this, we do comparative studies on incubation time, etc., and then optimization of how much the acrylamide which is present in food during high temperature is being reduced. So there are several steps in optimization of that protocol, and the analysis is done by LCMS. And acrylamide reduction was done in two types of products. One was potato-based products. So potato-based products are consumed extensively, like chips, French fries, etc., at home also, and in industrial, chips are the favorite, and French fries are very favorite. And the second is dough-based products, which are most commonly used in the bakery industries. So we found that the mutant varieties which were not treated with asparginase enzymes, they were having more reduction in the formation of acrylamide in the potato-based products as well as in the dough-based products. And also we try to do this analysis by testing them in buffer and also in water. So in the industry, when we take enzyme formulations and we are preparing the dough for baking, so just before the dough is being prepared, we generally don't use buffer. We generally use water. So we are not going to use buffer in our industrial process. That would be expensive and not feasible. But we are going to do it in water. So we have to test it whether acrylamide reduction takes place in water. And we were able to show that the detection in the final food samples, the acrylamide could not be detected. And this was tested on French fries before treatment and after treatment, and the reduction was possible. So experiments on dosage and the efficiency of reduction was also determined in potato-based products as well as in the dough-based products. We got good efficiency in potato-based products, and very little amount is required per gram of sample, which will cause 95% of reduction. So for information, during the baking process, we already use several enzymes during the steps of baking. That is in the initial preparation stage of the dough. So at that stage, this enzyme can also be added. So once you are sure that the enzyme which you have tested and validated whether the reduction is taking place, then can be used for the processing and for scale up. So in the industrial processing, whether it is working or not. So here at the first stage itself, this enzyme formulation can be added. So rest of the steps do not change because industrial processes they don't want to change too much. So this was shown for industrial processing. And once it was shown that the reduction of acrylamide was good enough, was 95 to 98% in the industrial processes, then we decided to scale up the enzyme formulation. And for scale up, we are doing it in lab scale biofermenter facilities in TBI. And for this, we are able to show that the sufficient amount of protein is purified, and using this particular purified enzyme for our subsequent studies for toxicity analysis, as well as other analysis for the validation of the enzyme formulation to be used actually in the baking process. So we have several plans for initially for proof of concept and for upscaling of this particular enzyme. But what are the difficult processes which are coming ahead are more of the validation, regulations, and then finally licensing. We have filed a provisional patent for this, and looking at the go-to-market strategy, which is, of course, for as a faculty, for me, very new terms. And I'm also trying to learn what are the different processes, and all these terminologies I'm trying to learn. So at this stage also, there is a lot to learn in this arena. So in summary, we were able to purify and downstream process and optimize the affordable process of purification of this enzyme to 95%. And it was thermostable, it was working in a high pH range, with high unit activity, and the quantitation and the reduction of the acrylamide was validated in potato and dough-based products without any changes or any changes in the taste. So basically, we are looking at when we are trying to work in the company, we are not just making baked food products. We are focusing on whether the food products which we are going to make, they are chemical-free, preservative-free, and they should be innovative. And whatever we are claiming that we are making safe foods, it should be actually there and not just say it verbally. So we are looking at several innovative products from different indigenously available flours. For example, I am in Odisha, and here millets or ragi is very commonly available, a flour, and it is a very high nutrition flour. And making baked foods out of them, these products are generally not available in the market. And these are gluten-free, having a longer shelf life, and using a chemical-free composite flour system. We are also trying to introduce chemical technology for toxin reduction so that safe foods can be made. So we have other social responsibilities also. We are trying to use these products for state nutrition programs, for midday meal schemes in schools, where high density nutrition snacks can be made for even household consumption or even at university level. And also there is social responsibility to create jobs for women, for children, giving them good nutrition. So malnutrition reduction can be targeted, and also for employment of tribal women whose concentration is very high in the area where I am. So we are also looking at various variants of this acrylate. For example, acrylate B variant is there, and acrylate P variant we are also looking at, which is one is the lyophilized version for industrial use, and the other is for potato and French fries based applications, and also some complex applications for different types of refined flours and different indigenously available flours. So we are also looking at developing these formulations. So currently our traction, we are on a daily basis feeding 1.5 lakh consumers daily. So even in the lockdown, these group of present customers were not operating, but we had so many people looking for baked products at their home, and we were delivering food to them. And that is one satisfaction which you get when you feed someone who's hungry, and you have the satisfaction you are giving them a safe food and not something which is not having any kind of nutrition. So all this would not have been possible if we were not having the support from funding agencies. And we have received a lot of support from BIRAC. That was the first big grant we got. Big means BIG also, and also the huge grant we got first time, and we were able to develop and do all these validation studies during that process. And currently we are upscaling with the Indian Oil Startup Fund. So these are some pictures from the gallery. We have from the manufacturing unit. So we have a team of faculty, we have team of management people, and this is my PhD scholar who was my master student and now is doing PhD. But he's really an entrepreneur, and he has taken this entrepreneurial journey head on. And I feel proud to say that I have tried to be an entrepreneur, and now I have also mentored someone to be an entrepreneur. So that gives me a lot of satisfaction. So there are acknowledgements for our founder of my university, who has also supported us in every possible way in helping us set up the manufacturing facility. So I'm always thankful to him. And a lot of funding agencies who have given us support, like NSTDB, BIRAC, Startup Odisha Fund, MSME, angel investors have been there, and Startup Indian Oil Startup Fund, who have always supported us. So thank you very much. And I would like to play a small video, which is just about a minute.
Did you know that presence of...
Is it audible?
Yes, very much audible, Mr.
food toxin acrylamide in our daily diet is a growing health concern globally? Strict norms have been laid down by the food regulatory bodies like FDA and EFSA to reduce the acrylamide content in the product. What is acrylamide? Acrylamide is a WHO recognized class 2A carcinogenic compound which is synthesized during cooking at high temperatures while frying, grilling, baking, toasting and roasting. The lack of awareness amongst consumers and their regular consumption of these hazardous processed products like biscuit, coffee, cereal, bread leads to these serious health diseases in all age groups, but significantly affecting infants. So what is the solution? Acrylate is a one-stop, award-winning, cost-effective solution capable of reducing acrylamide formation in food by up to 95%. So how does this work? Adding acrylate to the food production process converts the amino acid asparagine even before the heating process takes place. So, no acrylamide can be formed in the final product. Acrylate can be used over a wide range of products. No significant changes need to be made to your recipe or production process. Taste, appearance and texture of the final product stay unaffected, and acrylate does not require labeling. Pilot scale trials using acrylate and its variants are being conducted at Kriya Foods manufacturing facility. We at Kriya make food safer and help you choose the right bite.
Thank you. Thank you, Dr. Visakha. That was really great to speak about your journey from science to an entrepreneur. I think there's one question for you in the chat box. There's a real life application of microbe. Really great work. So the question is, what is the acrylamide contamination in processed food products elsewhere other than India? It means,
yeah.
Please go ahead.
Acrylamide is a known food toxicant, and it has been recognized by EU and US FDA. So whenever food products are manufactured in other countries other than India, so in India currently the testing of food products for acrylamide is not done with that intention that we have to check whether it is present in food or not. But in other countries, there is very strict regulation, and the content of acrylamide in food, that is about 1000 ppb concentrations, that's the limit. It should not be present beyond. So more than that. So it's a contaminant in food processes, which is very strictly regulated in other countries, but not very strictly regulated in India. And that's the reason why different food products in India, we continuously have higher levels, about 45% acrylamide contamination. And the highest contamination of acrylamide is found in street food, the food which you consume on the street, like chaat or golgappa, all that burgers, because these people, they are using oil which they are procuring from restaurants, which is already burnt oil. So this burnt oil has been heated at very high temperatures repeatedly, which results in excessive acrylamide formation already. And then they reuse it again to make street food. So that's the way they try to cut corners for not purchasing good quality oil. And that is where the acrylamide contamination enters into our health systems. So you may not be smoking, you may not be drinking, you may not be having any other bad habits, but the food which you are consuming is containing the toxicant. So at present in India, it needs a lot of, the FSSAI is now regulating the acrylamide content, which is now being formed and especially available in foodstuffs which are available in street foods.
Thank you. Thank you, Dr.
Mrityunjay,
Yes.
Can I ask one question? Yeah.
Yes, sir.
Yes,
Hello.
Yes, sir.
Congratulations, Vishaka, moving from also doing academics simultaneously and becoming an entrepreneur. What are the steps that you need to resolve in order to put now your technology to commercial use?
So at present, we are doing biofermenter studies for scale up. So we are at this stage right now. And so already we have already a food setup is there where we are selling our food products. This
Vishaka, you are unmute.
No, I think Vishaka, yes, once more, because it was not audible.
Okay. So I'm saying that at present, we have two things. We have, is it audible?
Yes.
Yeah.
Okay, so at present, I have two things. One is our company, which is making products. And for that, we have to use a different strategy for marketing. We have retail. We are selling our products in university and around the area, around the university. But when we are talking about enzyme, so currently our strategy is first to validate the production of the enzyme at different specifications, and then go for toxicity studies and then market strategy to sell this for prospective customers, that is different industries, etc. And that would be a different strategy for marketing, because there are competition in that arena also.
Thank you.
Thank
So
you very much.
We are using the purified enzyme at the moment.
Thank you, Dr. Vishaka. I think somebody is asking your email ID. Please type it in the chat box.
Okay.
Thanks,
Okay,
everyone.
sure.
So now
Thank you very much.
Yes. So Vishaka, please feed everybody good, safe food, and you make more money. And let us be all safe out from the food ecosystem.
Thank you.
So let us move to the next speaker, Dr. Utkarsh Sood. Utkarsh is, I know Utkarsh as a colleague from our laboratory from Professor Ruplal, PhD from Ruplal's lab, and currently the RA at TERI, New Delhi, and he's the co-founder of FixGen Private Limited. Very, very enterprising guy. And I request Utkarsh to speak about his experience with the start of FixGen. Utkarsh, very brief maybe.
Yes, sir. Thank you, sir, for such a lovely introduction. So I'm just sharing my slides. All right. Good evening, everyone. So I am Utkarsh, and I'm here to present the 3-year-long journey of our startup, which is named as FixGen Private Limited. So our startup is basically dealing in 3 regions and 3 domains. So we are doing academics, we are doing research, and we are also doing business. So moving forward, the basic origin of our startup, or the leads that we got in during our PhD days, our lab was working on 2 aspects, major aspects. One was genomics, and metagenomics. And the second was production of a rifamycin analog that is known as 24-desmethyl-rifamycin. And during our PhD days, I got associated with 9 metagenomics projects and 25 genomic projects. So our lab has an experience at that time also to do this type of studies. So I learned these techniques during my PhD days. And also I was working on a project where we were doing certain manipulation in the genetic architecture of a bacterium, from which we could produce 24-desmethyl-rifamycin. So we had 2 leads, but after those 2 leads, for commercialization, we needed a startup or a company who can work in this aspect. So as Government of India was promoting entrepreneurship, as we all know, so we decided to incorporate a company. So our company, named as FixGen Private Limited, got incorporated on 30th January 2018, and with a share capital of rupees 10 lakhs. So as I have already discussed, and that we are working in 3 domains: academics, research, and commercial. So I will be dealing each domain one by one. So the first domain, which is research. So we applied for this big grant in association with KIT TBI and under the guidance of Swar sir and Namrata Mishra ma'am. And during our experience, and when we applied this, so we wanted to upscale or scale up the commercial and commercialize 24-desmethyl-rifamycin, which is active against the major first-line treatment drug rifampicin and the resistant genes of Mycobacterium tuberculosis. So this is the producer bacterium which produces normal rifamycin, and after genetic manipulation, so this is the structure of rifamycin B, where you can see that at the 24th position there's a methyl group present here. So we genetically modified this organism, and after using a combinatorial biosynthesis approach, we removed this methyl group from the 24th position. And after the removal of this 24th methyl group, basically what happens is the binding of this molecule was found to be more adequate to the beta subunit of RNA polymerase, leading to effective killing of even the resistant strains of MTB. So these are the 3 objectives on which we are working. We are working to scale up the production of 24-desmethyl-rifamycin using fermenters. So we have proposed to first standardize the growth conditions as well as the growth media in a 2-liter fermenter. And then we have projected to go up to a 10-liter fermentation model. And after we achieve a good yield, then purification is also very important. So we have also proposed to devise or to standardize a protocol to purify 24-desmethyl-rifamycin from the fermentation board. And this 24-desmethyl-rifamycin is when converted to its further analogs, which are known as 24-desmethyl-rifampicin and rifamycin S, is found to be more, we can say more good molecule. So we want to convert this rifamycin to rifamycin S and rifampicin. So this is the methodology that we are doing, and we have already standardized all these conditions in the flask model. So from the flask model, we are moving to a lab scale fermenter, which has a working volume of 2 liters. And from these 2 liters working volume fermenter, we are projecting to a 10-liter fermentation. So we have just started on 20th February 2020. So we have just started to standardize the growth conditions as well as the growth of this bacteria and rifamycin production in this short fermenter. So these are certain side by side. We are also doing some exercise or some standardization of protocols using this organism in flask. So what we are doing, we are generating this molecule and we are standardize our protocol, and that if we start to get more and more amount of this molecule, how we standardize it and how we purify it. So this molecule is active against Staphylococcus aureus, and also we have tested it with the MTB as well. So in our setup, now what we are doing, we are just standardizing this step of purification. As you can see that we have set up a column chromatography, and from this column chromatography, we are obtaining different fractions. And as you can see in DCO, there are multiple impurities that we can say that there are pigments that the organism produces. And when we purify this pigment using this column chromatography, we are getting single bands as well. So the fraction 19 and 20 listed on the TLC plate can be seen to only have purified 24-desmethyl-rifamycin B. And this is a first project that we are working with the BIRAC grant, with the support of the BIRAC grant. There is another project as well on the other aspect, that is genomics and metagenomics. So FixGen is also providing its services to a project supported by National Mission for Clean Ganga, where we are partnering with CSIR NEERI, Accelris, MNIT, Charuta University, and we will be doing a GIS-based mapping of this microbial diversity across the river Ganges. And my coordinate or my colleague from FixGen, Vipin Gupta, will be giving more details about this project. So the commercial branch of FixGen, we are also associated with BGI. BGI is Beijing Genomic Institute, and we are their channel partner in India, and we provide all the types of sequencing services, whether it is DNA sequencing, RNA sequencing, or any customized sequencing project that our customer wants to initiate. So these were the two aspects. One was research, second was the commercial part, and third is academic. So usually what startups does, they don't pursue their passion of research. So what we are also doing, we are still continuing our passion of research, and we are still publishing certain papers. So these are a certain best publications from our group during the last 3 years, as you can see. So the take-home message for FixGen Private Limited is that we are working on major 4 domains. First is scale-up of rifamycin analog. Second is the genomic and metagenomic analysis and the sequencing services. And third is the training services. We also provide training services. We organize workshops and symposium for undergraduate and postgraduate students to give them a hands-on experience of how these genomics and metagenomics tools work. And we also provide technical services to our customer where they want to design their experiment setup in terms of metagenomics and genomics. So I would end my presentation with acknowledgement. So I would first like to acknowledge Professor Ruplal sir for giving us this platform to work and for showing us the path. And secondly, and most importantly, I would like to acknowledge Mrityunjay Swar sir, Dr. Namrata Mishra ma'am, and the entire KIT TBI team. As per their guidance, we moved, and we were, you can say, we were fortunate enough to get this big grant. BIRAC DBT, Government of India, for giving us the grant, and the National Mission for Clean Ganga for the second project. And these are my team members: Dr. Roshan Kumar, myself, Vipin Gupta, Princey, and Mona. So we are working in different domains. We have different expertise. So as a team, we are initiating both these projects. And with this, thank you very much.
Thank you, Utkarsh. I think well said, because if you look at your team, all are science-driven, and the mentor, Professor Ruplal, he also thought that let us build something out of my laboratory activity. They say, well said. I think right now the team is building up, and there's energy in the team. There's mentorship from the top. So I think definitely all the best for your company, and make it that one of the best successful company. Why BGI? Why not FixGen? The complete solution provider in India. Thank you, Utkarsh. I move to the next speaker, Dr. Bipin. Also, I know him from the same lab, Dr. Ruplal's lab, and he did a PhD from his laboratory, and right now the RA in a project. And I'll be requesting Dr. Bipin to give his journey as an entrepreneur from academia to industry and research. Bipin.
Thank you so much, sir. I hope my slides are visible.
Perfectly visible. Please make it full screen. Yes.
Myself, Dr. Vipin Gupta, and working as a director of FixGen, and also working as an RA in a project. And so I would like to brief about my journey as an entrepreneur from academia, industry, and research. As my colleague has already been Utkarsh has mentioned about FixGen and what areas are dealing. So major domains of FixGen are academics, research, and commercial. And with KIT TBI and their guidance, Professor Mrityunjay Swar, and he's also guiding us. We are taking good care of commercial activities. But in the research, FixGen is also active. And it is a good news to share that FixGen being included on a multi-million project where GIS-based mapping of microbial diversity across the Ganges for ecosystem services have been taken care. And it's a only research project that is going on nowadays over the Ganga. And being a private company, we have been indulged, we have been on board for this project to provide our services. So briefly, we all know that Ganga is the most sacred and holiest river in India. And we all know there are lots of rare and undiscovered species. And after giving so much of ecological and ecosystem services, you won't believe that Ganga is still one of the most polluted river in the world. And being this as a big challenge and a big defame for India. So the whole stretch of Ganga is 2,500 kilometers. It moves from Alaknanda to Diamond Harbour, and it crosses almost 4 major states of India and with 50 major cities, and lots of pollution from these cities just literally dumped to this river. and including from Ganga, Kanpur, including from Agra, Kanpur, Allahabad, and Haridwar. So these challenges were there, which made this project to happen. And National Mission for Clean Ganga framed out following objectives in the meeting, and these objectives were discussed where FICGen Private Limited gave the opinion that out of these objectives we can take up these 3 objectives, where bioinformatic analysis and metagenomic analysis we can take care, and we can design a pipeline where all these multiple objectives we can cover at once. So looking at the sample size, the type of sampling that they have framed, you see there the complete stretch of Ganga is divided into 7 segments. And from each segment, 3 locations, and each 3 locations, 3 different samples of microbial mat, water, and sediment to collected. And you see the only study a good study that have been conducted so far could contain only 26 samples. But this study we have planned to collect 403 samples from each sampling. And with this type of sampling, when we look at the sampling, we designed a pipeline. We designed a pipeline to cover all the major objectives for microgenomic, microbial, and bioinformatic analysis. And the pipeline we designed for National Mission for Clean Ganga could also do the assembly, diversity analysis, heavy metal analysis, AMR profiling, virome analysis, and lots of other analysis could be done by this pipeline. When in the meeting for National Mission for Clean Ganga, we designed this pipeline and we generate some result from previously available data. So it was found that our pipeline gives good result, which were not known earlier. Few of the genes that were not known earlier from the previously available data, we could figure out those important microbial communities. Also, our pipeline could manage to identify some of the indicators from animal gut, human gut, pathogen, and wastewater marker, which were difficult to identify earlier, previously tools. Thus, looking all these suggestions, when we put them all the suggestions in the meeting, it was approved that the pipeline is able to generate AMR profiling, heavy metal analysis, and lots of other analysis that were required to complete this project. And after looking into this project, special segment was also added for generating E. coli stereotyping. They wanted to know type of E. coli coming from human gut or animal gut. So we also added this component into our pipeline, so that we can have an idea of that how much of the contamination of E. coli is coming from animal during the lots of public meeting or gathering like Kumbh and other meetings happened in Allahabad and Prayagraj. And when we propose, we give all the result to National Mission for Clean Ganga, and we prove them that we are ready. Then only we were allowed to go for sampling. Our team with Professor Rupal, myself, Vipin, Utkarsh, Roshan, and other team members are Mona, Prince, Najar are there. Me and Utkarsh took this liberty and we went to the sampling site and we covered this area from just after lockdown went unlocked. From 15 to 30 November, we could cover this Devprayag and Haridwar region. We collected all those samples, and it was a great journey for us. And the sampling have been done and the data is under process, and we will soon share the result. And I would also acknowledge Namami Gange National for providing this opportunity, FICGen Private Limited, to get us on board and to give us services. CSI Navy to coordinate this project, Charusat University, MNIT Allahabad, and Excelis for all these big shots coming together and take up this major problem. Thank you. Our motto, the FICGen has a motto that you discover, we sequence. This is the contact detail. Our website is www.ficgen.com. We are constantly updating our website. These are contact details for me and Utkarsh. You can email us. You can give us your query. And if you want to give us any suggestion, you are also welcome. Thank you so much, sir. Over to you.
Thank you, Dr. Bipin. I think really is just very crisp one, and you've been doing great job. Keep on doing a great job. And I think any questions to both of them? To Dr. Utkarsh, Dr. Bipin, from any one of you? Because I see 114 members. I don't know, they are still awake or they're still sleeping. But any questions for them? They're young people, they've done PhD, postdoc, now creating enterprise. What best you can expect from AMI conference? I think, thank you. Okay, so I think the chat is going on, but not about the questions. Thank you, Bipin. Thank you, Utkarsh, for such a wonderful talk.
Thank you, sir.
I think what I can tell you a couple of things here. For all of you, because I'm in this game. I've been learning every day, sitting in KIT University in TBI. So we have a lot of things to be done, and researchers like us have to really come forward. So when we run a laboratory, of course, we are writing projects to get our publications, take students, PhD, but build some of the skills among students to think differently. That is the most important message I want to give to all the audience here in AMI conference. If some of you think differently like Son Chaudhary did, Visakha did, from scientific perspective, what Bipin did, Professor Ruplal, of course, he has already done so many things, and Utkarsh did. Can some of you create this mentality among your students? Or can some of the students like Rahul Chatterjee, what Visakha Raina mentioned about, the student who is a topper of MSc program and was offered for PhD in Switzerland or in Japan. He said, no, I want to do it. I want to build something out of my biotech education. This is the temperament you want to see in some of the, of course, everybody cannot do it, but can this platform like in AMI, when we are talking, like Dr. Manish said, money is not a problem. Siram said, I can really create enterprise, help you. Visakha said, all can happen here because my students are great, laboratory is there, I have brain, I have got human resources, funding is there. And Visakha also forgot to mention that being a lady from Srinagar, from Delhi, and she is now in Odisha, she got the Best Odisha Citizen Award with a 10 lakhs of cash award from the minister. So what else you can think as a scientist? So this is what all things can happen. You have to really form a right kind of team, and you all have to be coachable. When I talk to Utkarsh or BP, they're very coachable. They first listen to you. So if you really want to get into this innovation-led enterprise creation or startups, first of all, the first fundamental rule is that all of you should be coachable. You have to listen to people, then take call. So enterprise technology cannot happen from one individual. It has to be a group of individuals from science, from regulatory, from license, from you talked about FSSI, NABL, from QCI. Every year you have to go here and there. So with that, I can conclude this saying that India is now rising and shining. Be a part of it. Don't be a classical person in science that only publishing paper that you must do, otherwise no promotions, no accolades. Beyond that, you can contribute 20% of your time. Pick up your students and take them to this journey of innovations. I'm not saying everybody should build enterprise, but can you build up this entrepreneurial skill set among students? I'm very happy being the, when I School of Biotechnology at KIT University, most of my students are very, very enterprising. They take up fellowships called as innovation fellowships. The SIF fellowships, they don't take CSR and all fellowship. They're also taking this kind of fellowship and taking some of the interesting ideas into next level. And we play as a mentor for their initiatives. So with this, I thank everybody. Special thanks to Professor Ruplal, who has been my mentor, guru all the time. Still we share all the time what you can do in future. And let me finish, sir. I thank everybody. I know Sukanya Lal Madam also there. She is writing. Thanks, Sukanya Madam, hearing all of us, all my members, all AMI people, and everybody, Yogendra Singh sir. Thanks to my juniors, seniors, my mentors, everybody. Thanks, everybody. Yes, sir. My job is done.
Can I just make one comment now? Yeah, I'm not a mentor now. You all are becoming my mentors. And in fact,
that's a joke.
Very pleasing moment to see that yeah, no, it's not a joke. You all are providing now employment to so many. That is the need of the nation. Thank you for your encouraging words, and I'm sure you will continue doing all this work with the great.
Hello, Mrityunjay.
Yes, sir.
That you have done great job. So congratulations.
Oh, thank you, sir. I see VC Kalia sir also putting thumbs up. Thank you, sir. I think all are here. I didn't know all of you are here in this platform. I thought only Ruplal sir is there. Let me tell the audience, Yogendra Singh sir, VC Kalia sir has been always our mentor. I think they have always shared several platforms where they have been telling us do something differently. Of course, Ruplal sir is my mentor. Even Banwari Lal sir, I saw him in between. He asked the questions. These people are all in AMI platform. I didn't know about it. They have all been a motivator, and I have been inspired always by them. When I go to Banwari Lal sir, chamber, he talks about, but you see how am I building this technology, the oil japper, now getting into different business platforms. So this is what we want to hear from many of us, many of you. Thank you, sir. Thank you. Thanks for all the blessings.
Thank you. One statement I would like to make. Dr. Ruplal said very perfectly that now you are the mentor. You have the responsibility from Dr. Ruplal that now the things are under your jurisdiction, and you have done excellent work in the last, as much as I know, at least 10 to 15 years I have been watching your activities. And we were very happy when last time we all visited you, that you were showing that 6-story building, and that Vishakha was planning to bring out a bakery. So I was very happy that day, and today I can see that it is happening. And so, as we have heard Dr. Banwari-Lal also beautifully, from one stage where he was asking money from DBT. So he had paid 100 crores of tax to the government of India, which means we scientists are not that bad. We are also doing our service. And maybe the previous generation was not that entrepreneur, but now I think with you and Birak, Dr. Diwan, you people are doing a good transformation to the science, which this country has been struggling to take to a very high level. But I think that the change is coming, and you are the one people who have really taken it. And I say best of luck to Vishakha, to Dr. Banwari-Lal, and Dr. Vipin Gupta Utkarsh, and obviously Dr. Ruplal without saying anything. So he's the topmost boss, but he has done his duty beautifully. So I must congratulate all the team, and the AMI platform has done a very beautiful session, and Mrityunjay, you have done a beautiful job. So best of luck.
Thank you, sir. Thank you. Sir, as all of you are there, I thought I should share one more minute if I can take from all of you. Mr. President Yogendra sir, if you allow me one more minute. Sir, recently, yes, sir. Thank you. So recently, the PSA office to the Prime Minister is coming up with bigger schemes, and good that Bhubaneswar is now taking some of the lead, and the office has recognized us. So we are now incubator, and we are setting up something. Now you'll be very happy in due course of time. We are setting up a manufacturing hub, and we are taking the lead of setting up 100 acres of land, and all are scientists now. We are all, you can always call me scientist, even though I do other things, but all taking it up to build a manufacturing hub, which is driven by science. Now, recently, the PSA office has given us a Bhubaneswar City Knowledge Innovation Cluster, and we are setting it up, sir. So AIMS, and so not AIMS, IIT Bhubaneswar, Niger, KIIT. So we are taking the lead. I'm the chairman of the committee. And then the money came into picture. We will build up simple technology out of our local problems and building the right team. So IIT will design something, we'll get into something else, manufacturing will happen. And government is saying, I'll provide you land and buildings and other resources. It's a big budget, sir. I think Delhi cluster also coming up. Maybe some of you might be a member there. But Bhubaneswar is going to be one of the most vibrant because there is no bureaucracy, not much of leg pulling, and here is driven by us. So I think we want to bring more and more manufacturing so that Odisha will rise and shine with science and technology. So I'm very happy that getting PhD from Ruplal sir's lab, and always you crack jokes what should be done in future. He always says go and do something good job, and always I meet people like you, Kalia sir, always crack jokes, and Banwari-Lal sir also हमेशा चाय पे बड़ा अच्छे बात करते हैं. Yogendra sir always, when I go to his lab to do, I remember Western blot with somebody else, and he always cracks jokes. This is called, what do you call, the very authentic mentorship, that you provide freedom to do a good job. I think that's what KIT also provided. We're building it up. I think all of you are doing great jobs, sir. Keep on doing this. Mentor as many as you can. Thank you. And I'm very happy to be a part of science fraternity and doing something into incubation activities. And my team is very, very strong.
Congratulations from everybody, and thank you very much. Thank you.
Thank you. Best of luck.
Yeah.
Vishakha. Okay then.
Thank you, sir. Thank you. We're leaving now.
Yes. Thank you. Bye-bye.
Thank you, Mrityunjay sir, for your thought-provoking words and all the motivation. Special thanks for all the efforts you have taken to conduct this session. Without you, this session would not have been possible. I would also like to extend thanks to all the invited speakers on behalf of AMI and INSCR for accepting this invitation and delivering their talks. Now I would request Dr. Charu Dogra to begin with the next conference session.
Thank you, Princey. Thank you.
After such an inspiring session hosted by Professor Mrityunjay Swar, apprising everyone with newer avenues, newer perspectives, thank you so much for your thought-provoking words and all the motivation. Special thanks for all the efforts you took to conduct this session. We now begin with the conference next session on microbial pathogenesis and AMR. Dr. Anil Kaul, Johnson & Johnson, Belgium, and Dr. Namita Singh, Secretary AMI, will chair the session, and Dr. Utkarsh Sood from TERI, and Dr. Vipin Gupta, FixGen Private Limited, will co-chair the session. Welcome all. The first speaker of the session is Professor Yogendra Singh. Dr. Yogendra Singh is a professor at the Department of Zoology, University of Delhi. He worked as visiting scientist at NIH USA, supported by Rockefeller Career Development Award. He worked as a visiting scientist at Max Planck Institute, Munich, Germany, University of Freiburg, Germany, and National Institute of Allergy and Immunological Diseases at NIH. Over a period of more than 20 years, his group has worked on different aspects of Mycobacterium tuberculosis and Bacillus anthracis toxin biology. He held the chief scientist position at CSIR IGIB Delhi, India, before joining the University of Delhi. In his scientific career, he has published more than 130 articles and has several patents, including 2 technological transfers. I don't think that Dr. Vaising need any more introduction, and he is the man, along with Professor Ruplal, behind this conference, which has been wonderful. So over to you, Professor Vaising.
Thank you, Charu. Can you hear me? Charu, can you hear me?
Yes, sir.
Okay, let me put the slides. Can you see the slides?
Yes, sir, it's visible. Please go ahead.
Just a minute. So I'm sure that many of you know that many pathogens, and for example, those pathogens which causes cholera, tetanus, diphtheria, they all make toxins. And these toxins actually enter into the cells, and they have enzymatic activity, and eventually they kill the host if not treated in time. Today, I'm going to tell you about another factor which actually helps these pathogens to establish the disease. So my model I have taken is Bacillus anthracis. So I'm going to start with this pathogen and will take you to how that factor helps in the establishment of the disease, and together with toxin, eventually it kills. So anthrax is a disease which is caused by Bacillus anthracis. This bacteria is a sporulating bacteria, so makes spores, secretes toxin. As I said, many of pathogens do secrete toxin. It has a capsule, and this capsule actually prevents from the phagocytosis, so it helps in the establishment of the disease. Now the spores are very stable, even at very high temperature, and they can be aerosolized. So therefore, the spores are used, have been used actually in bioterrorism. And there is always a fear that these spores can be used in biowarfare. That is why our army always wants those remedies which can protect our army and even the general public also. If I tell you the pathogenesis, that how the disease happens, how the bacteria enters, so it is the spore which is inhaled, and those spores are inhaled by the animal or human. They are taken by the alveolar macrophages, and in the macrophages they germinate into the vegetative cell. I think you can see here the long vegetative cells, and these vegetative cells eventually make toxins. These are actually protein toxins, and these toxins bind to the receptor and enter into the cell, And as I said, many of these toxins have got enzymatic activity, so eventually they kill. I'm showing here what those toxins are. So there are three major proteins which make the anthrax toxin. Two of the protein have got enzymatic activity, which I have written here, adenylase, cyclase, and the protease. But alone, they cannot enter into the cell. Therefore, they need another vehicle, which is the third protein. So this blue color protein, which is called protective antigen, helps the remaining two proteins, and together they make edematoxin, which causes the swelling, and the lethal toxin, it has a protease activity. So I'm sure you can imagine if a protease goes into the cytosol, the cell will die. And this toxin actually, if, as I said, if not treated in time, it will be produced so much that the infected host is going to die. Now, what I showed you in the previous slide, that when this pathogen from a spore, when it germinates, it doesn't make a single cell like E. coli or other Bacillus species. Rather, it grows into a very long chain. And that's what I'm going to tell you today, that how these long chains are also kind of a virulence factor that help in the disease establishment and eventually.
So you're not audible. Sir, I think you are muted.
So should I go to the previous slide? Can you hear me?
Yes, sir, now we can. Please continue.
Okay. So about 4 years back, what was shown that if you produce the Bacillus anthracis chain of different length and you label them, or you can say bioluminescent they were, and if you inject in mice as shown here, so you can see here one was about 12 micrometer, the other was about 4 micrometer. And in the 12 micrometer, very quickly, those long chains actually accumulated in the lung, and those longer chains actually blocked the lung capillaries and they caused hypoxia, lung injury. So they, in a way, along with toxin, they are going to help in establishment of the disease as well as in the killing of the infected host. But what is not known until now is that what makes some of the pathogens single cell, some of them 2-3 cells, and some of them multiple cells, like I'm showing here. So in last 2 years, we have been trying to understand that what regulates the chain length of these pathogens. I'm just showing you the surface of the. So chain length is associated with the surface layer protein. It is called S layer. So I'm showing a figure which is taken from a Nature Review Microbiology, that in Bacillus anthracis, there are two proteins. One is surface area protein called SAP. Another is extractable antigen, EA1. And another protein is called BSLO, Bacillus, which has a surface layer O protein, which has got actually peptidoglycan hydrolase activity. And this BSLO is the one which is responsible for cleavage of the, when the two chains, when the bacteria divides, the peptidoglycan between those two bacteria are actually cleaved by this BSLO. And because it has got peptidoglycan hydrolase activity. So therefore, daughter cell separation is dependent on the amount of BSLO, which is Bacillus surface layer O. There are many types of BSL, so that's why BSL O type. It has got N-acetyl glucosaminidase activity. That's why it cleaves the peptidoglycan. Initially, we were not studying why these chains grow. Actually, we were trying to understand that what is the role of a kinase, which is PRKC, protein kinase C, you can say, in Bacillus anthracis, in pathogenesis, in biofilm formation, and many other studies. But what we noticed, before that, I want to tell you that what these kinases are actually. So there are two different kinds of kinases. which are very prominent, well known. They are responsible for sensing and signaling in bacteria. One set is known as two-component system. And the reason is that there are two proteins, one on the surface and another inside cytosol. So when a signal comes, this signal actually causes that signal induces a phosphorylation, autophosphorylation. These are actually histidine phosphorylation. And because phosphorylation happens, there are changes in the structure so that now it can interact with the response regulator, transfer the phosphate, and this regulator actually binds to multiple promoters in the genome so that it now makes large number of those proteins which are required to cope with this signal. So trying to say that outside signal conveys a message so that many genes are expressed, and those expressed genes actually cope with the situation. Now there is another set of kinases which are called serine, threonine, tyrosine kinases. Of course, there are arginine and many other kinases, but these are the major ones. So serine, threonine, tyrosine kinases, they actually also get signal. They get activated, but they work on the local level. They do not transcribe a large number of genes. Rather, they phosphorylate some proteins, and those proteins will do multiple jobs. They even do gene expression also, but not that many genes as the two-component system does. So therefore, basically, I want to say that phosphorylation and dephosphorylation regulates many things. That's what we are trying to understand, that what kind of activities are regulated by PRKC. Just to remind you that why phosphorylation does all these things. When a phosphate group is added, the negative charge is added, and that leads to a small structural changes, and that causes change in the interaction of the protein or activity of the protein. And that actually causes all the metabolic pathway and many other signaling gets converted into gene transcription of a gene. But what we noticed when we were trying to understand the role of the kinase, that PRKC, so we made a deleted strain of PRKC. And that deleted strain, when we saw in the microscope, we noticed the top one is here wild type, and the right side is actually deleted strain. So what we notice, if you look at the lower panel, you will notice that wild type has got very long chains, but the deleted strain, the chains are very short. Now we grew the bacteria from 1 hour to almost 30 hours. And we noticed that those chains where the kinase was deleted, they always grew as shorter chains throughout the growth cycle. And here you can see the top panel is actually wild type, and below that is deleted strain. So you can see how long here, and this happened actually throughout the growth cycle. Just to make the understanding better, we measured the chain length of the 100 chains. And you can see in this column here, which shows about the wild type, you can see that it is almost 4 to 5 times longer. So in here, you can see here it is pink color and the green color is wild type. So therefore it confirmed that PRKC has some kind of a role to play in the length of the chain. If the PRKC is not there, they cannot grow that long. We did other experiments also where we found out that the deleted strain actually doesn't grow as well as the wild type. But the more interesting phenomena we observed was that the deleted strain had got multiple septa. You can see here in the bottom picture, microscopy picture, you can see here that there are many septa here. You can see where you can see very clear septa into the wild type, but multiple septa into the PRKC deleted strain. It is more clear here. FM64 labeled cells fluoresce. So you can see here that the number of septa is much more into the deleted strain. Whereas in the wild type, you can see there are very little. And when we measured, we noticed that only occasionally there are multiple septa into the wild type, whereas the number of septa increases much more into those cells where PRKC, or you can say the kinase, is deleted. Question comes that why multiple septa? So I'm sure many of you know that when a bacteria has to divide, it produces a protein FTSZ. These FTSZ actually go to the center and make a ring-like structure. I have shown here in the green color. This is FTSZ ring. And this is the ring where other proteins are going to eventually join. And here, the separation of the cell will start into two. So two daughter cells will separate from where the FTSZ ring will be formed. So what we noticed that in those cells, this red color, where the kinase was deleted, much more FTSZ was produced. Now you can imagine if you make more FTSZ than what is required, it is going to make this kind of ring at many places. So we believe that increase in multi-septa is possibly due to the FTSZ mislocalization. We also noticed that the cell width and the cell septa, I have measured here in the graph, you can see here that wild type actually is larger, whereas the deleted strain is shorter. So the deletion of the kinase actually reduced the cell wall width and the septa thickness. Now this produced a hypothesis. We thought, why not test the hypothesis that if the cell wall is thin, then probably it will become more susceptible to antibiotics. So therefore, we tested the two antibiotics, actually three, but one was not the cell wall synthesis inhibitor. The two broad spectrum cephalosporins, cefepime and ceftazidimine, they inhibit the cell wall synthesis because they bind a protein which is responsible for the cell wall synthesis. They bind actually penicillin binding proteins. These are present in the bacteria and they help in linkage of the peptides. So when we tested, then we noticed that actually our hypothesis was true and they were more susceptible to these antibiotics. You can see in the red color, the kinase knocked out in the red color, blue color is wild type, and you can see that because it becomes more susceptible to these antibiotics. Now, dechaining in bacteria is actually related with, especially in bacillus, is related to SAP1. In the beginning, I said there are array proteins and extractable antigen which are present on the surface layer protein. The first slide and second slide, I think I showed you. So they are sequentially expressed and actually they go on the surface and in a way they restrict the presence of BSLO. They occupy all this space, you can say, and therefore BSLO doesn't deposit that much so that it can cleave the self. If there are more BSLO, it has a hydrolase activity, peptidoglycan hydrolase activity, so it will cleave the peptidoglycan to separate the two cells. But you can imagine that if the BSLO is actually less, then it is not going to cleave. That's what happens in the into the bacillus anthracis, that it go. And this, we tested this hypothesis where we measured these proteins and we noticed that the amount of this hydrolase is actually much more than the wild type. And that is the reason when it is expressed much more, it goes on the septum. These are deposited on the septum. So therefore, the anthracis, where in the wild type, where this was made much less, you can see the green ones. Where it is made much less, it is present in a small amount. Those SAP and even do not allow BSLO to deposit, but when it is expressed more. And that's what we found out, that when you knock out the kinase gene, actually the BSLO is made much more. So they are now able to enter on this on the septum, and now they can cleave. That's why the knockout strain grows into the short chains. So in conclusion, I will say what we have found out, that this kinase regulates many things, including the chaining phenotype in bacillus anthracis. And that happens by the increased expression of the that hydrolase, which actually cleaves the peptidoglycan. Of course, there are many other, as I said, this is a kinase, and it phosphorylates many other substrates, and those substrates play multiple roles. That's why we do see multi-septa formation, reduced cell wall thickness, and many other functions. Actually, before we did this work, it was known that this kinase actually is required, or you can say it helps in the germination of a spore. This paper, whatever data I have shown you actually is not published, but it is published only in bioarchives. So this is a very new data. Now we are trying to understand that how the expression of BSLO is upregulated. What is that transcription factor which upregulates the BSLO production? So with this, I think I will stop, and I will like to thank the funding agency SUB, for funding the project, for the JC Bose Fellowship Grant, and Institute of Eminence, University of Delhi recently has given some money, and we used that for this project. And of course, CSI and UGC always for the support to the students. Thank you very much. Charu, I am done.
Thank you, sir. Sir, we have some questions for you.
Yes, sure.
One is, why multiple septa created in BAS delta PRKC but not in the wild type?
So there's a protein called FTSZ. It is produced. So kinases regulate in such a way, they regulate the pathways and production of many things. And they are produced in an optimum condition. When they are produced in optimum condition, they will make only one septa so that the cell gets divided into two. But when this kinase is not there, things get deregulated. So much more FTSZ it produce. So instead of making just one septa, it is going to make multiple septa. And then of course, when there's a multiple septa, that's what I have shown you into the, what happens in the PRKC knockout strains.
Okay. One we have, as we can see, the septums in non-deleted PRKC are in uniform pattern as per your figure, while in deleted PRKC, the septums increased, but the pattern is very random. Could you please comment upon that?
So once you have something more, they will not know where to go. They will not recognize the center. They're not. So a sporulation process, the pole is decided where they have to make the kind of a septa. Similarly, division also, it is between half of the in between the cell. But when you have more, then it will not know where to go. So it can make anywhere.
Okay, thank you, sir. And I'm sure if participants have more questions, they can always, email you and ask you about the work. Thank you so much for such an informative talk. We move ahead. The next speaker for the session is Professor V.C. Kalia. Dr. V.C. Kalia is professor at Konkuk University, Seoul, Korea since 2016. Before that, he was chief scientist at Institute of Genomics and Integrative Biology, Delhi. His major area of research includes microbial biodiversity, bioenergy, biopolymers and genomics, quorum sensing and quorum quenching, microbial evolution. He has published 14 books and authored more than 120 research articles in reputed peer-reviewed national and international journals. He has also received prestigious fellowships like Danish Government Fellowship, French Government Fellowships, American Society for Microbiology, Indo-US Professorship, INSA Bilateral Exchange Program, and many more. He's Fellow of Association of Microbiological Sciences India, National Academy of Sciences, National Academy of Agricultural Sciences. Dr. Kalia has more than 8,000 citations with an H index of 52. He is the chief editor of Indian Journal of Microbiology, and under his leadership, the journal has gained tremendous repute internationally. Today, he will speak on novel drugs to target psychopathogenic bacteria. Over to you, sir.
Okay, thank you. One more step. How to make it full screen?
Yes, sir, it's visible. Please go ahead.
Okay.
You can put it on the screen slideshow.
Okay. May I start?
Please, sir.
Okay. Since 1st of February till 5th of February, you have been presented with enough science that it seems if so much has been done, then shall we either quit or do something else? By now, your mental gates must have been clogged with too much of science. And in most conferences, there is a cultural program for relieving and de-stressing the minds of the participants. So I thought I shall fill that lacuna in the conference. So my presentation will have less data and will give you some time to relax. But I sincerely apologize to those who have very serious scientific bent of mind. Microbes have a highly evolved genetic and metabolic system to encounter environmental stress. The major threat to human beings is from bacterial pathogens. These pathogens behave like sociopaths who mount a violent attack but leave the host very quickly, but crippled. On the other hand, there are psychopaths who are cold-hearted, make a very calculative move, and stay inside the host. So most infectious diseases are caused by psychopaths, and diarrhea and those things are caused by sociopaths. There is an interaction between plants, human beings, and bacteria. They can all live happily if they like. So bacteria, if they are gentle, we can be happy. So these are the bacteria. But a major challenge is protecting plants and human beings from pollutants and pathogens. Although we will not talk of pollutants today, we will talk of pathogens. We think we can protect ourselves by taking medicines. Medicines cure. But I have a very strong adverse view. I think medicines do not cure. Because even today, more than 80% of the world population has nothing to do with so-called modern medicine. In fact, this high-tech medicine has become prohibitively expensive and has failed to guarantee health. So what do we need? I will come to that very soon. You will be surprised that major causes of death are heart diseases, cancer, medical errors, doctors and hospitals, adverse drug reactions, and stroke. In US, a country with 325 million people, 250,000 people die annually because of medical errors. In Canada, 70,000 patients a year experience a death because of human error only. How many died from medical mistakes in hospitals? In 1999, 98,000 people. In 2010, 1,80,000 people died. In 2013, 210,000 people died because of medical mistakes in a US hospital alone. These figures are not synthetic. They are authentic. They are published reports in scientific journals. So I'm citing one of them. Well, India is not far behind in making medical errors. But, well, doctors operate on the wrong leg. 15 blinded after cataract surgeries. And it means the list is pathetically long and unfortunate. There is an interesting story, In Israel, there was a doctor strike for 3 months. And at the end of that, there were less number of deaths than which was happening otherwise. 40 developed industrialized countries, Italy has the greatest number of doctors and the number of people are the least healthy. Japan has least number of doctors and is the most healthy. So there are some more stories of that kind. So if medicines do not cure, then what do we need? What we need is clean water, a clean atmosphere, and adequate food. If we are healthy, we don't need to go to the hospital actually. Okay. So what do we need is a vision actually. In a vision, first I stand for intellectual, the other stand for innovative. Scientists, most of them are intellectual. They think knowledge is power, so they acquire knowledge. But there are people who are innovative, who think applied knowledge is power. So there is a difference between intellectuals and innovators. The way we feel, either we feel happy or we feel sad. You see on the left hand, the left hand side with the smiling face, on the right hand side is a sad face. You will be surprised that it is in our hands to be happy or unhappy. Human body has 1 trillion cells, but we have as many number of bacterial cells in our body, matching 1:1. And they are spread all over the body, including inside. We are happy when bacterial cells are silent, making no disturbance to our system, but we take notice when bacterial cells start talking and make a noise. That is expressed in the form of diseases. So this talking and noise we have to cut down. I will talk of that only today. So microbes on the planet called human beings. Skin microbiota, name the bacteria you will have on your body. Gut microbiota. Normally we have firmicutes and bacteroidetes in the range of 44 to 48% of the total population. But if they exceed and they go to 82 to 86%, then our body shape becomes from a normal human being to this or to this. So there are notorious bacteria causing infectious disease also. Pseudomonas, Vibrio, Staphylococcus, Yersinia, Streptococcus, and Streptococcus pneumoniae. Because the disease, you name it, you have it. Lungs, cholera, skin infections, fever, bloody diarrhea, skin disease, dental caries. So these are the dangerous bacteria which we need to take care. On the other hand, the planet called rhizosphere, the rhizosphere microbiome, there are good bacteria which give you healthy plants. Then there are bad bacteria which give you bad looking plants. And then there are the ugly bacteria which virtually kill you altogether. So growth and development, acquiring nutrients, protecting against pathogens, immune response, tolerance to abiotic stress. So they do everything for the plant. But if they go bad, they cause disease. But if they are ugly, then they cause food contamination. Bacterial pathogens of plants, fundamental and practical importance. Agrobacterium, Ralstonia, Pseudomonas, Xanthomonas, they cause different kinds of plant diseases. Okay. So it is better that if we have a plant, we want to grow it, nourish it beautifully, it will give you a good plant. But if it gets affected, then it will die and it will never give you any yield. So which means prevention is better than cures. So if we can prevent the plant from getting diseased, it will be nice. The battle now is microbes versus vaccines, antibiotics, antipathogens. Which means if we want to protect our body, we need vaccines. The vaccine way. It enables us to acquire immunity against specific diseases. The body is prepared for protecting itself in anticipation, but there are limitations. It's an inadequate immune response or lack of B cells to generate antibodies or may have adverse effects, and it takes long to develop vaccines. The host is left unprotected for a long period of time until resistance is activated. Antibiotic way. It simply says kill the bacteria. So it got the wonder drug, antibiotics. So we thought now the whole world will live happily, but bacteria become resistant to almost any kind of antibiotic which has been developed so far. So there is strong resistance. So what do we need to do now? The best is the antipathogenic way, which says don't kill the bacteria, fool it, let it live, but don't let it become pathogenic. So it will grow in your body, but it will not become virulent and pathogenic. So that is the objective of my study. The bacterial society, bacteria we think are unicellular. That's not true. They have a social interaction, which is called quorum sensing. And within a biofilm, it's a bacterial community. Plants, infectious diseases are caused under these biofilms. Medical concerns, cystic fibrosis, bacteremia, and implants, many respiratory problems happen because of these biofilms. Then toxic and recalcitrant pollutants can be treated with the help of these biofilms. So they can be good and can be bad depending upon how you want to exploit them. So it was in fact initially found out how quorum sensing works because of a squid. It could live with the help of bacteria and avoid fish from eating it. So that was the phenomenon of bioluminescence. So initial clues came from bioluminescence. That is what is quorum sensing actually. So if you have a bulb and no electricity, so it is darkness. But if you give 50 volt, it is slightly lighted, 100 volt, still more, 220, good lighted. Same manner, with low cell density, there is no luminescence, bioluminescence, but at high cell density, there is bioluminescence. So this phenomena is called cell density dependent gene expression. The phenomena is called quorum sensing. So many things can happen over talks. There are actually chemical signals through which they talk with each other. There are many phenomena where symbiotic nitrogen fixation is one of the most important. Then there can be biofilm formation, as I said previously. Antibiotic production happens with quorum sensing. Sporulation, competence uptake of DNA by bacteria, particularly bacillus. Now, bacteria talk to each other in two different manners. One is called intraspecies, personal and family language. As we inside, we go home, we talk of a local language. We don't talk, for example, in India, we don't talk of English most of the times. So that is called single and intraspecies. Then there is a trade and commercial language, which means when we go outside, we deliver lecture or we go to the malls, then we talk in different language. So there is a single system, there is a multiple system. So single photobacterium has a single quorum sensing system, but Pseudomonas and Cynorhizobium bacterium may have multiple quorum sensing systems. So if you want to attack the quorum sensing system, you have to check whether it is a single system or a multiple system. So what are the QS induced, quorum sensing induced worries? Agriculture, vegetable, fruits, as I said, medical, I had listed a few things. Even cleaning water through membrane, it gets clogged with bacteria, which is like a biofouling. And then even monuments, archaeological monuments get in fact affected by biofilms, and there is a deterioration which is much more higher. And the worst part is that bacteria within the biofilm are up to 1,000 times more resistant to antibiotics than they are in the free living form. So if they are outside, you can kill them easily, but if they are within the biofilm, then you have a tough time. So rhizosphere, I said many bacteria happen conjugation of DNA and antifungal proteins are produced, growth promoters are there, nitrogen fixation happens. So it's a very good system, for example, in Rhizobium and Agrobacterium. Now, can we jam this quorum sensing talking between them so that we can protect plants and human beings from pathogenic bacteria? They are the ones which we are worried about. So which means if we target quorum sensing, that can be the new drug targets. So we can, in fact, there are signals which I said are chemical molecules. For example, acyl homoserine lactones, then there are peptides. There are quite a few of them. We can, in fact, stop them from getting synthesized. So we can block their signal synthesis and accumulation. Or we can degrade them through chemical routes, metabolic routes, or enzymatic routes. There are many bacteria and plants which produce enzymes. For example, bacteria produce acyl homoserine lactonases and acyl homoserine acylases, which actually either open up the ring of the lactone of the AHL signal molecule or cleave the side chain. So in either situation, the signal becomes inactive and quorum sensing does not happen. And there is no virulence or pathogenicity happening. So this is either this is like a ring and a side chain. So if you cleave it, this is a acylase. If you open it, that is the lactonase. So Serratia, Vibrio, Ralstonia, Agrobacterium, they have different kinds of signals depending upon, and as I said, the systems can be single and multiple. Many bacteria have been known to produce lactonases, and Bacillus are among the top. Naturally, they don't, in fact, cleave every kind of acyl homoserine lactone, but depending upon the signal length, side chain length, it varies from lactonase to lactonase. Then there are acylases also produced by Streptomyces, Ralstonia, Pseudomonas, Shevonella. They also depend upon the side chain length. So which means opening up the ring through lactonase is much cheaper and easier than cleaving the side chain, which is highly specific. So then there are oxidoreductases also. Normally there are only lactonases or acylases which are found in most bacteria. So we thought if we can find a bacteria which has both the enzymes, that will be wonderful. So searching each one by one is a tough job. So we did a comparative genomic study. We took almost 300 to 600 sequence genomes and found out which of them has lactonase and acylase. And at the end of study, we could find out that there are 4 bacteria which have the potential to produce both enzymes: Deinococcus radiodurans, Hyphomonas, and Photorhabdus. And you will be surprised to learn that this was an in silico study which we had published in 2011. And in 2014, a group from Germany actually demonstrated that Deinococcus radiodurans has acylases and lactonases. So plants also can produce these enzymes which can inhibit the transcription process or can cleave the signal molecules, and these are produced by plants. Curcumin is one, cinnamaldehyde is another, Medicago sativa is another, which produce cannabinine, curcumin, and then there are limonoids, flavonoids, which are produced. These are all beautiful plant-based quorum sensing inhibitors. Even grapefruit juices, grapefruit have beautiful quorum sensing inhibitor present in them, so they are healthy fruits. Then there are many more bacteria which can do this job. Sweet basil is another. Herbal plants are there. Legumes are there. Edible plants and fruits like ananas, musa, ocimum, they are also very healthy fruits. So essential oils and hydrosols can do also the same job. We can also introduce the gene of AHL lactonase from bacteria to another beneficial bacteria which can express the gene and protect the plant from pathogenic quorum sensing inhibition, quorum sensing expression. So quorum quenching substances in torrential plants have also been found out. There is although a worry that, like many other antibiotics, bacteria may become resistant also to quorum sensing inhibition. So we have to be very careful when we make a statement that we can use these quorum sensing inhibitors and fool the bacteria. So the solution to outdo the bacteria is Bacillus is one of the bacterium which we have found out of more than 1,000 bacteria which we have tested one by one. We found out it has lactonase which can in fact take care of most quorum sensing inhibitions. And these works we have published in quite a few journals, and they have been very well appreciated and highly cited. And this Bacillus has other properties also, which we have worked for almost 20 years, that they can produce bioplastic, they can produce hydrogen also from bio-wastes. So the mission is to boost the morale of the scientific community who are eager to look for more efficient treatment with much lower doses of drugs. So when you use a quorum sensing inhibitor, the antibiotic usage can be reduced down substantially, as I said in the beginning. From 1,000 times, you can reduce it to 1. So this is the beginning of a long journey, but I believe together we can achieve everything, as this symposium itself was not done by one person, but it was a team effort. So we, although congratulate the team leader, and I am thankful to the team leader and the organizers of this conference for inviting me and giving me this chance to share my thoughts. So I am thankful to them, although they can invite actually a number of people in the country and abroad, but I am thankful to them that they invited me also. So I would like to thank obviously a very special person, Professor Ruplal, and his team members. The list is very long, so I will not take much time. So that's all. Thank you very much.
Thank you very much, sir, for such an interesting talk, presenting science sprinkled with humor and wisdom. We have one question for you. Can we use quorum sensing to characterize the microbial interactions in the community?
Yeah, sure. There are many kinds of quorum sensing. So systems, signals, and transcriptions. So we can, in fact, use them.
Can you give an example?
For example, Cynorhizobium, there are 3 kinds of quorum sensing systems. Pseudomonas have 2 kinds of systems, but Bacillus has 1 kind of system, for example. So these spore formation, whether it will happen or not, uptake of DNA, agrobacterium has another system. So which means based upon these also, we can quantify that whether it is a quorum sensing producing bacteria or it does not have.
Okay, so we take the last question.
I could answer, although very difficult questions to answer, but I will try.
Okay, sir. How quorum sensing interfering strategies can inhibit a pathogenesis of biowarfare agents?
If they are operating through these quorum sensing signals, the best is that quorum sensing signals are actually exuded into the climate and outside the bacteria, which means you can use the inhibitor, cinnamon aldehyde, for example. It's a very simple thing. You can give it outside the body and still kill the bacteria.
Thank you very much, sir, for patiently answering all the questions. And I'm sure that if participants have more questions, they can email you directly. Thank you very much for yet another interesting talk, sir. It was our pleasure. We move further. Our next speaker for the session is Dr. Parveen Rishi from Punjab University, Chandigarh, India. Dr. Parveen Rishi is professor and ex-chairperson, Department of Microbiology, Punjab University, Chandigarh, and also served as ex-dean, Faculty of Sciences, Punjab University, Chandigarh, India. Her area of specialization include medical microbiology, immunology, with special reference to the development of biotherapeutics, prophylactics, and diagnostics, particularly against Salmonella enteric sorobar typhi infection. She has been honored with several accolades, such as the Best Publication Award, Srimati Premlata and Professor DVS Jan Research Foundation 2020, Professor S.R. Vyas Memorial Award conferred by Association of Microbiologists of India at University of Hyderabad in December 2018, and ICMR Dr. Y.S. Narayan Oration Award conferred in 2013. She has been recently elected as a member of Central Council of Association of MI. In fact, she is the elected president this year for a term of 3 years, and is also the academic editor for PLOS ONE and coordinator of e-Pathshala program for MSc courses in microbial pathogenesis. The title of her talk today is Betals as Drivers for Emerging Antibiotic Resistance and its Clinical Validation. Dr. Rishi, welcome, and over to you. Dr. Rishi, we can't hear you. Dr. Rishi, please unmute yourself. We cannot hear you. Huh. I'm sorry to interrupt, Dr. Rishi. We cannot hear you.
Hello. Hello.
Yes, ma'am.
I'm audible now, yes?
Yes, ma'am.
All right.
You need to share your screen, ma'am.
Yes. All right. Thank you, Dr. Charu, for the kind words, the kind introduction you have given. And at the outset, I would like to thank the organizers for giving me this opportunity to participate, you see, in this prestigious meet, and to share my views on the topic that is metals as additional drivers for emerging antibiotic resistance and this clinical validation. You see, 2 days back, we had a wonderful talk by Professor Guleria on emerging antibiotic resistance. And you see, ever since the discovery of antibiotics, these drugs have actually proved to be miraculous drugs for treating the infection or infectious diseases. But due to the emergence of antibiotic resistance in the pathogens, the clinical medicine is now facing the challenge. And WHO has also reported that growing antimicrobial resistance is a life-threatening crisis, as well as it's a costly costly concern also. And it is also being speculated that by 2050, more deaths may occur due to AMR rather than with cancer. So in addition to, you see, the intrinsic mechanisms which are there within the pathogen to have to show the antimicrobial resistance, there are several factors which we know can contribute to the emerging resistance. That is the inappropriate use of antibiotics, overuse of antibiotics, for example, in agriculture, livestock, and fish farming. And there is inadequate public health measures or poor sanitation along with hygiene. And of course, you see, we are not able to identify or discover the newer antibiotics at the same rate at which the pathogens are mutating themselves or they are modifying themselves. And in addition to that, there is a role of multitude of substances such as heavy metals or biocides, which are occurring in the environment or sewage. They are also being put forward now. And in this context, I just would like to share my views on the heavy metals and the antimicrobial resistance. The heavy metals, you see, they enter the ecosystem through geogenic sources and anthropogenic activity. Geogenic, as we all know, that is a natural phenomenon. For example, volcanic eruptions or abrasions in the rocks, and anthropogenic activities, including the industrial ones or the agriculture. So as a result of this, the heavy metals, they enter the ecosystem. And, because the metals, the metals can maintain the selective pressure for a longer period. So metal contaminants function as the selective agent, leading to the survival of the metal tolerant cells due to mutations. So in the literature, as far as the metals and antibiotic resistant determinants are concerned, this association has been reported for several pathogens. For example, the genes tolerant responsible for tolerance to arsenic, copper, and zinc have been observed in methicillin-resistant Staphylococcus aureus, and the same has also been reported for Salmonella enterica typhimurium, which was isolated from retail foods. So, as far as this association is concerned, we can call it co-selection mechanism, which may be, co-resistance. Co-resistant means exposure to one toxicant can lead to the resistance to another insult, maybe plasmid mediated. Then it can be cross-resistant, that means physiological coupling wherein similar resistant determinants confer resistant to multiple agents wherein efflux pumps may be involved, and then the co-regulatory pathways wherein expression of resistant determinants are governed by same regulatory pathways. So thus there are concern about the the metals the metal induced antibiotic resistant genes in the environmental as well as the clinical settings. So in this context, we have studied this association in case of Salmonella for two reasons, because Salmonella is the organism which spreads through the contaminated food and water, and secondly, by WHO, it has been put on the list, which is priority 2, as far as the antibiotic resistance is concerned. So Salmonella enterica typhi, we all know that it causes typhoid fever, and it is transmitted through fecal oral route. And then this is actually the major concern in the developing countries, where we have inadequate sewage treatment facilities. And then as I told you, that the metals, they enter the ecosystem, and then they enter the food chain and the water, because Salmonella can persist in the sludge of sewage treatment plants for a longer period. And when minimally treated sewage or the effluents, that is used for the agriculture practices, then these organisms, they enter this along with the metals one, they enter the contaminated food and water from where it is transmitted to the host. And there are several antibiotics which are, of course, advocated for its therapy. But then the hypothesis of our study was that if the organism, they are exposed to metals in the environment, pathogens, they are rendered metal tolerant, and metals being non-degradable exert long-lasting selective pressure, and then that can make the pathogen resistant to various advocated antibiotics inside the host later on. So then there are concerns, of course, which are both of an environmental and clinical concerns. So for this, first of all, we procured the clinical isolates from various hospitals, for example, from Chandigarh Medical College, Sector 32. We also procured the isolates from AIMS as well as from Maulana Medical College. So we had gone for the antibiogram of these clinical isolates along with the standard one by disk diffusion, that is a conventional method, as well as we determined the minimum inhibitory concentrations. And by both these methods, the isolates which have been shown in blue, they are the sensitive ones, or the pink one, they're intermediately sensitive, and the rust or the brownish one, they are more resistant. That means they may be resistant to 3 antibiotics or more than 3 antibiotics. So they were categorized like that. After evaluating the antibiotic sensitivity pattern of all the clinical isolates, then we estimated the presence of various metals in these isolates. And out of all these metals which you are seeing, the cadmium was predominantly seen, high levels of cadmium were predominantly seen in case of the clinical isolates. We can see the first one, TY2, the level was much lower as compared to the other isolates. So correlation of antibiogram and presence of metal, it was seen that the level of cadmium was almost double in case of the clinical isolates, and the concentration was, the level was more in the resistant strains, and it was much lesser in the standard strain. So cadmium, if acquired by the clinical isolate, it may be because of the longer exposure, of the organism in the environment where metal contamination is there, and from there it can enter the host through the food chain or water. So that indicated that cadmium is one of the factors for inducing the antibiotic resistance. So to further ascertain this association with cadmium, we adapted the isolates to higher concentration of cadmium for 10 successive cycles, so that the maximum permissible cadmium accumulates within the cells. So when we characterize these cadmium adapted cells, the lag and the log phase, the lag phase was delayed, and there was a prolonged log phase, and that may be because of the metabolism dependent uptake metals, which is a slow process. And then when we characterize it by transmission electron microscopy, wavy appearance in the membrane was seen, along with the narrowing of periplasmic space, and some electron dense areas were also seen, indicating the accumulation of cadmium inside the cells. Then you see, when cadmium was accumulated in the all the clinical isolates, and we can see that the range was from 3.102 to 28, and accordingly, when we re-evaluated the MIC of clinical isolates, there was a many-fold higher fold of MIC. MIC, that means increased by several folds in the clinical isolates, where the cadmium concentration was much higher. So this variation in the MIC to these antibiotics were seen, and you see in this case, if we look at ciprofloxacin, 76% of clinical isolates show the variation in the MIC of CIP, and so on. So going by this correlation, we can say that there was an alteration in the MIC of the conventional antibiotics, and this is very important. This was an important observation, that sensitive strains became resistant, and the resistant strains became more resistant as per their MIC, as per the CLSI guidelines, in the standard as well as in the clinical strains. So further moving on, in order to see the phenotypic changes which may be induced by the cadmium accumulate in the cadmium accumulated cells, we carried out native gel zymography of SOD, because this is a metal associated enzyme, and otherwise in the unadapted cells 3 forms of SOD was seen, whereas in case of the adapted ones, only one isoform could be seen, and that may be because that on adaptation the cadmium may induce certain it can help in initiation of the cascade of reaction, whereby there is release of iron and resulting in the reactive oxygen species, and that may be through the Fenton reaction, and this was also seen in case of clinical isolates shown below. And then you see, if we talk about the reactive oxygen species, or maybe the organism tries to counteract that oxidative stress by enhancing the antioxidants, for example, catalase, glutathione reductase. But then there was a reduction in the SOD in the cadmium adapted cells, and that that is very well correlated with the zymographic study. Similarly, the biofilm forming potential was also seen to be more in case of cadmium adapted cell as compared to the cadmium unadapted cells. Then intracellular killing assay. Intracellular killing, we know that Salmonella is an intracellular pathogen, and it can survive and multiply within the macrophage milieu. And here we see that by 90 minutes, the intracellular killing was reduced in case of cadmium adapted cells as compared to the unadapted cell. So indicating that there was more survivability of cadmium adapted cell in the inside the macrophages, and that may be correlated due to the higher levels of antioxidant, because by producing the higher levels of antioxidant, the organism may try to combat the oxidative stress which it encounters within the macrophages. This was an ex vivo test. And then looking at the protein profile, protein profiling of cytosolic membrane, inner membrane, and outer membrane, several changes were seen as far as the expression of these proteins were there. But the noteworthy change was seen in the expression of porins, and maybe the organism tries to downregulate the expression of porins in order to inhibit the influx of the molecules like antibiotics. So then we studied the quantitative by quantitative PCR techniques, the expression of various genes. For example, SMTA gene, which encodes for metallothionein, is the metal binding protein, and that confirms the binding of cadmium to the cells. And then YBL gene, which is cation proton antiprotein, and it encodes for potassium antiporter in Salmonella, and its role has been defined in maintenance of cellular homeostasis, and indirectly its role has been associated with the type 3 secretion system of Salmonella pathogenicity islands. So we can see that there was a change in the expression of these genes also. Further going, because we wanted to see that is there any role of efflux pumps by which the organism can throw the antibiotics like molecules outside. So we had gone to we checked the gene expression of SOX, these genes, and ACRB and TOL-C, these are the efflux pumps, and the other two are the genes which encode for the transcription regulators. And here also we can see the enhanced expression in case of cadmium adapted cells. So since the efflux pumps, the expression of efflux pumps was inhibitor, we wanted to identify, we wanted to check the efficacy of certain efflux pump inhibitors, 2 chemical agents and 2 biological, 1 biological agent was selected, naphthylamide, PA beta, and you can say, and naphthyl methyl peprazine and NPM, sorry, NMP and plumbagen. So these inhibitors we evaluated, and after standardizing all the concentration, we can see that when we use these inhibitors at 1/4 of their MIC, again the expression of these genes or which are involved in the efflux pumps, that was increased even in the presence of inhibitors, because the organism may sense that insult inside and still try to enhance the expression. But you see, that means one indication was definitely there. That means these inhibitors, they are interacting with the efflux pumps. So we checked the toxicity of these inhibitors on the basis of hemolysis as well as the cytotoxicity assay using the cell lines. So it was found to be perfectly fine. And then, if this is the role of again efflux pumps, NPN is phenylnaphthylamine, which otherwise cannot get permeabilized within the cells. So we wanted to see the efficacy of these efflux pumps inhibitor, whether they allow NPN to go inside the cells or not. So what we did, because NPN otherwise it doesn't give you much fluorescence in the environment which is outside, but under the once it is in there, inside the cell in the hydrophobic environment, it gives the strong fluorescence. So in the presence of NMP, for example, hardly any fluorescence was seen. That means it did not allow NPN to go inside the cells. That is how we selected this. And then again, ethidium bromide. Ethidium bromide, you see, is a substrate for several efflux pumps. And again, we wanted to see whether in the presence of inhibitor, ethidium bromide is able to come out or not. So again, if you look at the upper one, the NMP, you see the fluorescence units were much more as compared to the other. That means this is because of the fluorescence which was given by the ethidium bromide. That means ethidium bromide efflux was inhibited in the presence of the inhibitors. So after, when we see that it does not allow the ethidium bromide to go out, that means there should be a very interesting phenomena. That means if we use these inhibitors in combination with the antibiotics should not be allowed to come out. So otherwise, you know, efflux pumps, they try to throw the antibiotics outside, thereby increasing the concentration of the effective concentration of the antibiotic which is required. But in this case, when we use the combination of inhibitors with the conventional antibiotics, the effective concentration of the antibiotics were reduced, thereby giving the synergistic relationship. So if we look at the FIC index, though it was found to be synergistic. So that means inhibitors and the conventional antibiotics, they make a very good combination, which can be, you know, for which the strategies can be made to add it to the existing antibiotic, or you can say existing battery of antibiotics, rather we can say that. So ultimately, finally, once that relation, that synergistic relation was seen using the standard strains, we validated this combination, NMP or antibiotic combination, with the freshly procured clinical isolates, and again they were found to be synergistic, except 2 strains where the association was found to be additive. So what we can say from this study, that in the presence of cadmium, when the cadmium is present at higher level, and we make the cells cadmium adapted, so the sensitive strains, they became resistant, and the resistant strains, they became more resistant. However, when EPIs or antibiotics are there, so these EPIs along with antibiotics, then this relationship could be reversed. The resistant strains became sensitive, and the more resistant strains, they became less resistant as per their MIC. That means this could be reversed. So the key points of the outcome of the study, that metals, they are another driving factor for inducing antibiotic resistance, and efflux pumps are maybe involved in the co-selection of metal and antibiotic resistance, and when used in combination, they may act as yet another effective combination which might strengthen the reservoir of existing antimicrobial compounds. Because you see, when we use the combinations, these combinations, they act by multi-pronged approach. So the organisms, lesser chances are there that the microorganism will try to become resistant to it, because microorganism, we all know that they are smarter, but then we have to be smarter, because we have to attack the microorganism from different angles, from cell wall, from DNA, protein synthesis, and all those things. So we see here that the microorganism is having the efflux pumps. When the antibiotics are given, the efflux pumps, they are activated. They do not allow the effective concentration of the antibiotic to come in, or maybe the antibiotic, if that is in, that is thrown out. And if we are using the EPIs in combination with the antibiotics, so these efflux pumps, these EPIs, they interact with the efflux pumps, and they do not, even if the antibiotics, they try to throw the antibiotics out, it doesn't become possible. So on the left side, you see that the organism remains happy by, throwing the antibiotic out, but in the right hand side, the pathogen really becomes gloomy because uski ab chalti nahi hai. So these are some of the best practices which are required to be implemented. As Professor Guleria also mentioned, that any perturbation in one of the habitats which are meant for humans, in plants and animals, because that can result in the well-being of the other habitat because of the interconnectivity or interdependence. And there is need to have the we need to reframe the stringent guidelines as far as the or fixed policies as far as the management and the disposal of effluents are concerned. Then there has to be a dialogue between the experts of public, animal, and plant health. They have to be a crosstalk between them. Then we not only, you see, the government or the policy makers, the general public has to be made aware, and we are equally responsible, because we are the ones who are also responsible for contributing in the emergence of antibiotic resistance by not following the ways. As Professor Virdi, I remember he has posed the question that, like in the Western countries, over the counters you cannot get the antibiotics, but here there is no problem at all. So in addition to that, that is why there is a need to spread the awareness, and we have learned several lessons from this pandemic, and there is a need to maintain the hygiene to avoid the incidence of such infections. So this study, was published in scientific reports, and that news was also flashed by India Science Wire. This is a new syndicate venture of Vigyan Prasar. This is by syndicate, and this is how the news came, that how heavy metals are helping spread drug resistance. And here you can see she is Dr. Ujjwal, who has carried out the work, and she is the another co-investigator. She also used to happen my student who worked on antimicrobial peptide, and now she is an assistant professor. And Dr. Virdi came for her viva, and this is my team. I'm working the AMR group. I'm working on the antimicrobial peptides. I'm also working on the probiotics. Amrita has presented her work with the probiotics, because when we are, looking at the association of salmonella probiotic at the gut-brain axis, and these two girls, Reena and Shania, they are working on the vaccine candidates. This is the biosensor group, and Steesh and Preeti, in collaboration with IMTech, we have carried out this work and filed the patents, and we have also transferred the technology to the industry. And she is the girl who has just finished her PhD with the cocktail of microbial enzymes, which we used as the anti-biofouling agent. Anti-biofouling agent, that means to remove the black chunk from the kitchen pipe, as well as for the biofilms, for removing the biofilms from the dentin discs or catheters and contact lenses. So this is what I am doing here. Thank you so much. Thanks for your patience.
Thank you very much, ma'am. That was really an interesting talk, but just due to paucity of time, we will not be able to take up questions now, but I'm sure that they can always post you and write to you directly if they have any queries related to the work you have shared or any other thing. Thank you very much for this informative talk. We move on, and next in the series, we have Professor Vibha Tandon with us. Dr. Vibha Tandon is Professor in Special Center for Molecular Medicine, Jawaharlal Nehru University, New Delhi, India. She has long-lasting experience in the field of chemical biology, radiation biology, and cell signaling drug development, and study of their mechanism of action based on genomics and proteomics. development of antibiotics for gyrase-resistant strains targeting topoisomerase 1A gene in bacteria. She has many prestigious publications and 4 patents to her credit. She has held many academic and administrative positions. Professor Tandon has been awarded with Fulbright Senior Research Fellowship to visit Georgia State University, Atlanta, USA, Royal Society Fellowship for a visit to Cambridge University, UK, under the Bilateral Exchange Program of INSA, Professor D. Nasipuri Memorial Award by the Indian Chemical Society, Mid-Career Award of UGC for excellent research work from 2018 to 21, and many, many more. She's also a member of National Academy of Sciences, India. I now request Dr. Vibha to kindly deliver her talk on a multi-scale approach to combat the mechanisms of antimicrobial resistance through ABT119B, a known bacterial topoisomerase 1A inhibitor. Over to you, ma'am.
Am I visible to everybody?
Yes, ma'am, you're visible and audible. Please go ahead.
Okay, so thank you very much. I would like to, on the onset of my talk, I would like to thank organizers, especially Professor Ruplal, who has given me opportunity to present my last few years work here. And I would also like to say congratulate the whole team of AMI and NSCR that you have gone very well, and you have the kind of speakers you have collected. They are like every lecture is a wonderful lecture, and I have learned a lot during this say conference. So with all these things, I would like to move ahead and like to present my work here in next 20 minutes and say, as I don't have to, the stage has been already set for me. Dr. Joshi, and before that Dr. Kalia and Dr. Weissing, all speakers have already talked about the antibiotics, and I would say that the just preceding speaker has talked a lot about the say antibiotic development. My talk is also about the antibiotic development, and I will be talking here about a small molecule which we have developed in our laboratory, and that particular molecule was synthesized in our laboratory, and then we have taken it to the preclinical level. And we have you will also find that the most of the novelty in this work will be for the benefit of the students. I want to tell you that we have identified some new say target. So we are not targeting gyrase, or we are not targeting the topo 2, we are targeting topo 1A, which is a new target and has not been clinically well validated. So with all these things, I won't explain about it, but I would, as already all of you know very well, that that there is a lot of bacterial resistance is there. And looking at the bacteria, I'm only showing here you the map of say E. coli resistance to aminoglycosides, because aminoglycosides are supposed to be still working against most of the microbes, but you can see that still India also have become a gray color. You can see the almost 20% of the resistant in Indian strains are also fine. So there are a number of pathogens which have been identified by different agency, and specifically WHO. And WHO has identified those pathogens into 3 different categories: critical, high, and medium. So we have been working on the antibiotic development from almost last 10 years. But when this report came in 2018-19, we what we did that we have screened our molecule against some of the critical and some of the high and some of the medium, strains of the WHO list. And when we screen our molecule against these pathogens, we have found that our molecule was showing certain activity, good activity. So this will be the story of the activity of these molecules against these WHO priority pathogens. So now, first of all, going into the story, I would like to tell you that the target of our molecule is topoisomerase 1A. And I'm trying to show you here the structure of topoisomerase, bacterial topoisomerase 1A. And it is it has 4 domains. I'm trying to show you E. coli crystal structure here. It has 4 domains, and it has a DNA binding domain, and that is called as the active site of the molecule. And it causes the DNA relaxation, and after DNA relaxation, the replication procedure starts, and then the religation occurs, and that is how the daughter cells are formed, or the different bacteria divide. And then there is a human domain. Human topoisomerase is also found, and they have they also have almost 3 major domains in that, and they also have an active site over there. So, but there is, these are structurally different, but there is some kind of a homology between them. So the idea of this, that we wanted to use a new target here, because most of the topoisomerase inhibitors which have been developed, they have been developed against the gyrase, and because of the overuse of such antibiotics, the gyrase has got mutated, and there is multiple mutations are found in the gyrases. So that is why we started looking into the new target, and that was a topoisomerase 1A. Now and the another point which I would like to raise here, that when we develop this molecule, we try to see that this particular molecule should be selective to the E. coli topo 1, and it should not act on the human topo, or it should not act on the gyrase. We wanted a molecule which should be selective to the topo 1 only. The reason for that if a molecule will act simultaneously on the topo 1, human topo 1, then it will not be a safer molecule, because it will kill simultaneously the human topo also. And the gyrase, as all of us know, that if it acts on the gyrase, then it will not be a selective molecule. So with all these things, we started synthesizing a number of molecules. And while I was synthesizing a number of molecules, so out of a library of some 150 molecules, we could come out with a molecule which is known as PPF, and we have given it a name PPF, and it is chemically it is a bis-benzimidazole molecule. So while we were doing this work, we have evaluated this molecule almost till now against the 600 bacterial strains, and out of the 600 strains, almost 500 strains are the members of the WHO priority pathogen list. And while we were doing this work, we have observed that this molecule did not show very good activity against a certain A. baumannii strain. In some of them, they have shown very good strain. I have also removed from here the Pseudomonas aeruginosa, but we didn't get good result, at least from this pie chart, against the baumannii and the K. pneumoniae, Klebsiella pneumoniae. But rest of the cases, whether it is gram positive or gram negative, we have got wonderful results, and the results were very, very good against the S. aureus, and that was like MIC and MBC was very good against the multi-drug resistant VRSA or the MRSA bacterial strains. So these are the few results which are shown here, and you can see here that this is our molecule of interest, and these molecules are showing the MIC and MBC of 2 mg per liter. But if you look at the ciprofloxacin, imipenem, and vancomycin, either say intermediate or they are resistant to these particular strains. So we have, you can see here, Acinetobacter, E. coli, Klebsiella, methicillin-resistant Staphylococcus aureus, vancomycin resistant Staphylococcus aureus, Salmonella typhi, Shigella, Enterococcus, Streptococcus. So you can see that we could develop, we could find out, or we observed during experimentation that this molecule has shown wonderful activity against a couple of say WHO priority pathogens. So then this is just for this the interest of the students, that whenever you do MIC or MBC with the clinical strains, you have to find out the first, you have to say decide their resistance according to the CLSI guideline, and you have to follow the CLSI guideline and CLSI diameter there. So if you do it on the say on a particular disc, then you have by the size of the zone, you decide that it is sensitive or it is intermediate or it is resistant. So one has to do these experiments before taking the clinical strains or any kind of a bacterial strains for the different work. So we have done it with that, and then we have done the with the PPF also. These are the methicillin resistant Staphylococcus aureus, and you can see that these methicillin sensitive Staphylococcus aureus, which are susceptible to this, and some of them are which are resistant to these antibiotic, but they are showing such a good MIC and MBC. It's a PPF. They are showing such PPF is showing such a good MIC and MBC against these Staphylococcus aureus. Similarly, then we move to the VRSA, the vancomycin resistant one. You can see here that again this particular molecule was showing good minimum inhibitory concentration or minimum bactericidal concentration. This is again the results against the Enterococcus species. So this things just goes on, and then what we did that we collected the some 600 or 700 strains from the different places. The major source is Safdarjung Hospital. Professor Rajni has provided us those say strains, and otherwise we have got it from the say this European say health department. We have got some CDs from CDC. Then we collected it from different say academic institutions also. So we just kept on doing this work. And last year, under the AMR mission, this was funded by the DBT and BioRec. And then an industry partner has also entered into that, and that industry partner is Anthem Biosciences. So they also had a bank of say a lot of strains. I'm not including their strains, but I'm showing my results over there. So like this, we just kept on working on the different kind of strains, different pathogens, and then we showed our result on Shigella flexneri, Salmonella typhi. You can see that they are resistant to these many antibiotics, which are the which are given as a panel by the WHO, and they are showing the resistance against those panel, but they are sensitive to the PPF. So like this, we just kept on going, and we just kept on working on the different strains of the different pathogens, and and we could see that this molecule is acting against the number of pathogens, and out of those, say, 12 pathogens, it has shown activity against the 7 pathogens, 7 very good, 7 activity against those pathogens. So now what happened, when we do such kind of a work, now in the case of antibiotic, there is another parameter which is called as the post-antibiotic effect. post-antibiotic effect is this, that if you give a molecule and how longer it is effective against a particular strain. So we did this experiment also, because this experiment clinically decides that what should be the domain, what should be the dosing regimen of a particular antibiotic, and how sooner you have to give the second dose of a particular antibiotic. So any antibiotic which has a post antibiotic effect for several hours, that can be a good molecule, because it is still active and it is still working against the bacteria inside the human bodies. So this is the result of certain standard antibiotic, say trimethoprim, gentamicin, ciprofloxacin, ampicillin, kanamycin, tetracycline. And this is the average rates of the ppf, that how many hours it is found inside the say against a different standard strains, mtcc, so Klebsiella, Staphylococcus aureus, or E. coli. So we have taken some standard strains and just gave you a glimpse of it. But if you go into the, say, my further slides, you would find that still this particular molecule is giving, say, very good, say it is still found in some cases even for the 18 hours. So you can see that its post-antibiotic effect is very good in against these pathogens also. So it is showing very good, it is staying somewhere, most of the places it is staying for the more than 18 hours in certain clinical strains. So with all these things, we have a good results, but we had to, I just want to show you that how it really acts on the target protein. And here my target protein is topoisomerase 1A. And when we did this so here the topoisomerase 1A is responsible for the DNA relaxation. And you can see here that when there was no PPF was added, then there was the whole, say this is a supercoil band, and when you added the topoisomerase, this is a relaxed band. But as soon as we added the PPF, the supercoil band has come back, which is showing that it is inhibiting the topoisomerase 1A. So then whenever you do the, so we have done it with the SRase topoisomerase, we have cloned E. coli, Acinetobacter baumannii. So we are still working with the topoisomerases, because these topoisomerase 1 are not available in the market and you cannot buy it. So we have one has to clone it and express them. So then say, in order to see that where this particular molecule binds in the topoisomerase, we did the docking and MD simulations with these molecules, and we could find that it binds at the with the complex of the benzene, with the complex of the say protein and DNA, whereas it binds in the human topoisomerase at a completely different place, and it binds only with DNA, and it doesn't bind with the complex of the topoisomerase and the DNA. So it is binding differentially. That is why its effect is also different in the between the bacterial topo and the human topo. So then we started doing certain experiments, and because of the paucity of the time, I cannot show you. But we did a number of experiments, and when we did number of experiments, we created the, say, mutant of the active site of the protein, and we have created double mutant and single mutant, and then we did the we tried to see that the cleavage of the DNA is occurring in the mutants or not. So when we did it, we found that the cleavage was less in the case of the mutant protein, but it was higher in with the case of the wild type protein. So then which suggests that this molecule goes and binds at the active site of the protein only, which was a good news for us. And we are still working on this. We are trying to do the X-ray crystallography, and with all thermodynamic parameters, we are trying to establish what is the binding site of this molecule. Then we did the, how much time I have, Dr. Charu?
Ma'am, I'm extremely very sorry because we are actually running short of time. We have a valedictory function scheduled. So please, if you can just wrap up in another minute or so. I'm very sorry for that.
So give me 2 minutes, fine. Because if I'll wrap up like this, it will look very.
No, go ahead, ma'am.
So I'll give 2-3 slides more.
Thank
So
you.
then when we did the number of experiments, we could find that the PPF is subjected to the efflux pump also. And you just listen my previous speaker, and she said that all antibiotics are subjected to the efflux pump. So we also found that it was subjected to the efflux pump. And then when we have taken a drug, which is when we have taken the such strains which are having the deleted efflux pumps there, then it will look like that we would find that our molecule, the uptake of this molecule was quite increased. If you look at the deleted Tol-C, you can see that the uptake has increased. And if you take the E. coli K12, then the uptake was less. So it was very clear that it was being effluxed out. And when we used it along with the efflux pump inhibitor, its uptake has gone very high. So that was very clear that this molecule is also suffering with the efflux pump. So with all these things, I would like to give some important point over there that this molecule has a better, say, kinetics than the ciprofloxacin, and it is going for it's efflux kinetics, and its killing kinetics is having a different kind of a curvature as if you compare it with the ciprofloxacin. Ciprofloxacin is is killing the bacteria in a longer time, whereas the PPF is killing it in a very straight kinetics and a very fast kinetics of killing kinetics of PPF is quite faster. So then I won't go into the all detail. What majorly I want to say discuss with the people who are like all the audience who are listening it, and specifically with the students, that what we did, that whenever you try to develop an antibiotic, and this was an idea which was given to me by Professor Yogen Singh, who is the one of the mentor and who is the president of AMI, and he told me that when you have got this molecule, why don't you give a long-term treatment and with this molecule to the bacteria and try to see that what kind of a mutation has been generated by the long-term treatment. So we did long-term treatment, and we went up to the 900 generation of this bacteria by every time by treating with the PPF, and whatever colonies were remaining, we were just trying to grow them. And that is how we did it, that on the we use half of the MIC, 0.5 of the MIC for generating these kind of 900 generation of the antibiotic. And another thing we did simultaneously, that we took the 1x MIC of PPF, 2x MIC, 5x MIC, and 10x MIC, and then also we tried to see that whether any bacteria is remaining over there. So none of the bacteria remained over there, but by the de novo generation, which we did by long-term treatment, the good news for us was there that we did not find any mutation, neither in the topoisomerase nor in the bacteria. And that has given us a strength that this molecule is not generating mutation in the bacteria very fast. So that was a good news for us. But we have found that when we have treated the bacteria, and the efflux pump got upregulated as soon as we treat the bacteria. So you can see this is E. coli K12, and this is E. coli K12R, that is a resistant bacteria. With all these things, now I won't present the NGS data. I will directly go to the animal data. And what we did, that we have generated the thigh infection model and the septicemia model in the animal. And when we did that and we treated those animals with the different doses of the PPF, you can see here that more than 80% of the animal have survived at the 20 mg per kg body weight, and say at the 15 mg, almost more than say 60%, 50% animal have survived. So this worked in the thigh infection model, and this is the murine sepsis model. And this is only the survival, this is the murine sepsis model, and this is the thigh infection model. In thigh infection model, you do the colony counting to see that how much of the infection is still there after the treatment of the antibiotic. So you can see here that in the treated one, you can see that there is the several log reduction of the CFU was observed in the treated one. So similar thing, it is being shown to you in the SRAS infection. We infected the mice with the SRAS, and there was one severe septicemia model, and another was the thigh infection model. And you can see that we have got the very good activity, and you can see almost 100% of the animal have survived at the 20 mg per kg body weight. So that's all we could say, that this that this molecule is able to work against the bacterial infection even in the mice also. So with all these things, I won't go into the LD50, ED50 data. The summary of this work is that this molecule targets the the bacterial topoisomerase. And other than targeting the bacterial topoisomerase, it is also inhibiting the glucose uptake pathway. I did not present the NGS data of the treated organism, treated E. coli and treated S. aureus. So that suggests that it does not allow the glucose uptake. It also causes the inhibition of the replication, transcription, and recombination and decatenation of the DNA. This data we have got from the NGS a challenged organism. And we can also say that PPF stabilizes the topo-1 DNA cleavage complex, and it doesn't allow the religation of the DNA. And that is how a lot of bacterial DNA gets damaged and bacteria die. So with all these things, now I would like to, I think while I was presenting my data, I have already told you what were the salient feature. One more last example, last thing I would like to mention here, that we have done the pharmacokinetics and the toxicity with this molecule, and it is working. It has shown that it has a half-life, it has a Cmax at 1.5 hour, and it reappears after 4 hours. And it is safe up to, in the oral route, up to 2000 mg per kg body weight. But at the IV, the LD50 was observed at the 125 mg per kg body weight, which is quite a good range if we want to, because it is showing the ED50 at the 10 mg per kg body weight. So with all these things, I'm open to this question. I work here, this is a place, and these are my few of the collaborators, and I have been, so I have given here the name, Professor Rajnigan, Yuk Ching, Dr. Yogendra Singh. These are the students, Raja, Urvashi, Vikas, and Pramod, and I have shown their faces during my presentation. And BIREC and SCRB and DBT have largely funded for this project. With all this, I would like to say thank you to the organizer, and I am open to the questions.
Thank you, Professor Tandon, for sharing very interesting work. And again, I apologize for cutting you short. Participants can directly write to you the questions, and due to paucity of time, will not be able to take it. I really sincerely apologize from everyone here. I'm quickly call upon last speaker of the session, Dr. Richa Mishra. And Richa, I really request you to keep it short. Dr. Mishra is Assistant Professor at Department of Zoology, Shri Venkateshwara College, University of Delhi, India. After completing her graduation and post-graduation from the University of Delhi, she joined PhD at CSIR Institute of Genomics and Integrative Biology. Her current research focus involves studying different aspects of gut microbiome, dysbiosis in tuberculosis, and working on effective alternative of TB diagnosis by stool PCR and TB patients. She was recently interviewed for her work on extrapulmonary tuberculosis for a Nature Medicine report. She has 23 publications and 3 book chapters to her credit, and serves as a reviewer for many reputed journals such as Scientific Reports, PLOS One, Peer J, among others. Today, she will speak on understanding the gut-lung axis in tuberculosis and its implication. Welcome, Richa, and very sorry, but please keep it short.
Thank you so much, Aru ma'am, and I'll try to keep it as short as possible. So I'll just jump straight. Okay, so a very good evening to all of you. I mean, through many wonderful lectures, the background has already been set, and we all know the vastness and the potential of harnessing the knowledge of microbiome. So I'm going to add a little bit about my work on the connection of microbiome with tuberculosis. So as we all know now that apart from environmental surfaces, microbes are present over our body surfaces, especially concentrated in gut. And over the last few years, the focus has been to delve deeper into not only microbial diversity, but also looking at the diversity of their genetic content, because that is vast. The vastness adds manyfold because of that. So we know that there are many factors which influence microbiome, and based on that, its implication has been already highlighted in many such studies, many such diseases. And during all these studies, two unobvious but prominent interorgan axes have been fervently explored, one being gut-brain axis, and the second, which I'll be focusing on, is the gut-lung axis. Now, when we started our work, already some connection was obviously known about the gut-lung axis, but the focus was mainly on the respiratory disorders like COPD and asthma, whereas we got interested in studying its role in the infectious respiratory disorder like tuberculosis. Now, we know that India leads the TB burden in the susceptible as well as the multi-drug resistant TB milieu. And apart from that, there has been a consistent trend in the increase, or there has been a consistent rise in the extrapulmonary TB cases, which is infection of MTB at sites other than the lungs. So, so the plan was that we initially recruited TB patients, and we recruited as controls the healthy household contacts of the same TB patients in order to negate the diversity which can be accounted for due to genetic changes as well as the dietary preferences. So, so that's why healthy household contacts were taken as controls. And the same TB patient was also evaluated for up to a month for the after the treatment starts. So most of the all the patients were the sampled before beginning of the ATT regimen, which is the 6-month long DOTS regimen, which is given for all TB patients. And we also sampled them for because we wanted to see the diversity which gets perturbed due to the multi-drug regimen, which is administered into the DOTS program. So all these samples were simultaneously observed by 16S ribosomal gene sequencing to look at the microbial diversity, as well as whole genome sequencing to look at the functional aspect. This was a collaborative project between 3 labs: Professor Yogendra Singh's lab, Professor Ruplal's lab, and also Professor Sharma's lab in IISER, and the results of which is published already in EMI a few years back. And although, because of the paucity of the samples we had at that time, we did not see any demarcated taxonomic variations looking at the 16S ribosomal data, but we observed demarcated differences of certain microbes in through our whole genome sequencing data. So this taxonomic variation highlighted 2 important points. One was the baseline microbial composition in Indian population of healthy individuals consisted mostly of Previtola species, whereas surprisingly there was an increased abundance of butyrate and propionate producer, which in short we can call SCFA producers, in the TB patients. Apart from this, at that time, it was quite a revelation, because most of the studies at that time were done in different geographical locations, mainly targeting at the Western population. So where Previtola was said to increase in many of the disease conditions as well, especially in oral microbiome. But surprisingly, the core community in Indian population is consisting of the Previtola family. This was also corroborated by other studies which were done later on a much more larger meta-analysis scale across different geographical population. And there also the core population of Previtola was prominent in Indian population. This taxonomic variation was reflected again in the functional analysis, and we saw reduced biosynthesis of important pathways like vitamin and amino acid metabolism in TB patients. And this compromise in turn was enhanced by enriched metabolism of butyrate and propionate in the TB subjects, which initially sounded like a very different result, because most of the studies done till then had always highlighted the good beneficial role of SCFAs. But when we delved deeper into the literature and saw the diverse role of SCFAs in various pathways, we started connecting the dots. So what might be good for a Western population as a baseline may not be so good. So basically, balance is the key. Abundance, increased abundance might have differential role in different disease pathologies. Now, this was also obviously corroborated by other studies where after our study and along with our studies, few reports were published, and a major cue we got by looking at an extensive epidemiological data analysis, which was published in NEJM, where they had looked at the association of risk of death from respiratory infections in almost 2 lakh Indians and Bangladeshis. And it was seen that low BMI confers increased risk, whereas high BMI did not have any such observation, which goes on to say that increased abundance of those SCFAs might be detrimental by affecting various pathways for infectious respiratory disorders, whereas it might have a completely different beneficial role in a disease pathology like obesity. So I'm skipping the other corroborative results which were got after our report was published in different other studies. And in summary, we can say now that there has been substantial evidence of the dysbiosis of microbiome in TB patients, as well as the additional perturbance due to the long DOTS regimen that is given to all TB patients. And we can say that this metabolic activity, this change in metabolic activity at the microbiota level becomes an important risk factor for development of active TB. Then another lead we got from our whole genome genomic analysis was the increased relative abundance of MTBC organisms per se, like Mycobacterium tuberculosis, in stool samples of TB patients, which was initially a big surprise, because obviously we know at the diverse level of microbes present in stool of any individual. So, so we took that as a clue, and I thought that why not look at it from different perspective as a potential diagnostic sample tool. Now, the general perception is that the diagnosis of TB is known and the treatment is very much standardized. But I'm sorry to add that it is not the same. Whereas the diagnosis of smear positive cases is fairly simple by FB microscopy. But as you can see, there are diverse diagnostic algorithms one has to employ, the clinician has to employ for detection of paucibacillary bacteria. Paucibacillary diseases include extrapulmonary cases, which I mentioned, which are now increasing more and more in India as well as other parts of the world. So to look at the potential of stool as a potential alternate sample for diagnosis, we recruited for testing TB patients, pulmonary, extrapulmonary, diversity of extrapulmonary patients, as well as we took into account false positive cases, and so we recruited some non-respiratory patients, non-infectious respiratory disease patients like COPD and asthma patients, as well as obviously healthy individuals. This stool was tested by 2 methods. One, which was done for all the subjects recruited for the study, was through the insertion element based PCR, because it is present in multi-copy numbers. It is easier to detect by PCR. Also, we tested this with stool. We also tested stool in the GeneXpert system. Now, GeneXpert system has been a game changer in the diagnosis of possible bacillary bacteria, because it's molecular biology, so it's DNA based. But the important thing was the procurement of sample is extremely difficult in extrapulmonary cases as well as the smear negative cases. So we were actually quite encouraged and happily. So obviously we were quite happy when we got very encouraging results with both the techniques. Since we could not at that time, and as you can see in the slide, normally stool is not recommended as a sample to be tested in GeneXpert. So it was very difficult to initially convince the clinicians to allow us to check stool. But since it is non-invasive, it does not, people usually, it does not require any invasion. So, so ethical approval was much more easier, and we could test it for a small subset. And to our surprise, we got good results with stool expert as well. But stool PCR definitely was much more accurate in terms of because it was tested on a large number of samples with diverse cases, looking at also false positives. And now, although expert is not expert does not include stool as a sample, but we see in practical settings, doctors are using stool to check for pulmonary TB in children or pediatric TB patients. So this result, this study was also published in scientific reports, and so I'm just focusing on the conclusions. So the major encouraging result that we got was the high efficacy of stool as a specimen. Then 100% specificity in expert MTB, which is definitely an encouraging result, because normally stool is not preferred looking at the diversity of microbes it has. But it for all the cases, we saw very good specificity and definitely good efficacy as well. So sensitivity was also there. And also the potential to detect even EPTB cases. So stool looks like a good alternate specimen which can detect quickly pulmonary and extrapulmonary cases. Also, obviously, we need more such studies on a larger population to look at its potential in a much more serious manner. So obviously the next steps are, so I'm skipping all this part. So the next part is that we are following it up and also looking at the metabolic changes, which are more practical to evaluate and like study intervention methods. And all this work obviously would not have been possible without the encouragement of my PhD guide, Professor Yogendra Singh, who had the faith in me to allow me to work on any of my crazy ideas at that time. And obviously, the 3 PhD students who now I kind of mentor and who do all the hard work of going to hospitals and getting all the samples from the TB patients, as well as the healthy individuals. So at the end, I would like to thank all the organizers and the organizing committee for providing me this platform to talk about my work and my journey into studying the role of microbiome in tuberculosis. And
thank you so much, Dr. Mishra, for the nice presentation. And I'm really extremely sorry for cutting you short. And thank you for considering a request also. I thank the chairpersons, co-chairs, and all the respected speakers for this fruitful session. This brings us to the end of the session and the conference, and I'll call upon Dr. Jaswinder for initiating the proceedings of the valedictory function. Thank you. Good evening, everyone. I'm pleased to start the valedictory ceremony on behalf of the organizing committee to the 61st Annual International Conference of Association of Microbiologists of India and 5th Annual Conference of Indian Network for Soil Contamination Research, jointly organized by University of Delhi, DU, Indian Agricultural Research Institute, IARI, the Energy and Resources Institute, TERI, and the Indian National Science Academy, INSA. The conference adopted a very well-timed theme this year as well: Microbial World, Recent Developments in Health, Agriculture, and Environmental Sciences.
who else could have been a better and a more appropriate speaker at the valedictory session than Professor Randeep Guleria himself? Your presence, sir, is indeed a source of great motivation for all of us. We look forward to your valedictory address today, and once again, a warm welcome to you, sir.
Thank you very much. Good evening. The guest of honor, Professor Victor Dirita, the other dignitaries, Professor R.S. Kurat, Professor D.K. Singh, Professor Praveen Rishi, Dr. V.C. Kalra, and of course, the organizers who have done a huge work, Professor Roop Lal and Professor Yogendra Singh, and Professor Namita Singh, the General Secretary. At the onset, let me really congratulate you for a wonderful conference. I think you've had a very enriching experience over the last few days, and the 61st Annual International Conference of the Association of Microbiologists of India, which is in collaboration with the Indian Network for Soil Contamination Research and with TERI and with the Indian National Science Academy, I think has been a huge success. The quality of papers have been really of great use. And the theme itself, microbial world, recent development in health, agriculture, and environmental science, I think is very, very topical. What COVID-19 has shown us in the last 1 year, that we really need to look at microbes in a very holistic manner, whether it be agriculture, whether it be plants, whether it be the animals or humans, and correlate that with the environment. Because we have not done that, and we have not looked at microbes in a holistic manner, a lot of opportunistic infections, if I put it that way, have started occurring in humans. And this is happening not only as far as antimicrobial resistance is concerned, but new viral infections, new infections which were not being previously seen are currently being seen in humans because of the interaction which are occurring in different parts of what is One Health. And therefore, this is something which is more and more important. Also, I think it's important for us to understand that as we look at the research that is being done and the papers that were presented, how can we really move them to the next level? How can we really look at what is of translational use? I would really urge people to look at research not only from the point of view of a good publication in a high impact factor paper, but how can it be useful for the common man? What research can we do which can subsequently lead to something which can make our researchers entrepreneurs, make them into doing something for society, which is the way forward? There has been, for example, there has been a lot of interest now in phage therapy. It's felt that bacteriophages can actually play a huge role as far as taking care of resistant bugs or bacteria are concerned. And there is a huge area of research that can be done as far as developing phage therapy in our country is concerned. One could take the lead as far as this is concerned. There's also a huge interest in looking at the microbiome, not only the gut, but in various other parts of the body, and see how they interact and how do they influence the body and the disease relationship. One interesting factor is what is the cause of the decline in cases of COVID-19 in our country as compared to the West. We know that from the data which was just released yesterday, we have not achieved herd immunity. The national seroprevalence is only around 21%. So only 1 in 5 individual has antibodies or immunity. The remaining people are still susceptible, yet a number of cases are low. So there has to be some sort of a host response, which is also driving the infection rate as far as humans are concerned. And could it be related to not only our immune system, but the microbiome which we have? Has that really given us, our immune system, ways to fight the virus more than what it has done in the West? We already have the so-called hygiene hypothesis, which really puts forward the concept that because of the immune reaction that we have in the developing world, we see very few autoimmune diseases. We see fewer allergic diseases than is seen in the West. Is this immune system also protecting us from emerging infections? So a lot of interesting areas and questions that are there. And I would really urge the researchers to look at these in the form of translational utility. The papers that were presented, they were really very diverse. And I think the delegates must have had a great feast as far as knowledge is concerned. I must also congratulate all the faculty that were part of this conference, because I think they did a stellar job in the presentations that they made. So in the end, I would really like to congratulate Professor Ruplal and Professor Yogendra Singh for actually having this conference in this trying times and making it such a huge success. So congratulations to you all, and thank you very much.
Thank you very much, sir, for such warm words. I would like to extend a very warm welcome to our dignitaries as well, the chairman of FAMC, Professor Ramesh Chandra Kuhar, Professor D.K. Singh, President AMI 2122, Dr. Praveen Rishi, President-elect AMI, and Dr. V.C. Kalia, Editor-in-Chief, INJM. Welcome your good selves. It is rightly said that, tell me and I forget, teach me and I remember, involve me and I learn. So it is on occasions like these that we learn, we get opportunities and look forward to getting a scientific exposure and interacting with unique and talented mix of participants from almost every corner of the country and abroad. Summarizing the aim and intent of this year's conference once more, the event targeted the recent development in the microbial world. The horizon of the event was extended by inviting students from different schools across India to propagate microbiology and raise awareness at the grassroots level. Raising the awareness and microbiology literacy is important, and the conference successfully achieved that, with students and faculty joining us from schools to higher education. The themes picked up an applied science dimension with throwing light upon the role of microbiology everywhere in our lives. The various themes covered the role of microbiology through topics ranging from aid in agriculture to One Health, medicine, and maintenance of the environment, as well as the innovations in biotechnology industry. Science and everyday life cannot be separated. We saw how microbes work at the level of the community, that is, ecology and epidemiology, at the level of the cell, that is, cell biology and physiology, and at the level of proteins and genes, that is, molecular biology and bioinformatics. To learn about recent advancements, we were fortunate enough to bring renowned scientists from across the globe on the same platform to discuss microbial advancements and to provide potential solutions to the current challenges related to health, agriculture, and the environment. It is critical that the current youth be aware and literate of microbes. This is essential for both individual well-being and the society. Moving on, for the welcome address, I would like to request Professor Ruplal and Professor Yogendra Singh, Organizing Secretaries, AMI-Inscr 2021, to welcome the participants and say a few words about the conference. Professor Lal.
Professor Randeep Guleria, Professor Archie Kohar, Yogendra, Sunil, Namita, all past presidents and participants and members of AMI and INSCA, and also Dr. Dikshit and Rajender and Professor Agarwal, who are very closely associated with the INSCA. I'll not take much of your time, but I feel much relieved today. 6 months before, when we decided to take up this responsibility, we never thought that we will be able to do this job so well during this COVID-19 pandemic. But the association of many of my colleagues, the help that I received from AMI, INSCR, TARI, Delhi University, IARI, and from INSA, from many of my colleagues, made me to achieve the goal that we set 6 months before. Not only this, over the last 6 months, but I had not done anything. Let me tell you very frankly. In fact, it was the inspiration from AMI, from Archiquar, from our president, Bai Singh, and many colleagues who really helped me. And along with this, there was a team of more than 30 to 40 young students who did PhD with me, but later, now they became my colleagues. I'm almost every minute I'm getting a message, the wonderful job my young team has done. So it's a really very thrilling experience. Probably with this, the organization of this conference and the pleasure that I'm driving out of the work my young colleagues have done, शायद इससे मेरे और 10 साल जिंदगी के और बढ़ गए हैं. मुझे इससे ज्यादा आप लोगों से कुछ नहीं चाहिए था. So that's what actually, and not only that, I just wrote simple mails to many renowned scientists, not only from India, but from abroad. Dr. Guleria, I know despite his busy schedule, I never thought that he will spare so much time for the inauguration as well as for the valedictory function. Thank you very much, sir. And I also got mails from many of the great scientists, renowned scientists, who has just written, how did you get all these great scientists who are doing such wonderful things in microbial research, especially on environmental microbiology and on microbiome. So it was their passion and association, and also probably my good luck that all of them, they came under this particular platform and it did the job so wonderfully that really I'm feeling very happy today. And as I mentioned, this was a very unique conference in the sense that we had very unique sessions, and probably Professor Guleria and Professor Kohal and all other scientists or participants will agree with me that through this conference, we ignited the society and the children. We told them the importance of microbiology in their daily routine life to keep good health. And not only this, even we also conveyed this message to our doctor fraternity that you need to understand the role of gut microbiome, as Professor Guleria mentioned, microbiome on skin in oral cavity, in order to provide better health and better treatment. I'll not get into details. Probably my colleagues, they will let you know what all happened during these 5 to 6 days. So with these words, again, I want to actually mention once again the important role and my young colleagues, they played in organization of this particular conference. The experience they gained, not only they helped me, but the experience they gained probably will make them better persons in the society and take up bigger challenges and bigger jobs. But I know actually, because this was a new experience conducting such a big conference online, and it was our first experience, everything moved so smoothly. Nothing happened. Speakers who came, actually, they were so happy with the organization, and immediately they were on the screen. So it went so well. I'm not really able to, I mean, even now I'm not able to believe that all this has happened despite all these limitations. But if there had been glitches or any mistakes, we actually take up full responsibility. And once again, I thank you, Professor Guleria. I thank all AMI members and my colleagues, including Archie Kuhar, Yogendra Singh, Sunil, Namita, and Dr. V.C. Kalia, who were always with us to take up this bigger challenge. And today, as I mentioned, I'm much relieved. Thank you very much.
Thank you so much, sir. Over to you, Professor Singh.
Yeah, thank you, Jaswinder. I welcome Professor Guleria and all the members of the AMI.
Sir, your voice is gone.
Yeah, So I again welcome Professor Guleria.
Sir, your voice is getting muted. Could you please check at your end?
Just a minute, there is some.
Okay, it's okay now.
So I said, I again welcome everybody who is attending this session. Finally, every event comes to end, and so do this e-conference. I am honored to share some quick thoughts with you about the past few days. To start with, I will say that I am overwhelmed with gratitude to each one of you who choose to be with us on this journey of facilitating this conference. In fact, words of gratitude won't suffice for our eminent speakers who took out their precious time and presented their talks. Microbiology literacy, as Dr. Guleria said, this remains one of the biggest problems in this country especially. Interest in microbes is growing nowadays after the recent pandemic. This kind of change needs to be
Sir, could you please check the voice again?
uncovering
go ahead.
more details. All of these and the inject issues have been thoroughly covered with our learned speakers. I am sure that the various sessions and talks have infused us with the greater interest towards the subject. It has indeed been a stepping stone towards a more inclusive society in terms of sustainable development goal. So we had these sessions for last 3 days and the pre-conference meeting for 2 days. However, I will say that these were not the last word. I'm sure that again next year and in future, every year, new things will come and AMI and INSCR will actually give knowledge to everybody who is working in this area. Finally, I will thank all the people, all those students, all the people who are helping to organize this conference, which is extremely successful, as Dr. Rupal mentioned. So thank you, everybody, for joining this. And now over to you, Jaswinder.
Thank you, sir. We are delighted to have with us Professor Namita Singh, General Secretary, AMI. Ma'am, you have been a member of the association and General Secretary, playing a very pivotal role, and hence your remarks from the association's point of view would be most pertinent today. A special warm welcome to you, ma'am. Over to you. Ma'am, we are not able to hear you. Could you please unmute yourself?
Is it okay? Am I audible?
It's okay now, ma'am. Thank you.
Thank you. So most respected chief guest of the day, Dr. Randeep Guleria, Director AIMS, New Delhi, and our guest of honor, Professor Victor Derrida from the Michigan State University, USA. We are honored to have all our distinguished dignitaries with us, Dr. Ajay Mathur, Director General TERI, Professor R.C. Kohar, Chairman, Academy of Microbiological Sciences, Professor P.C. Joshi, Pro Vice Chancellor, Delhi University, Professor Rakesh Bhatnagar, Professor Appa Rao Pundil, and Professor A.K. Singh, Director and Vice Chancellor of ICR, Indian Agriculture Institute, New Delhi. Dr. Vibha Dhawan, Distinguished Fellow from TERI, Dr. Banwari Lal from TERI, Professor Yogendra Singh, our President AMI, Professor D.K. Singh, President-elect 2021, Professor Rupal, President INSCR, and also the past President of AMI, Professor J.S. Virdi, past President AMI, and the President-elect 2022, Professor Praveen Rishi, and our all other past president Professor S.S. Dudeja, Professor T. Satyanarayan, Professor B.C. Kalia, and our Treasurer, Professor Sunil Pabbi, and other Center Council members, AMI. We have with us INSCR past president also, Professor Prahlad Seth, Dr. Rajendra Prasad, and Professor H.C. Agarwal, and Dr. A.K. Dixit, Treasurer from the INSCR. On behalf of the Association of Microbiologists of India, I would like to express my gratitude to all of you have come together here in a virtual platform from different countries and the cities of India. I really appreciate your cooperation, which has enabled us to conduct a highly fruitful conference in this pandemic era. I am sure that the talks delivered in this 5 days have infused us with greater interest toward the subject. It is essential that scientists and students set themselves ready for this new era of micro. We will be listening to many eminent speakers delivering in the different area of microbiological research. Main issues raised during this or addressed during this conference, I just listed some of them to share with you. The international environmental problem in response to man-made disaster and how we can manage it with the help of microbes. The second address is the health issue in response to outbreak of infectious disease that cross international boundaries also, and the different application of microbes in the field of industry and the agriculture, and the global microbiome and its importance in the health, sustainable environment and agriculture field. All these issues raised by our speakers, and they explain very well to enlighten our knowledge in the changing era. As we know that disaster is unpredictable, the possibility of their occurrence in the Asian Pacific region is very high, and horribly tragedy is anticipated if and when they do occur in the nation. There is an urgent need for us as a microbiologist to cooperate across borders to contain the spread of such disasters, tragedy, and infection to absolute minimum possibility, to educate people about microbiology, what is it, how it will help in well-being of human, environment, and climatic health, without any difference in nationality, races, politics, religions, and gap in wealth. It means nothing when it comes to save human lives. This should be our basic philosophy as a microbiologist. This is clearly, you can say, spread it in the current 61st conference of AMI. In this conference, we picked the topic for yesterday and today of disaster and the status of profession. Each region picked their own, and obvious there was much in common within the region. It is my hope that basis on the virtual relationship established through this conference, in the future, we will be able to respond together to issue that we have in common and achieve our common objective to spread the knowledge of microbiology in the common society. I would like to express my sincere thanks and appreciation and gratitude on behalf of AMI to all the moderators, presenters, and participants for their very active and frank discussions, which I believe had made this conference very fruitful. I would especially thank to all the foreign speakers and participants who, despite their busy schedule and time difference, have joined us from their respective countries. Once again, I thanks to Professor Ruplal and his dedicated team for organizing such a wonderful conference. Thank you so much, sir, and thanks to all for your cooperation. I, with this word, I don't have any other word to say. A special thanks to Professor Lal. Over to Dr. Jaswinder for declaration of our results, AMI poster presentations. She will read out all the results. Every year we announce for the AMI poster presentation and a R.S. Rana Memorial Award. Over to Dr. Jaswinder, please.
Thank you so much, ma'am. We would proceed with the award ceremony shortly. Right now, we have with us Professor Ramesh Chandra Kuhar, Vice Chancellor of Central University of Haryana and Chairman of Academy of Microbiological Sciences, FAMSC. Professor Kuhar, please enlighten us with your perspective and experience. Professor Kuhar.
Thank you, Jaswinder, for giving an opportunity. They have highlighted the various programs which have been carried out by AMI. Gularia.
There's some technical issue, sir. Can you please rejoin?
Presidents of AMI have joined this program, and this could only be possible that having this really very, very delighted to welcome of the microbiological sciences in our country, Johari. I have been getting messages from Professor B.N. Johari and others that quality and high standard seminars or labs have been organized by this time. I congratulate them. The microbiological sciences in our country has really reached to a stage, as Professor Guleria also mentioned, that now this is the era and we need to propagate further the microbiology and its prospects, so that our youngsters can take up this as a career. This conference is a unique kind of conference. Not only that it has been organized online, but if you see, the emphasis has been given majorly to the environment, agriculture, and the health sciences. These are very, very important areas. If you take environmental aspects itself, that covers everything. The human life, the plant life, and rest of the environment as well. And the experts which have been selected, I think from each corner of the world, and the quality of lectures have been, which have really advantages to all our students, specifically the youngsters, and of course for the seniors like we also enjoyed, we tried our best to listen whenever we got time. This is an opportunity for all of us. And I think the way this webinar has been organized, this is a wonderful, opportunity for all of us. And in the history of microbiology, we never thought of that we will be organizing sometimes the whole program, the whole conference, 5-day conference online. I think this is a beginning, and now we are quite confident that certainly in the time to come, we can organize such seminars, such conferences. And I congratulate Professor Ruplal, Professor Yogender Singh, and the whole team of the young students, young researcher from TERI, as well as the students and the faculty from the Collaborating Institute, University of Delhi, IRI New Delhi. And now, of course, everybody is waiting for the various poster awards, which is a usual feature of AMI. And I congratulate the award winners as well. I also like to take liberty to congratulate all the AMI awardees, as well as, say, our fellow of the Academy of Microbiological Sciences. Once again, thank you very much for giving opportunity to say a few words among you. Thank you very much, and I wish you all the best.
Thank you so much, sir. Let us now proceed to the winner announcement for various award categories. First, we will talk about the AMI awards. Just a moment, please. So under theme 1, microbiome and One Health, the award goes to Silvia Talavera, University of Madrid, Spain. Under theme 2, environmental and agricultural microbiology, the award goes to Varsha Tripathi, CSIR, Indian Institute of Toxicology Research. Under theme 3, industrial microbial biotechnology, the award goes to Alka Devi, Indian Agricultural Research Institute. Under theme 4, advances in microbial systematics, the award goes to Akshita A. Champaneria, Nirma University. Under theme 5, virus and vaccines, the award goes to Reena, Punjab University. Under theme 7, microbial pathogenesis and AMR, the award goes to Ritika Sahi, Punjab University. The AMI Dr. R.S. Rana Memorial Best Poster Award goes to Rakhi Dhankar from MDU Rohtak. For the AMI Oral Presentation Awards under Faculty Presentations, the award goes to Dr. Gaurav Sharma from DST Inspired Faculty Scientist IBAB Bangalore. And for the Students and Scholars Presentations, the award goes to Sachin Khurana and Harpreet Kaur. Now we come on to INSCR Awards, INSCR. The Best Poster INSCR Award goes to Sri Pratyusha Gandham, Punjab University. The INSCR Oral Presentation Awards, Faculty Presentations, the award goes to Dr. G. Yamal from Karodimal College. And under the Student Scholar Presentations, the award goes to Diya Roy. We have also conducted two awards under quizzes for microbial literacy for the school students and college students. For the school students, the award goes to Arya Kashyap and Bharti Kumari from DAV School, Kent area, Bihar. And second prize goes to Simranjeet, Guru Nanak Dev School, Punjabi Bagh, Delhi. The third prize goes to Priya Sinha, DAV School, Kent area, Bihar. We have also conducted awards for college students. It goes to Richa Hans from DRDO Gwalior and Muhammad Ashraf, Central University of Rajasthan. Congratulations and best wishes to all the winners. We would contact all the winner awardees on their given contact details for further proceedings. It is my pleasure to now welcome next Dr. Banwari Lal to give the concluding remarks. Sir.
Thank you, Jaspreet. I think now we have reached at the end. So every good thing has to come at the end. So in this 5-day journey, we have gained a lot of knowledge and we have more confidence. I think this is a really a good experience, a learning experience for all of us, that how we can do a virtual conference during pandemic time. If we would have done this conference, probably it would have really become difficult to manage 1,000 people, but it has become so great and we were able to get the good speakers globally. So I think it's a very good learning for all of us for future AMI conferences also. So I think the best thing is that in this all 100% participation, those who has given their list for talk, everybody has presented. There was not a single absentees. So in total 10 themes, total 164 talks were given. And all these talks, the quality of talks were really very, very high standard. So in 10 themes, the first theme was microbiome and One Health. So in these 2 inaugural lectures were given, as Dr. Guleria has given a very high quality lecture about antibiotic AMR. And in agricultural and environmental and agriculture microbiology, so 3 keynote lectures were given, 4 plenary lectures and 13 lectures were given. In session 3, children, this was a special session, children-centric microbiology, in which 9 lectures were given. And 4th session was industrial microbiology, in which 5 keynote lectures were given and 7 lectures were given. In 5th session, Ami-Inscr Innovative Research, in which 23 papers were presented. In session 6th, virus and vaccines, in which 3 papers were presented and all papers were very high quality. Session 7th, on advance in microbial systematics, in which 1 keynote address was given, 1 plenary lecture were given and 8 papers were presented. In 8th session, Ami-Inscr Innovative Research, in which 1 plenary lecture and 62 papers were presented. In 9th, translating and innovative ideas to enterprise, in which 7 papers were presented. And in the last session, microbial pathogenesis and AMR, in which 5 papers were presented. And 10 AMI awardee lectures were presented. So total 164 lectures were given in this conference, which was a great 100% participation. So I think this is a really very good learning for all of us. And as Professor Ruplal, our mentor, he told that today we feel so confident and very happy that this conference we have conducted successfully without any problem, without even any technical glitch in the virtual mode. So I think it's a really good and in future also, I think we should learn the lesson that how we can arrange such mega conferences virtually. I would like to thanks once again for all of you. And let's hope next year we will have a similar good conference. Thank you.
Thank you so much, sir. No duty is more urgent than that of returning thanks. I would now request Professor Sunil Pabbi to say a vote of thanks.
Thank you, Dr. Jaswinder. Of course, all the, I would say, speakers in this particular session have done the job of giving the thanks to all the organizers, but this is my formal duty to propose this vote of thanks. This is a culminating session of AMI 2020, the 61st Annual International Conference being held as a e-conference in association with INSCR. We are thankful to all the organizers, the Energy and Resource Institute, University of Delhi, Indian Agriculture Research Institute, and Indian National Science Academy for their wholehearted support in successful organization of this event. The AMI fraternity, along with INSCR, place on record a deep sense of appreciation and gratitude to organizing team led by Professor Ruplal for the wonderful organization of this e-conference, which was on one hand had an excellent blend of international and national speakers, and on the other hand was full of such rich and diverse academic flavor. The task of arranging this e-conference, which was as Professor Kohad also mentioned, it was a maiden experience, which was made easy by the untiring efforts of course, a lot has been said. I would say the Never Say No brigade of 30 odd young microbiologists who worked day and night, and we had nearly smooth and uninterrupted scientific discourse for nearly 5 days, including these pre-conferences and the workshops. And a big round of applause and thanks to all those young people who really took this on their shoulders. And they have really set a, I would say, very high benchmark for the future generations. I thank all the office bearers of AMI, IAMSC, INSCR. I'm not taking names because everyone has already said about their contribution, but we are very thankful for their continuous support and guidance for this mega event. I also thank all the speakers who gave their valuable time and input, both international and national speakers, and accepted our invitation to be part of this e-conference. I am thankful to all the faculty, the students who presented wonderful posters during this scientific event. Thanks are also due to all the young school students and the authorities who participated in our special session on child-centric microbiology, which was a great success and appreciated by one and all. At the end, I thank one and all who are directly or indirectly associated with this particular conference and helped in its successful organization. Jai Hind.
Thank you so much, sir. Indeed, this conference was a great success and an informative one. Thank you all for being with us on this journey of knowing the world of microbes. It means a lot to us to have all the speakers, the team members, and our wonderful audience with us. This conference was a basket full of knowledge shared by our eminent speakers, and their perseverance in answering so many queries was truly heartwarming. Thank you so much, everyone. This conference wouldn't have been possible without all of you. Before signing off, I would like to mention here that it is imperative for all of you to fill out the feedback forms. The certificates would be received by you in the coming 15 days. We would appreciate your patience. To capture this moment, I would request everyone to switch on their cameras.
Sir, I need permission from you.
Please, I request the host to give permissions.
To leave.
I hope my friends have captured this moment. Closing off, I would reiterate, science and everyday life cannot be and should not be separated. I would end the program with a quote by Aristotle: "We are what we repeatedly do. Excellence then is not an act, but a habit." Signing off, fare you well.
Thank you all.
Thank you and congratulations.
Thank you all of you. See you next year.